platform. People who do not want to do too much of coding, especially for biologists and chemists and science people. Of course, we don't want to get into more of Python. So there 9 can be used. It's a plug and play module. So I am sure that will be useful for you also. Any other questions?
Thank you so much, sir. Can you share your PPT as well?
I will be sharing the links in the chat after my presentation so that all the slides are there.
Thank you so much, sir.
So any others?
Hello, sir.
Yes, sir. Please. Yes, sir.
Sir, this is Professor Harishchandra Patil, Communist College, Pune.
Okay.
Sir, I would like to just ask about the computational techniques or the feasibility for the crores of the fingerprints which are there in the India, maybe for the ATM banks in future. So, what computational techniques will be playing a vital role in such a big data analysis?
Sir, your voice was very feeble. Can you repeat it again?
Hello?
Slowly?
Can you hear right now?
I can hear, but it's a little bit feeble. Yeah.
Hello?
Yes, sir.
Now?
Yeah, better.
Can you hear?
Yes.
Yes, sir. I would like to just your views on explore about the data analysis techniques for the crores of the fingerprints of the Indian people for the ATM kind of application. There we will have a biometric system to the ATM. What do you think? What do you think? Which computational techniques will play the vital role for us?
I'm so sorry, sir. Can
Hello?
you type it in the chat? Can you type it in the chat?
Okay, sir. I'll ask later on. No problem.
Yeah, thank you so much. Because there's so much of background noise, I'm not able to recollect the words. Sorry.
Sir, yes, sir.
Yeah. Anything else before we start?
Yes, sir. Good morning.
Good morning.
I am Dr. Senthil Kumar, heading a plant science department of St. Joseph's College, Trishrapalli, which is in Tamil Nadu state.
Yes, I have visited your institution. Yes.
Yeah. I have few clarifications regarding this point. Because in the biological science, School of Biological Science of our college, there are many students are working with the phytochemistry part. Even the chemistry students and even the physics also now they are working. The computational group also they are working with the annotation and all other part. See, now the scholars, once they identified the compound or isolated the compound either from the plant source, they go directly to the publication.
Yes.
Then later they think about the patent. Then later they think about the technology transfer to the industry. So what's actually the sequence, whether should go to the connect with the industry or then patent or patent with industry, then publication. So what is the lacuna? Because not only this issues which is in our college, but most of the institutions.
Very true. Very true.
Kindly just to flash a few things.
Absolutely, sir. Absolutely. Thank you so much. The reason here is being it's all only faculty community here. I want to review, and everybody knows that most of the time we have to publish, maybe even without quality, just because of survival, or just because we need to do it, or just because of points or promotions. Let it be anything. I'm not generalizing it, but generally I'm telling it. But when we want to do quality publication, it takes time. We cannot publish 30 papers in a year. It's difficult for us. We need, for a productive and quality research publication, we require time because we need to do several analysis. Now that time we might think, oh, I cannot do it alone. Of course you cannot. That's where you have to collaborate. You have to collaborate with the right people. So it's not always me, or I. It should be we in research, basically. Now, as Dr. Senthil Kumar rightly mentioned, when you extract, characterize, and publish, that doesn't have a lot of potential there. The first thing you have to ask questions, like I am starting my slides. I'm just asking first what you require, what you expect. The same way, you should ask companies like nutraceuticals or pharma companies, what is your expectation? I'm coming up with a molecule. I'm planning to patent. What if you have what all information will you purchase or technology license my patent? So try to understand what is the requirement first and then work towards that. And one more point I'll tell and then we will start. Another point is that we have to identify what are the possible targets for the extracts that you have done, because that is called systems pharmacology or network pharmacology, because it is a compound to disease target approach. That's where we use a cytoscape and all these tools to understand how far these compounds are already relevant to existing compounds to find out possible targets. Generally, phytochemicals, we always look at either anti-cancer, antimicrobial, antifungal. This is what majority papers talks about. But it is not that way. Many compounds have different other activities also. So we have to do a proper analysis. That is again machine learning based analysis. For you, you won't need to do any machine learning. It's already being done. And I will discuss a little bit touch base on that also, because I can understand many of you are interested in that. So I will also touch base that. So thank you so much for those really interesting. Over to you, Gaurav.
Thank you so much, sir. Let's begin with the session. So
please.
We are welcome to Dr. Pillai, sir. Dr. Pillai is one of the coordinator for Drug Discovery Hackathon 2020 by MHRD, researcher at Nairo Research India, director and founder of Zastra Innovation Bangalore, and formerly a Marie Curie Research Fellow at Malcode Limited, Estonia. His expertise in the area of machine learning, molecular modeling, software pipeline development. Currently, he has PhD student working under him in the area of aging and biomarkers. Apart from the scientific position, he is a member in academic councils of few academia, review to ACS, Willy and Elsevier Journal, also a mentor at IIT Kottayam, Government of India, Niti Aayog, ATL school and research advisor. He carried out his PhD at University of Florida with, and he holds 40 research papers in peer-reviewed international journal, 1 US patent, and co-authored a book chapter on mosquito repellent. So I welcome and inviting Professor Dr. Pillai sir for their scientific session.
Thank you so much,
sir. Yes.
Thank you. I'm just sharing my screen. Are you able to see my screen?
Yes, sir. Yes.
Oh, great. Yeah, all faculties who are joined here, feel free to put your responses in the chat. I'll be asking questions, a lot of questions. So, let us make this more interactive session rather than me just simply talking and going. So throughout my session, if I ask you a question, if the answer is yes, you can type number 1 in the chat. If the answer is no, you can type number 2 in the chat. So throughout my session, any questions that is coming out from me, 1 is for yes and 2 is for no. And there are a few other questions that might come up. You can unmute and discuss. And don't wait for the last moment for question and answers. Feel free. And I'm sure we have to interact a lot better than students. Students feel shy and students do not speak thinking that they are not confident with the questions. But here we are to learn each other, I have to also learn. So it's better that we interact a lot. I'll have my chat window also in my presentation window. So absolutely fine if you send the chat messages, I'll be able to see that. Anyway, without much ado, because we have to finish it before 10 also, 10 o'clock. So role of ADME and toxicity in drug candidate selection in AI approach. I didn't mention here for a drug discovery area, because I cannot predict a drug, or I cannot come up with a drug. I can come up with a drug candidate only. A possible, it could be called as a drug later based on some other analysis. But initially we can look at what is the affinity, what is the ADME properties and toxicity. The sad part here, in most of our paper publications or research, I could see majority of them. I could see ADME and toxicity is just used for predicting some values, but we never made any analysis based on that. Because ADME toxicity is something, the next factor that to be considered more important in order to call a chemical compound to be a lead molecule. Unless a lead molecule, then only it goes to a drug molecule. So when we are talking about AI or ML, machine learning in drug discovery, it is not yet completely ready. So don't worry that if you or me don't know about the machine learning or AI, because it takes time. There is a big hype there. because that is the era we live, because every time we have different hype. But then it changes as the things goes ahead. So it takes time, like Human Genome Project, but slowly make yourself understanding about the technologies. And ML means, don't think that it is programming or Python. No, never, ever. ML means it is statistics for me completely. Until unless you understand statistics, people cannot do ML. That's the beauty of it. So make sure that your good expertise in statistics, because it's a modern or a modified version of statistics as ML, which I'll come to go through my slides. And my slides might contain pictures and others taken from other web articles and lectures and tutorials. Why I specifically mentioned this sentence? Because we are all faculty teaching others. We used to write. Make sure that whatever pictures that we taken from web or other articles, they're cited properly. If it is yours, you claim it is yours. If it is others, just mention it as others, because there is no wrong in using others' data if you could cite them. And that is very important, and it is very much ethical. We have to teach our students also, because they also do not know the importance of this. Because nowadays web internet is completely catching up the plagiarism automatically. No need to find and say this is copy. No, it's automatic. So better let students know earlier also. And when students go for higher studies, they have to write several documentations like statement of purpose, research statement, and many other. So if they learn from the day one about plagiarism, copyrights, ethics in publishing or articles, it would be nice. Most of the time we give about IPR and all those at the last semester. It's always to give at least a 40 minutes of lecture in the beginning of their curriculum, just to understand what is ethics about, not to copy from others, or when you're adapting, how to cite them. These are very important things. Now, the most biggest challenge that we can see is a gap between academia and industry. Even though we talk a lot, we bring in a lot of sessions, we give a lot of talks, but in action, very few things are happening in action. Even though in action, if it is happening, also it is either training or either some workshop, that's all it is ending up. We want to come up with a collaborative project which can come into as a product. That is what I am trying to tell it as a bridging the gap. Now, consultancy projects might come to some faculties, but we don't know what happened next, or they might not come back to you later. Why? What is the reason? So all these things has to be analyzed. Then only a relationship between industry and academia will be strong. And that is very much important for industry, for a relationship with academia. But unfortunately, we are not yet there. Of course, some are doing really great, some are not. So it's as a faculty, it is our duty to come up with these, because this is not good for the management or the institution or even for the school or for the students. It's mainly you get a better exposure, the faculty who is involved in that interaction. You might get a better patent, better remuneration for the research innovation that you have done with the industrial collaboration. So it's basically mainly that you can get benefited. So you have to get a take with the initiation. Okay. So first we will look at what is machine learning and what is there in machine learning in drug discovery, as well as what to do next. When we look at machine learning, the word learning itself denotes the changes in a system that enables a system to do the same task more efficiently the next time. The same way we learn, right? Or the students learn. They learn, we put a test. If they fail, we give them some suggestions to improve. And next time when we put a test or exam, they improve their marks. So every time we are enabling a system, or we giving an environment to the system to do the same task more efficiently the next time when they redo it. But for this, similar to human beings, we have to understand and improve efficiency. This is something similar to improve the methods for teaching and tutoring people. Discover new things or structure that were previously unknown to humans. That is called data mining and scientific discovery. One madam was talking about big data. That is something that helps a human to go for unearthing or looking into unknown data. Fill in incomplete or skeletal specification. The biggest challenge in biological domain or drug discovery is that We have sparse data. We don't have much data. Even though we have much data, they are not reliable. The reason is most of the experiments are carried out only once. What is the rule? Because in our +2 or pre-degree itself, we learned by doing titration 3 readings. At least 3 readings were done or a titration reading. Why? Because we were able to calculate the median and also the standard deviation or the error value. Then only we will have a significance on the result that has come out. To generalize in layman terms, in mathematics or in computer science, 1 + 1 is always 2 only. We don't have any other answer for that particular calculation. But in science or in biology or physics or relatively drug discovery, 1 + 1 has 3 different answers. One is 1.9 plus or minus 0.1, 2.0 plus or minus 0.1, 2.1 plus or minus 0.1. All 3 answers are acceptable, even beyond that also. Because that plus or minus 0.1 tells you about the error and the deviation that we can accept because of the variations in the experimental analysis or human error. It could be anything. But if you're saying in biological results also it is 2.0, it is unreliable because there should be an error value because you have to repeat or replicate the experiments. It's very, very important. Now, if you blame that you don't get enough funding for that, so ask for more funding, let them give the funding for you to repeat the experiments. So this is what we have to look at for the next upcoming era. So here, when we are specifying the learning system, you have to specify what task to be done, how it should perform, what is the experience it has to learn. So the exact what knowledge is to be learned is to be looked at, and how this knowledge to be represented, and how this knowledge to be learned. So these are different stages of artificial intelligence, machine learning, and deep learning. I'm not going deeper into it because you might get all this information in many places. Machine learning is a branch of computer science which deals with system programming in order to automatically learn and improve with experience. Because of many advanced technologies, we need not to do this programming nowadays. Lot of plug and play, already programmed plugins are there. You can need to just plug and play. And rather, the important here is the system or the model has to self-learn. And we are also looking at the iterative process to produce a result, evaluate and tweak the system. That is what ultimately what we're looking at. And these are the few examples of different algorithms and different applications. When you look at the types of technique in machine learning, they're supervised, unsupervised, semi-supervised, reinforcement, transduction, and learning to learn. And when you look at algorithms, they are decision trees, neural network, probabilistic network, nearest neighbor, support vector machine. Don't worry, I'm not going to discuss about any of these things because of more of the communities with biology and science background. And moving forward, how to look at machine learning in drug discovery. This is one of the issues that came in 2018. Is machine learning overhyped? And when it was being taken a good amount of survey, they strongly agreed that it is more hyped. So that is a fact also. Machine learning is overhyped, especially in drug discovery and in science area. The main reason is we don't have enough data. Without enough data, machine learning doesn't work, but statistics much works better there. So you have to keep in mind that you have to also still start learning, like what we all learned about statistics. You have to learn machine learning also in your studies, because that is going to be very important for data analysis. Now, when you look at drug discovery companies, just a minute, let me hide this. Yeah. So when you look at drug discovery companies worldwide, there are many, many small and big and moderate companies who are working in drug discovery. It could be target identification, it could be disease analysis, it could be pharmacology, or it could be software related, or even it could be a small molecule design, including peptides. So there are multiple arenas where all these companies are looking forward to. In majority of them, they have come out from academia. Means the academic lab research work got converted into a company in many of them, right? So that's what we have to also look at. how we can convert our research into a possible commercial model. And if we are looking at the cost saving analysis, they are implementing machine learning and AI into their research in order to reduce the cost. And it's tremendously high. So they are utilizing computer aided drug discovery services in order to reduce the cost in pharma industry. And similarly, you can also look at the upcoming trends where artificial intelligence, deep learning, and machine learning is going to be used for a long term in order to look at the drug discovery process. So it is yet to come. So we are on the right time, right track to learn about it. Cloud-based technology platforms are also available. So recently I have published on the web about you can do molecular dynamics and certain extensive computational studies using Google Colab. So Google Colab is something cloud-based technology platform. And then you have Amazon AWS, then Google Cloud, then you have Microsoft Azure. And some Indian companies also like AICTE-ATAL and few others, they have their own cloud-based system within India also. So these can be now used because you can run your calculations, everything using a mobile onto their computers, we can submit the jobs. But when you look at the distribution of the CADD service, you could see here, there is very less people working on target identification and target validation. And this is a reason why I included this slide also. Target identification and target identification requires a lot of biological experts because chemists cannot do that. Chemist is mainly having a role in the drug optimization, but biologists have a major role in target identification and validation in order to understand which mechanism, which pathway, which receptor is upregulating and downregulating the mechanism, and whether it is going to be the reaction mechanism like opposite, and also whether it is going to this one agonist or antagonist like inhibition or activation. All those things are explored at target identification and target validation. So when you're doing anything on phytochemistry or phytochemical compounds or extractions, and most of us are missing to do this part, target identification and target validation. So make sure that you go through this so that your research work will be complete, because you're trying to understand reverse like reverse engineering, what we call it as, right? So we reverse look at how, based on the structures that is present in the plants, what could be the possible targets available already reported. And similarly, deep learning algorithms can be used to correlate between your compounds with respect to the biological activity or biological affinity, so that we will be able to produce or predict the affinity values for the new molecules which is not yet tested. So even for the plant extracts, if you don't know which could be the possible drug target, in that time you can use several models to predict the activity data to find out which could be having a better activity. But keep in mind, it is a prediction. Make sure that you consider the error value also. Now, the biggest drawback here is many of us know how to do machine learning or statistics. But the question here is, we also need to know about the analysis part. We need to know how to predict things. We need to also know how to come to interpretation of things. And that is very important. So when you look at this particular representation or this representation or this one, we need to know how we have to interpret the data, or like a dendrogram like this or a cluster like this. So we have to understand that. So result analysis is something very important for each of the methodology. And later, based upon COVID data as well as toxicity data, many people are doing different models. So keep in mind, different approaches of machine learning models can give you different accuracy levels. So don't think that one single approach is going to give you the best. In machine learning, you have to compare between multiple models. Which one is giving the best one, you have to take. Moving forward, we have some recent news, especially in April 2020 and September 2019. There are few companies claim that AI identifies a drug candidate and within weeks. Here they were able to identify this compound within 46 days. And also exploring the chemical space using AI. Chemical space means which contains around 17 billion compounds, small molecules. So how do we convert our chemical structure or drug molecule into machine learning understandable language? That's where we call it as fingerprints. So we convert our molecule to fingerprints so that this will be binaries, which is zeros and ones. So this is how we convert it. So we do have algorithms certainly, even to represent each of the region of the molecule to be of each individual fingerprints. Now, looking at an overview about all these machine learning aspects in the application of drug discovery, let us look into what are the actual challenges that we have in drug discovery. Now, it is a question time. One of the biggest challenge that we are facing is, apart from all these analysis and research and whatever we are doing, the biggest challenge a civilian is looking at is, how can I afford the cost of a medicine? As a researcher, maybe computer science or statistician will think about how can I get a better model? How can I better R square, or better accuracy? But in the case of biology, people, we always look at how can we get some positive results, or how can we get some reproducible results. That's what we look at, right? But a civilian, does he think about it? No. Civilian thinks about whether this medicine is going to cure my disease, can prolong, and how affordable it is to me. So now a question to everybody. Just think within you, okay? Don't tell about government. Don't tell about policies. No, that is not within our hands. If I tell that, it is not going to change tomorrow. Within your limits as a researcher, how can you reduce the cost of a medicine? Because if you're working on biology also, tomorrow it's going for drug discovery, or it is going to define a disease mechanism. So how you think that you can reduce a cost of medicine by your research contribution? Either you can unmute and tell me, or you can type in the chat. But please don't take time. Otherwise, we'll be wasting time. I have more slides to cover. Nobody?
I think, hi, this is Abhimanyu.
Yes.
I think one of the approach could be like finding the literature, and removing those targets which have already been investigated. So it's better like to remove those, and if it is really not working. And also the sharing of data is like it also plays a crucial role, I think. Like as a biologist, I'm not computer science.
I understand. I completely agree.
Yeah. Thanks.
Thank you. Thank you. Any other perspectives? If you're taking time to think, I can give you time. I'll continue with my presentation. I'll come back to this question later.
Hello, this is Dr. Arup. Am I audible?
Yes, sir. Please, very much.
Good morning. Good morning, sir.
Good morning.
The perspective of conducting the machine learning, as per my concern, is that first of the design of the molecule in the lab, and thereafter there must be a synergistic approach between the two domain of the science and a computer vision person along with the biologist, and as well as the deep thoughts of literature. Thank you.
Okay, great. Thank you so much, sir. Any others?
Yeah, I think one important cost for medicine, I suppose, will be the control trials and all that, where from computer science point of view, we can evolve better methodologies, which may cost less. I don't know whether we're asking only related to drug discovery or beyond that, but I think
it's generally within our capacity. That's only my
from a computer scientist point of view, I feel there is a lot of opportunity to have better techniques, better sampling, so that the overall cost can be reduced quite significantly. And even for infectious diseases, we can have much better surveillance methods to understand how the disease is actually spreading and all that. So that potentially is again underexploited as of now.
Very true. Very true. Thank you so much. Anyone else want to add something else? One more. an opportunity for women scientists and researchers here? All faculties are researchers too, right?
So good morning, sir.
Good morning.
Sir, I am Muthulakshmi from Kalsalingam University. Sir, for drug discovery initial stage itself, they are spending more time for screening of drug targets and everything. For screening itself, and we can combine together with computational and biology, that they can reduce that time as well as that cost also. So maybe it's a combined research work. It's good for reducing the cost of medicine.
Okay, that's also a good perspective. Thank you so much, madam. So moving forward. So thank you so much for all those answers and different perspectives. I'm sure everybody one else also would have thought of answers, but you're not opening up your mouth. Absolutely fine. But at least you thought. That is very important. So moving forward, I'll come to that back a bit later. Moving forward, the biggest reason for increasing the cost of medicine is because of failure. But the failure is happening due to the reasons what all you mentioned. Very true. Many of you pointed out the same point, collaboration. If you drug discovery is not a single man job. Every field of expertise has to come in, including computer science, including statistician, biologist, chemist, pharmacologist, or pharmaceutical chemist, toxicologist. Everyone comes into the picture, but we don't know where to fit it. We think that I'm doing my job, that's enough. I get a publication, enough. Why should I worry about the drug? Why should I worry about the molecule? That is not the case. As a faculty, I hope many of you will agree with me. For our sustenance in research, we have to work on a specific topic for more than 6-7 years. Then only we can call, or we can self-call ourselves that we are an expert in that domain. At least 6-7 years continuously we have to work on that particular domain. Sometimes people continue to work with a PhD topic. Sometimes they start with a new topic. In fact, I have started with a new topic after my PhD, completely new topic, and I'm establishing on that. For the past 4 years, I've been working on that, published few papers. Now we are working on a research product out of it. So it is of our interest. But what I wanted to tell you here is, in early between 1960s and 1980s, as one of the faculty clearly mentioned, we want to know existing data, analyze it, learn from that, and apply something new, where he mentioned about computational techniques. But I want to correct one only thing. If you have a confusion, don't think that you can replace your wet lab experiments with computational studies. Never, ever you can do that. But computational studies helps you to understand how you have to optimize things. It gives you some information about optimizing the parameters, but it will never replace your experimental data ever in future. Never. It cannot be possible, because computational are mostly predictable or with errors. So we have to consider that error value. And error is much more than experimental errors. So when considering the FDA compounds between 1960s and 1980s, we could see ADME properties was much more 40% reason why the drugs were being failing. And the second reason was under the phase 2, efficacy was poor efficacy of the compound was one of the reason of failures. So in this stage, I want to tell you that most of us would have been publishing papers based on activity data or bioassay data or concentration data. Did I mention anything about concentration, activity, or affinity here? No. So just finding out affinity or activity doesn't mean that you found a drug. There are many stages yet to come. So now, how did we overcome this? That's where computer science, machine learning, statistics was being implemented. So after 80s, people started generating a lot of statistical models and machine learning models for ADME and PK studies. And that we could see in the difference also. For the FDA approved drugs from 2000 to 2010, the cause of failure completely changed the factors. Since due to the implementation of statistical and machine learning models, A reason for ADME reduced from 40% to 4.9 or 5%. Right? And efficacy were also to get reduced because of this is only because of the implementation of machine learning and statistical model. In our terms, what we call it as QSPR or QSAR. But we had two other different challenges. Because of the computational implementation, we started putting in more junk data because we were able to explore more space of chemistry, more space of data. When we explored more data, we started generating unwanted structures, or we started exploring unpromising data. That's because of that 40% of the failure is just because of toxicity and strategy. And this is one of the biggest drawback with computational studies. Strategy is the biggest drawback. The reason I'll tell you, I might do from a workflow from A to B to C to D. But due to other computational availability of tools or methodology for other people, the other people might do the work plan from C to A to D to C. But the strategy is different there. Now, which strategy is good? It depends upon what is your objective. So that the kind of experience we need to achieve. What we try to do is how to best incorporate the existing knowledge. We look at a paper, replicate the same methodology, different molecule, different protein, different disease. That's all. But the problem, we are not realizing that that methodology worked for that paper or for that work, but may not be for your objective. It might differ. We are not exploring that. So that is the reason for a strategy failure. So here about 30% of the drug failures because of the poor strategy. Likewise, we also need to decide on what kind of methodology you have to choose. It's similar to what kind of machine learning methodology you have to choose for generating models. It's very important. If you do not have a specific data and you apply some methodology, it doesn't work. So this is something like we already have, for example, we already have an equipment with us or an instrument with us. We are just finding a way to how to make use of it. In order to tell it as a joke, you would have spent crores of money to buy an equipment in your research facility, but nobody's using it. After 1 year, the report came, you have not published any paper using this equipment. So publish some papers. In order to make use of that equipment, you will be writing some project. That's what I'm trying to say. It shouldn't be that way. We need to have a need from a research problem. Based on the research problem, utilize what we have or collaborate with others whom they have or what they have, and then come up with the result. And that is very much applicable in drug discovery also. Sometimes I have seen people say that I have this software, so what project can I do? That shouldn't be the approach. That is a very wrong approach. What data you have, based on that you choose a software, based on that you choose some method or approaches. That is very important. Now, how many of you understand what is a protein here? So the question is for 1 and 2. Yes means 1, no means 2. So how many of you understand if I say the word protein? Okay. Many of you understand that. Okay. Why I asked you just to whether I should go with the basics of proteins or not, but I could see unfortunately many are here attending, but only 6 of 7 has responded. Oh yeah, there is a no also. Okay. Okay. So I will tell a little bit basics. There are people who are saying no also. Okay, great. So proteins are something that is building our body, right? In a normal layman terms, when we explain it to a person who doesn't understand protein, it is a building blocks of amino acids. There are 20 amino acids, in fact 21, but 20 amino acids. So building blocks of all these amino acids which join hands together between carbon and nitrogen. These are elements or atoms, and then that's where a long chain forms. That long chain is what is that you see here. Oh, sorry. This is the long chain. So this is called a protein 3D structure. So these each one will contain lot of amino acids. So some protein might contain 300 amino acids of different among these 20 different combinations. And some might contain 1000 amino acids depending upon that. But a protein is a 3D structure which has a combination of multiple amino acids with different functions. So inside our body, we will be having several proteins which will be responsible for certain function. It's a simple function, I'll tell you. When you have, let's say, when you have fever, your temperature rises. That as a layman, I identify fever based upon temperature, right? Based on like thermometer, we try to understand. But internally, why the temperature increased? That is a question. Why your body temperature increased? That is because of many of the changes in the metabolic pathways, and our biological system is a bit disturbed. How this is disturbed? This is based upon different protein action functions, enzymatic functions, and changes in metabolic pathways. So these are the reasons for the change in the biological system, which increase the temperature in our body, which we again feel tired and many things. So protein is also one among, has its role there. And protein is also called as a target for several diseases. So if you have a specific disease, one specific protein is responsible for causing that disease. I'm not getting into deeper things, but for the people who said no, just an overview. Okay, that's enough. I'm not going beyond that. So how to get the structures of this protein? Because it's in our body, right? How do we get these structures? So biologists, molecular biologists, biotechnologists, they try to extract, characterize, and then they purify, and then they crystallize it, whatever crystallizable, not all of them are crystallizable. And then they put it in X-ray diffraction studies, that is called X-ray crystallography studies. And then they elucidate the structure, which can look like this. So that is what I called as 3D protein structure. If I have a X-ray crystal structure of the 3D proteins and the structure of the small molecule means it could be phytochemicals, it could be flavonoids, it could be synthetic molecules, it could be from a database, or it could be something that you designed yourself. These are all small molecules. Small molecules are not biomolecules, but basically chemical molecules or small peptides. So these are called as ligands. So when you have a 3D structure of the proteins and the ligands, we try to perform molecular docking. I'll tell you what is molecular docking a bit later. And then we try to perform molecular dynamics. That approach is called structure-based drug design because we have both the data available. Let's imagine that you have a 3D structure of the protein, but unfortunately, you tried all possible compounds, small molecules, nothing is interacting with this pocket or nothing is interacting with the protein. In that stage, we try to come up with a new approach called as fragment-based drug design. Now, you might think, sir, what is the point of me learning all these things? Because these are all well and widely used in pharma industry. And if you ask me, is there any success stories? I have more than 100 success stories to talk about drug discovery. If you want to know about the success stories of all these compounds in CADD, I have a podcast. I hope you know what is podcast. It's just an audio. You'll be able to listen to it maybe from 5 to 10 minutes of the data. So my podcast name is Drug Discovery Research. It is there in Amazon, Geo7, even in other platforms like Ghana and Apple, Google Podcast, Spotify. There you just search for Drug Discovery Research, you will get it. So there I'm talking about the success stories of drugs, which is approved by FDA based upon CADD. Now let's imagine when you don't have a 3D structure of the protein, but you have the small molecules with you. At that time, we have to use a different approach called as ligand-based approach. There we use pharmacophore, similarity studies, and QSA, but not docking or molecular dynamics because we don't have the protein there. Let's imagine when you don't have protein and when you don't have small molecules. We completely use the high throughput screening method, try to combine everything, mix and match, and come up with what are all other possible targets coming in. So this is what we are looking at when you're going for combinatorial drug designing approach. And this is very important when you don't have much data to compare or to be available. Moving forward, what do you mean by molecular docking? The word docking, I'm sure everybody knows that, right? It is being used everywhere in our life. A docking means when you're locking your pressure cooker's pressure valve into the valve, that is also called as a docking. When you're locking your pressure cooker's lid on the actual bottom part, that is also docking. So anything that can go and bind to another material is called docking. But why we call it as molecular docking? So we want to understand how a compound chemically react with proteins in a human body or any others, which is having some interaction and good binding affinity to make an impact on the function of the protein. That is very important. Until it makes an impact on the function of the protein, it doesn't have any value. But in 18 earlier days, and still in our books, we learn it as key lock hypothesis. But unfortunately, things have changed. It is only half the truth. The reason is multiple keys can go into a lock. Multiple locks can fit into a single key. That is where we are heading to molecular docking. Now, to simply explain what is molecular docking, we are trying to find the possible okay, let me put it this way. So when you have a compound, simply you purchased a compound, right? Or you purchased a drug, a medicine that has a crystal structure, okay? A specific 3D structure it has. But when you consume it and when it goes and binds to your protein, which is called as a biological target, the structure of the compound might change. I mean, not the chemical composition, but the conformation, the geometry will change. So that geometry is called bioactive conformation, right? So docking is a process to find out the bioactive conformation of a molecule, which can bind to the receptor, and we quantify the strength of the ligand receptor interaction. Just a minute, Gaurav sir, how much more time I have?
Yes, sir. Sir, you can take, sir, about 15 minutes more.
Okay, thank you.
Yes.
So if you are trying to explain a molecular docking, let's say a biologist is explaining to a computational computer science, or if I am explaining to my grandmother who doesn't know about science, but she knows about history, so she might ask specific questions of molecular docking on a layman terms, where my drug will go and fit or sit. How my drug, whatever I have consumed, how it is going to bind or how it is going to sit there with the protein. How strong it can be there, whether it goes out and come back or how strong it can sit there. What is the role of solvation? Because we have a lot of water molecules or solvents within our body system. So how this drug is going to react with these solvents? And what makes my compound bind to the specific protein rather than other protein? Because there are thousands of proteins in our body. How can this drug find exact protein inside our body? What is the changes in the geometry? I told you, the geometry outside our body will be different, and when it's consumed, it will change when it interacts with the protein. So what are the changes happening to the ligand on its geometry in order to look at translation rotation of the compound? So this is a normal question that we are trying to address from molecular docking. But unfortunately, what we do is molecular docking means docking score. Molecular docking means binding affinity. That's all we look at. That we have to change, because transfer of knowledge is something very important. When we are conveying our research outcomes, it should be understandable interdisciplinarily. Then only it is reproducible. Don't think that only a biologist can do biology. Others can also do if they are able to understand it. Now, machine learning. Do you think that all the biologists are sitting and doing machine learning coding? No, computer science people. But computer science people not able to say whether the results are correct or not. That is where we have to understand causation versus correlation. It's an interesting phrase, actually. If you search online, you'll get a lot of interesting and funny examples. Causation versus correlation. One simple example I'll tell you. The rate of death in a city is correlated with the rate of divorce. There is no correlation, there is no cause there actually. right? How can you correlate a death with the divorce happening in that city? But on a statistically, you can correlate with anything, and it correlated well. So in biology, you cannot do that. In biology, causation has a very much big importance, and including the correlation. So when a computational scientist or a data scientist or a statistician might not know about biology, but he just looks at data and looks at how far it is correlated or accuracy. But only a biologist can tell, or a chemist can tell, whether that correlation has any physical significance or any meaning on that correlation. That is very important. So, okay, considering the fact we have, so this is what is called the protein. So black region. So I'm taken only the small portion. Don't think that is only protein. The whole protein might look like this, actually what you're seeing here. So I took only a small region where you can see a pocket, a space, and this colored region with blue and red and white, this is the ligand or the small molecule. So after doing docking, I could see that my molecule is entering the small surface area of the protein, which can go and bind. Now, binding means how it is going to change my function, how it is going to cure my disease. That is because this, as I already told you, this ligand or small molecule is able to form some chemical reaction with the certain amino acids. I told you, protein is building blocks of amino acids. So we already know that the biologist already identified that certain specific amino acids in this region of the protein is responsible for a mechanism or a disease. If you can block the action of those amino acids, you can correct the mechanism, right? In a simple layman terms, I'm explaining, not using too much of scientific words. So that is ultimately what we are trying to do, docking. And if you could answer all these questions based on those analysis, that makes wonders. But one thing we are missing in many of our studies is conservation studies. For example, we identified an anti-cancer drug. We claim that this is very good for maybe for lung cancer, right? But the question comes, you might test it only for lung cancer. Now the question comes, how confident you are that your compound binds to a lung cancer target, not to a breast cancer target? We don't have an answer because we did not test it. And we cannot tell about selectivity, specificity also. Even if it is going and binding, we will say that my drug is a multi-target. How can it be? Did you test it? No. So conservation study is something very important nowadays, or even earlier also, but we never looked at it, because we have to make sure that our drug is going and binding only that specific target. So this is what this question, what makes a ligand bind to the receptor better than the others? Why it is not going for other receptors? That you have to research and tell them, very important. Until unless you're doing that docking, it doesn't have any meaning. And then other factors are there. I'm not going deeper into it. This I'll just skip. Too many equations, people might not like it. But let me explain how all these things I was able to put it in a proper way. So this, whatever gray color you are seeing is the protein pocket, or the place where the key amino acids are there to go and interact. Whatever you see here in green colored circles and some golden color stick and balls, they are the compound or the molecule which entered into the pocket of the protein and interacted. Apart from the binding affinity, as well as apart from all your docking score, the main key factor is we wanted to understand whether this the compound which is binding with my protein after molecular docking, can it be crystallized or not? Can it form a co-crystal or not? Until it forms a co-crystal, we cannot confirm whatever docking score you give, whatever binding affinity you give, it doesn't have any meaning, right? So basically, we are also trying to quantify whether these structures or different isomers or different conformations are crystallizable or not. If it is represented by green, we say it is fully observed in Cambridge structure. It is just a X-ray crystal structure database, Cambridge structure database. If it is yellow, we say it is unusual. If it is red, we say very rarely observed. So we have to be very sure. Another thing, these whatever the green and red color things are represented, it is trying to tell you how far the affinity or the energy is being contributed from this atom. This also helps you understand things. So I'll just quickly skip these because this is again, we are talking about big data in drug discovery. We can have billions of compounds for analysis. I'm just skipping because of interest of time. And another, this is if you designed a compound computationally, if you wanted to know whether this is synthesizable or not, because the biggest challenge between a modeler, chemist, and biologist is they having miscommunication. There's a good gap between the communication. So in order to reduce that, if you're designing some new molecules, if you could convey to the chemist that these are the possible retrosynthetic pathway, it would be nice. With this slide, I will end up because our time is already up. Can you please mute? Excuse me. Can someone mute you? Okay. So,
what difficulty are you facing?
I think someone is switched on the mic.
Dear participant, please mute your mic.
Okay, fine, great, thank you. So with this slide, I'll try to end up my session because it's already time. I can take up some questions if you have. So when you look at the objectives of the drug discovery, make sure that we learn 3 different terminologies here. One is hit molecule, lead molecule, and drug molecule. So quickly, because of no time, can all of you tell me what do you mean by a drug molecule? At what stage you tell or when you call a compound to be a drug molecule? Either you can unmute and tell me, or you can put it in the chat. But please make it quick.
Hello, sir. This is again
Yes.
Dr. Arup.
Yes.
Answering your question, a drug molecule is a molecule that interferes with the biochemical pathway of our body and try to change the clinical manifestation of the disease.
Okay.
And it hits the target portion of the body to which the efficacy of the disease is diminished.
Great. Thank you so much, sir. Any others? There is an answer from Dr. Srikanth. Is the only way to cure a disease is to influence a block specific protein? Oh, that is a question. I'll take up the question a bit later. Okay. So when you call drug, I'll come to that. What sir has given is a wonderful explanation, but I will also give one more perspective there, but a completely acceptable explanation. So when you talk about hit molecule, the hit molecule is basically the initial screening. Let's say you have 1,000 molecules and you did a bioassay studies or docking studies in order to find hit molecule. Hit molecule is never a one molecule. It could be 10 or 20 or 30 molecules actually. So that is called hit molecule. When it comes to be lead molecule, if you want to call it to be a lead molecule should be a hit molecule for sure. That means hit molecule should have good potency. It's very mandatory. Unless you have a good potency, you cannot call a compound to be a hit molecule. So when you have good potency along with safety, safety means toxicity should be taken care. Then absorption means your human small intestine absorption, human intestine absorption rate should be good for the molecule. Metabolic stability, because your compound can pass through P450 or liver metabolism. So you have to make sure that your compound doesn't break down and form a metabolite. If a compound is breaking down, when it reaches the target, it might not interact properly as you expected in the potency studies. And the compound should be soluble, permeable, and flexible. Once all these are satisfied, that's where your hit molecule can be called as a lead molecule. So lead includes potency too. And we call it as a drug molecule once we get an approval from NDA, as well as when it passes through efficacy, all those things as already mentioned. Unless then we cannot call a compound to be a drug. So in your research paper, you cannot call your compounds to be drug because you did not do clinical trial. You can tell about lead, you can tell about hit molecule. That is within our capacity. So we want to identify compounds which has an optimal balance of all these properties. That's very important. Then only we can call it as a drug candidate. That's why my title was drug candidate, because I cannot call it as a drug because I didn't do clinical trial. If you are not able to identify such situations, a balance of all these properties, then stop that compound. Don't work on that compound. The problem here is since we started, we feel a little bit lazy or maybe time constraint. We try to end up with something. I don't say jugad, but still we try to end up with something moderate activity. We're not getting a better activity. I hope many of you will agree with me. You're not getting a best activity or better activity than the marketed drug. That is because we did not plan our time accordingly, and we did not find out the balance property. We always looked only at the affinity, but affinity is only not required to call that compound to be a lead molecule. In order to get into lead molecule, not only cytotoxicity, I know everybody does cytotoxicity, but no, we need many more other analysis to confirm that this is a good candidate. And final one question, and then we will stop the presentation.
Sir, I have a question. Sir, if there is a very small molecule, which we call the marker molecule, in terms of the proteomics, when you do the mass spectrometry, because most of them are the garbage proteins. Suppose there are collections of the small molecules, very small molecule and short-lived molecule. For example, if you take in case of the phosphoproteins, they are very short-lived, and they are presently they are mostly role in the signal transduction in the cancer. So, can we predict a model of the small molecule targeting the organs through the drug discovery, machine learning approach, or AI approach? If you can light on this question.
Yes, sir. But there is an answer both yes and no. The answer yes is for if you have already analyzed proven data, both successful and failure data, both should be there. If that data is there, you can train a model and you can predict for the new ones. That is possible. The answer no comes when you don't have already known experimental data. If you don't have much means you should have at least more than 1,000 data points. If you don't have, then AI machine learning does not have a role there. So it's very important that you have a good experimental data base in order to generate any models in machine learning. So that question has two different answers based on the data availability. That's why I gave you a stress on my presentation that based on the data only we can talk about any analysis. Right now, a question time. Imagine you have 10 different compounds. Okay, no, let's say 20 different compounds, 20 different small molecule compounds. You have to carry out 10 different experiments. Okay. All these experiments are required in order to call this compound to be a drug molecule. All 10 is required. Now, tell me, the computer experts and statisticians here, if any, tell me what is the best approach you can apply here. Keep in mind the condition is you have 20 compounds, you have 10 different experiments. All experiments are required in order to make a decision. I want to choose the best 5 molecules from this 20 molecules. What is the approach that you have to apply? In a normal sense, if I have 20 molecules with just one experiment, it is very easy for me because either the higher value or the lowest value I have to choose. These are the best 5 molecules I can conclude. But here the challenge is I have 10 different experiments to be considered. So how can I take that? Or how can I do analysis? This is a question for computer science, machine learning experts, and statisticians. Only you can answer this. So this is how we bind interdisciplinary aspects. So this is a challenge for the drug discovery or biologist. Now, how can you solve it? Any answer? Any thoughts? No? Okay, because of time, I'm not waiting for that. So let me quickly finish this. So when we have a lot of data from in silico, in vitro, in vivo, we have to look at the importance level. Which data is more important and which data is less important based upon uncertainty. That's how we do this analysis. Due to interest of time, I have to skip a little bit, but make sure that when you do a filtering approach, you have to be very cautious in this method because you think that in each step you are filtering the bad ones or keeping only the good ones, but ultimately you might end up the bad one. In most cases, it is happening in that way. So make sure that you do an approach, something called as rule inference based method. That is called a multi-parameter optimization method. Multi-parameter optimization method is something called as rule inference. It is not machine learning. It's completely into statistics. So when we use this rule inference based method, we could give the importance to specific property more important to decide which is a drug candidate proning property or not. This is already proven back in 2010 on CNS drugs. So it's already 11 years. The question is, are we using here in our research? Unfortunately, no. None of the Indian publication have seen this. People are using this. So we have to make sure that we start using all these techniques for our better prediction. I'm not spending too much time here because of less time. And these are the success stories of computer-aided drug design. There are more than 112 molecules which is available. You can take a screenshot of this slide because these are the two papers, review papers. I have taken this particular data to talk about the success stories of CADD. And when coming to machine learning, these are the software which I was telling you, 9, where you can use machine learning models. You can generate many drug discovery as well as biological data, including data mining, text mining, protein data mining, string database, systems pharmacology, all things can be explored. All these are available in the link that I have already shared in the chat window. I have already shared. So don't worry, probably later we can again get connected with the session to cover those slides. But to conclude, kindly consider 25% importance to score and quality and 75% to diversity as a size, as a lead residue. Consider 75% for score and 25% for diversity. So make sure that your different chemistries involved in the selection of compounds. Consider enrichment factors for your virtual screening. Good synergetics between human expertise and computational tools are very important. Avoid missed opportunities because don't do unnecessary things and then don't lead to come to understand the wrong strategy. Understand the significance of parameters or properties properly. Evaluate and decide the tool or the approach. Check the reliability of the data that you have used, which is very important, especially for these teachers. Until you try yourself and train others, we can never be an expert. We will be keeping on learning, but we have to apply it somewhere to be as an expert. So that's all from me. Due to the interest of time, I had to move a little bit quick. Sorry about that. Okay, the link you want to share, I'll share it again. Gaurav, I'm done. Sorry, at the end, I had to go a bit quickly because other speakers has to come up with the timings.
Thank you very much, sir, for sparing your invaluable time and suggesting us about different approaches to the drug discovery. So there are a few questions from the chat box that I would like to address to you.
Yeah.
Yes, sir. So
I'll read it.
Okay,
there is one question from Dr. Srikanth. Is the only way to cure a disease is to influence a block specific protein? Is there any other people can get cured? Dr. Srikanth, don't think in the way of curing a disease. No, it is actually, we are doing a very preliminary studies to understand at the protein level, because we have to look at the gene level. We have to look at how organs are going to react with the drug and how the drugs are going to react with the organs. These all things has to be analyzed. So, but on a computational perspective, we are actually looking at the specific protein only because we know that that protein is taking part in a specific mechanism or the pathway. So targeting that receptor or the protein can lead us to that mechanism. But the challenge here is a single disease is not a single receptor is not responsible for a single disease. There are multiple receptor or multiple mechanisms are responsible for a disease. So only a specific portion or a phase of the disease can be cured, not the whole disease. Now, yesterday there was a good news came in the news that FDA approved Alzheimer's drug where they were being researching for the past 20 years, right? That is a breakthrough. But even though as a normal person, we understand Alzheimer's drug has come, but it is only controlling the growth of that. So only the growth is being controlled. So only a space can be controlled in a disease using a drug design or a specific drug. Now, another question is, Is the process of vaccine discovery or drug discovery similar? No, sir. Vaccine discovery is more of a therapeutic way. Drug discovery, therapeutic way in the sense we are more looking at peptides and trying to understand the specific regions from the affected species and then trying to do it. But drug discovery is mostly from the small molecules or biosimilars. What use of ML for vaccine discovery? Vaccine discovery, ML can be used only to identify the new possible peptides or epitope that could be coming in, possible ones if you want to design a new thing. But most of the time, as I said, we take it from the species. Are AI and ML used there? Slightly used basically on the analysis. What strategy of ML can be applied for vaccine discovery and drug discovery? ML mainly can be applied on prediction of activity data as well as this pharmacokinetic property data, which I discussed. And also you will be able to analyze a target identification. So by giving using NLP or by looking at similarity data, you will be able to use machine learning approaches in drug discovery. Okay. Thank you so much for your feedback. Can you suggest some current researchers on protein binding such that protein bindings become dangerous? Sajina, protein binding on computationally, it's a bit challenging. Even though we have a lot of studies came up, we always look at the shape electrostatics point of view. But protein interaction is much more beyond that. So combination of computational and binding studies on the wet lab studies will give you a much better understanding of protein interaction. How can we identify whether DNA of a disease is created artificially or not? Mutagenicity or genotoxicity that is happening in the damage of DNA because of certain molecules or certain food particles of higher dosage. So that could be induced actually, right? So I think that you are asking for artificially or not. After symptoms happens, we are in drug discovery, but before the catastrophe happens, if there is any method to monitor the protein level in human body. See, that is where we are using all the existing data to understand how we can predict something there. But unless because our human body is more dynamic. Of course, there are simulated things to understand what could be the symptoms that might come up. But different variants, for example, in the case of corona, different mutants are giving different symptoms also. Sometimes it is unpredictable, even though we predict things based on that drug is very difficult. Now, coming to corona itself, COVID-19, we don't have a one single drug to solve everything. At different stages, we need to have multiple drugs that has to come in. Can you suggest books? Books rather than books. I would recommend you to start with some video lectures and reviews first, and then get into books. Sorry, I really have not got into deconvolution, so I really cannot comment on that. Sorry about that. Yeah. So thank you so much. I think all the questions I have addressed.
Thank you so much, sir. Thank you so much, sir, for addressing all the questions of the participants. Now, I would like to invite Professor Ranjana ma'am to give an e-memento to sir.
Thank you very much, Dr. Pillai, for such an informative lecture and accepting our invitation for this FDP, and kindly accept our e-memento from us.
Thank you so much, madam. Thank you so much. A pleasure talking to you all, and thank you so much for your patience listening and for your interesting questions. Thank you. Best wishes. Take care.
Thank you very much, sir.
Thank you so much, sir.
Thank you, sir. Thank you very much.
Thank you. Bye-bye.
I am requesting all participants to kindly on their videos. That's why we can take a group photograph with the Girinath, sir. Pillai sir, are you able to on your video, sir? If not comfortable, then let it. Okay. Thank you, sir. Thank you so much.
Okay. So the next speaker for day 3 will be Dr. Abhimanyu Thakur. He's from University of Chicago, ILUS, and he will be enlightening us on the topic of artificial intelligence in exosome-based disease diagnosis. I would like to give you a brief introduction about him. Dr. Abhimanyu Thakur, he obtained his PhD in biomedical sciences at the City University of Hong Kong by investigating the role of glioma-derived exosomal surface proteins as biomarkers and functional mediators. Next, he joined as postdoctoral fellow at Hong Kong Institute of Science and Innovation, followed by in the Pritzker School of Molecular Engineering, then made Department of Cancer Research at the University of Chicago, USA. His postdoc research is focused on the crosstalk of stem cells derived exosomes in health and disease. He is a recipient of International Research Abstract Award by Korean Cancer Association, South Korea. He was awarded with Young Investigator Award in 2019, which was conferred by American Association for Cancer Research, Service Appreciation Award by Hong Kong Precision Oncology Society in 2018. He has Outstanding Academic Performance Award in 2018. City Chao Yes Ching School of Graduate Studies Research Tuition Scholarship was awarded to him in 2018. Hong Kong University Grant Commission for PhD was awarded from 2015 to 18. Various travel grants for the prestigious Global Young Scientist Summit in 2018 was awarded. He was also a member of the 109th annual meeting of AACR 2018 Chicago, USA, and All India Council for Technical Education Graduate Pharmacy Aptitude Test. Scholarship was given to him in 2013 to 2015. We would like to welcome you, sir.
Thanks, Bharti, and thanks, Dr. Gaurav, and thanks to Galgotia University for providing this opportunity and this platform, and thanks to Dean as well. Yeah. So let me share the screen, I think. Can I share this?
Yes, sir. You can share, sir.
Okay. So is it visible? My screen is visible to everyone?
Yes, sir.
Yes, sir. Yes, sir.
Okay, so so it was a nice talk by Dr. Pillai. First of all, thanks for a wonderful lecture. So I'm not a computational scientist, and neither I'm not even an expert in artificial intelligence, but I have been using this technique for last two years, and I published 2 papers in this field. But I'm still a novice in this field, so so pardon me if I'm going somewhere wrong, because there are several experts related with artificial intelligence and machine learning and this computational stuff. So actually, in this talk, I'll be mainly talking about how we can decipher some important biological questions and how we can understand biology, because biology is more complex, like what we think about, like 1 + 1 = 2 in mathematics, but here it can be like 1000 or it can be like -1000, something like that, you know. So biology is unpredictable. I mean, it is predictable to some extent, but we cannot model completely. Yeah, obviously many data scientists, they have put a lot of endeavors, so I'm not denying that. But yeah, we have to understand what sort of biological questions we are going to address, and what is the molecular events and what is the phenotype we are going to expect. So all these things we have to consider, you know. Okay. So there are a lot of diseases. There are a lot of fatal diseases like cancer, or even like a lot of progressive diseases like Alzheimer's disease. You know? So all these diseases needs a lot of attention. because the survival rate is low, and even the cost of treatment is very high, and there are like even like some added even challenges, and it puts a lot of burden on the family and by the time we detect this disease, it already surpassed the advanced stage. So there is a dire demand for early diagnosis of this kind of I think someone has to mute yeah. Hello.
Sir,
Yeah,
you
Doctor.
can continue. I have withdrawn the unmute.
Yeah.
command.
So there is a dire demand for early diagnosis of this kind of fatal and progressive disease because an early diagnosis will open the door to future care and the treatments. So here I would like to take an example of cancer, of a particular cancer, that is glioma. It's basically brain cancer originating from various different kind of cell, mainly neuroglia stem cells in central nervous system, and and as you can see with the as per the latest statistics, there are around 1.7 million patients are affected by this glioma. And the one of the most aggressive form of glioma is glioblastoma. So this is the fourth stage of glioma. It's also known as astrocytoma, because there are many different kind of cells in brain apart from neuron. Because when we talk about brain, we only talk about neuron, but there are supporting cells, like microglia, astroglia. So I think I know there are a lot of non-biological people. So pardon me, and let me know if you have any questions, if I'm going somewhere around. So so the aggressive form of the glioma is glioblastoma. And importantly, even though there are combination of treatments in market, for glioma, the drug available is temozolomide. And even in terms of treatment, there is radiation therapy and even surgery. But in spite of the availability of this temozolomide, there are different limitations. Like it can extend the lifetime only for like 1.5 years, and also there is a chance of relapse. And in spite of all this, the survival rate is only around 14.6 months. So we can understand how a person suffering from this disease like brain cancer is already on a deathbed, and therefore it's very important to develop a diagnostic method for early diagnosis, and right from beginning of any molecular event before it leads to any particular clinical symptoms. So it's very important, because by the time we detect this already surpassed the advanced stage. Like suppose someone has brain cancer and they suddenly feel headache, and by the time it is in second stage or in the primary stage, we think, oh, it's due to some like normal headache, but that headache might be due to some other constraints in the brain. So that's why it's very important to understand how this molecular signature can be detected, like before the appearance of real particular clinical symptoms. So here I would take another example, that is Alzheimer's disease. Dr. Pillai just talked about this briefly. So it's a neurodegenerative disease, which is a progressive one, and it involves a slow degeneration of neurons, leading to various clinical symptoms, including dementia, that is loss of memory. And the two important molecular events which occur are one is the decreased level of this amyloid beta protein and the increased level of phosphorylated tau in cerebrospinal fluid. Well, the available treatments for its diagnosis, including either invasive they are either invasive or costly, which again creates a huge burden on patients and their family. So therefore, again, there is a demand for developing a non-invasive or minimal invasive based on like biofluids methods, which should be like robust and can be easily handled. So from these two slides, we understood like there is a demand for developing non-invasive and some like very simple biofluid based detection system for early diagnosis of this disease. So as we can see, there are several imaging devices such as CT and MRI, which are although gold standard but suffer with various limitations, such as patients like those who get exposed to this harmful radiation, and even they need the injection of contrasting dye, you know. And moreover, this tissue biopsy is highly invasive, and they require professional handling, and most importantly, these are painful and incurs like discomforts to the patients. So again, this also shows that there is a need for that non-invasive diagnostic method. So now there is a game changer, that is liquid biopsy. So what is this liquid biopsy? So liquid biopsy is basically using different biofluids from the body parts, like blood, or even cerebrospinal fluid, or even ascites, or even other kind of fluids like urine, the pulmonary fluids. There are many different kind of fluids which can be used for liquid biopsy. And interestingly, these fluids contain several mutant proteins, or even circulating DNA, or microRNA, or long non-coding RNA. So these are all nucleotides, and some are like coding, and some are non-coding. So for non-biological people, I can tell in very simply, so these are all like we have nucleic acid like DNA or RNA. So similarly, there are some non-coding things like long non-coding RNA, or even ctDNA. So because those who code, they form that protein, you know. So but those who don't code, they do not take part. But interestingly, these also take part, you know, so in several different biological events. So that's how these important constituents of the biofluids can be used for the early diagnosis of several fatal diseases. So more recently, advances in the detection and characterization of circulating tumor DNA have finally enabled the introduction of liquid biopsy, and they have also even come into the clinical practice. And recently, FDA has approved this for finding like genetic alteration in plasma, using it as a companion diagnostic match to specific molecular target therapies for cancer. And how this can be useful? This can be useful like they can investigate the abnormalities, and which can even detect on imaging examinations such as mammography or lungs computed tomography. And it can be also used for disease surveillance, and it can also guide the selection of most appropriate treatment and monitoring like treatment response. It's more like a prognosis, you know. After giving the certain treatment, you have to like track how the response of that particular treatment is, like post-treatment, you know. So among the biofluids, there are various constituents, as I already talked about it. And one of the there is a very paradigm shift recently, that it's about exosome. So which I will talk about in the next slide. So what are these exosomes? So these exosomes are very tiny extracellular vesicles, which are around 30 to 200 nanometer, and these are released probably by most of the cells types, you know. Like just you name it, it is released by most of the cells, like 99.99%. So, and it was discovered in early 1900, I think 1990, around 1990 it was discovered. And after that, it was in the dormant phase, like for the beginning, like 11 years. And after that, there is a boom in this field, like this exosome. You just search it in Google, you know, exosomes or extracellular vesicles. So interestingly, these exosomes can be excreted by most of the cells in the extracellular space, and these are also present in the biofluids like blood or cerebrospinal fluid, and they take part in the various biological events, like they communicate, you know, they take part. They also act as a messenger. That's why they have also been investigated as a very important drug delivery system, like just loading the drug and for targeted delivery, or even they have also been used for nucleotide delivery system. Interestingly, these exosomes carry several mutant proteins. For example, in case of glioma, this EGFR-V3 is a very specific mutant. I think it was discovered. It has been discovered in early 2016 something. And there are also several microRNAs like microRNA 21. So these are very specific for glioma. And similarly, like for EGFR for in case of lung cancer, and in case of ovarian cancer, there are some, like different cancer have different kind of mutant protein and mutant this microRNA. And similarly for other like diseases like Alzheimer's disease or even Parkinson's disease, there are several mutant proteins which can be detected, which have already been detected in the exosome as a constituents. Interestingly, these exosomes can cross the very like different kind of barriers, including blood-brain barrier, blood CSF barrier, and they are found in the peripheral circulation, and they can mimic their parent cells. So they are very good at impersonating the cells from which they have derived. So we can say that these exosomes are very good. This can be a very good platform for the detection of biomarker for this for the diagnosis of disease, or even to track or monitor the disease progression. So in 2020, I published a science paper, and based on this exosomes exosome-based liquid biopsy for glioma, and it got highlighted in several different platforms, including Times Higher Education and also Asian News Research. So which I will be talking briefly why I'm talking about the exosome and the artificial intelligence. So very soon I'll be introducing machine learning also here. So you can see in picture my boss is there, my PhD boss. So, and these are our collaborators. So in 1931, Otto Warburg, he got Nobel Prize because of his postulate, because of his because of his theory that that all forms of cancer are characterized by two basic conditions, that is acidosis, acidic environment is there, and hypoxia, that is lack of oxygen. And the lack of oxygen and acidosis are the two sides of same coin, where you have one, you have the other. So basically he talked about aerobic glycolysis, which I will talk here in this next slide. So for most of the cancer, one of the key hallmark is in the malignant progression is closely related with metabolic reprogramming. So what is this metabolic reprogramming? So in the metabolic reprogramming, different cells in the vicinity, because when we talk about cancer, we only talk about the cancer cell, but we don't talk about the environment. But we have to consider how the cells in the vicinity are interacting. So what happened is there is a high glycolysis due to the energy requirement, and therefore the this cancer cells, they increase the glucose consumption and lactate production, and this lactate causes the environment as acidic, and this lactic acid has to be gone from out of the cell, and therefore for its exocytosis, these two important proteins, MCT1 and CD147. I think just now someone was talking about is there any fatal disease which can cause due to the protein-protein interaction or protein-protein binding. I think some Saikia or something someone was asking about. So here I'm talking about these two glycolytic protein, MCT1 and CD147. They interact, and they take part in the extracellular. They take part in the exocytosis of lactate, and they form a couple with the cells in its vicinity, and together they cause the tumor progression in most of the cancer cells. So therefore this metabolic reprogramming is very crucial in the progression of cancer cells. So MCT1, it's a lactate transporter because it transports lactate, and CD147, it's a helping protein. It is binding this MCT1 and MCT1, and together it is causing these three important phenotypes. Of course, there are many other phenotypes, but these are the three important ones, like migration, proliferation, and angiogenesis. When we talk about cancer, so these three experiments we generally do to examine how the cancer is behaving. So these MCT1 and CD147, they take part in the tumor progression. Of course, if we block this, so they can be one of the target and as an anti-cancer agent, and they can, at the same time, they are crucial indicators of cancer malignancy. Interestingly, most of the diseased condition, including these cancer cells, they release high number of exosomes compared to their control group, compared to their normal healthy cells, but nobody knows how. Of course, many people have deciphered. So how how these are helping? So for that, this is our experimental, sorry. So this is our experimental result. So we detected the MCT1 and CD147 under normoxia and hypoxia. So let me tell you what is normoxia and hypoxia. Hypoxia, we use 1% oxygen. So this is a tumor this is a microenvironment in the tumor. That's why we used 1% hypoxia. Generally, we use carbon dioxide incubator and 37 degrees centigrade temperature. So in case of hypoxia, we use 1% oxygen. So interestingly, what we found is that MCT1 as well as CD147, they are highly upregulated. So this is the immunogold EM data. Like these dots represents the protein. All these dots represents this MCT1 protein, and these dots represents CD147. And this is how the exosome looks like. This is the electron microscope structure. These are around 100 nanometer. This shape. And this is the CD63, one of the marker protein for the exosomes. So if these proteins are available, that means exosomes, what we have isolated are indeed exosomes. And again, we used a very common technique, that is Western blot, and we found that there is a dramatic increase of MCT1, CD147, and we used CD63 as a control group. And at the same time, we did ELISA experiment, and here you can see that there is a significant increase of MCT1 and CD147 under the hypoxic environment. Well, so far we understood there are several different techniques. So these were the biology. So there are several different techniques. So like in the clinical method, there are MRI and CT. And for the biochemical methods, there are like electron microscope or ELISA or even Western blot. But most of these clinical methods, they cannot detect early, and they are limited in resolution. Because actually the resolution is such that by the time this MRI or CT scan they detect, the disease is already in the advanced stage. And similarly, these biochemical methods, they are either time consuming, or they are direct, or they even have limited sensitivity, or there is a chance of protein contamination, or there is a requirement for large volume of sample for the analysis. So all these together challenge the detection of precise molecular signature of cancer progression. Therefore, we need a very robust method and with very high sensitivity. And we propose to use LSPR and AFM, which are like non-invasive, precise, good for precise monitoring, and they have an added advantage of label-free, and they can be used as a biosensor for detection of exosomal constituents for disease progression. So in my next slide, I'll be introducing what is LSPR and AFM. So LSPR, it's a technique. It's a bioengineering techniques. Which detects the changes in refractive index at the surface of a sensor due to antigen-antibody interaction or protein-protein interaction. So this is a typical diagram for the sensing chip. Here we have a monochromatic light, and whatever the event takes place on the surface of this chip, it can be detected in the form of a sensogram. And this sensogram is very important, because even the minor change can be detected. So suppose if you want to detect any molecular events like in a very early stage, this can be very helpful. One important point about this method is this is already available in the cancer diagnosis in most of the laboratory setting. Of course, not normal lab, but like high profile lab, they already use surface plasmon resonance, but this has got some added advantage. So what mostly people use gold surface over the glass. So we use self-assembly monolayer gold nano island. So there is a nano island. So here you can see this yellow mark. These are all nano islands. So these are all like 10 to 20 nanometer islands of gold. So how this can detect the molecular events? If there is a interaction, there will be an association curve, and if there is a dissociation, if the analyte detach from the substrate immobilized on the surface, there is a dissociation curve, and again there is a regeneration happens. Next is AFM. So AFM, this is also another bioengineering tool which has a very high resolution. It's a very high resolution scanning probe microscope, which can detect with a resolution of fractions of a nanometer. So the typical setting has one cantilever, and one sample disk. So we immobilize the samples on this sample disk, and this is the scanning electron microscope image of a typical AFM tip. So this is around 10 to 20 nanometer diameter. So typical, like conventionally people use mica surface or even gold over mica surface. So we used very advanced one, that is gold nano island sample disk. And this is a typical AFM force curve. So how this detects the molecular event is, like if there is an interaction between the analyte and the substrate immobilized on the surface, on the sensing surface, on the sample disk, there will be this kind of curve. And when it detaches, it forms a retraction curve. And if there is no signal, that is like non-specific interaction has happened. So this is again our experimental data. So this is the typical AFM curve, AFM image. So you can see all this signing ball-like structure. These are all exosomes, you know. So these are all around 100 nanometer. And this is a 3D diagram of a single AFM image. AFM image of a single exosome, and this is a 2D 2D image, and this is a height curve. Here you can see that this is around 100 nanometer, and this is around 300 something nanometer. So interestingly, we detected the enhanced level of MCT1 and CD147, like using this LSPR as well as AFM very significantly. So you can see that this hypoxia has high level compared to the normoxia. So compared to this molecular characterization, these methods are highly sensitive. So again, you might be wondering why I'm talking about all these molecular events. So let me conclude this part. So we already found that this MCT1 and CD147 could be important targets, and already there are several ongoing clinical trials related with these two important glycolytic protein. And our lab for the first time detected MCT1 and CD147 in the glioma derived exosomes and from a mouse model, and we established this as a non-invasive tool for the diagnosis of brain cancer. And in another research, we detected EGFR-V3 with a high sensitivity and in the glioma cells derived exosomes. And this got published in Advanced Functional Material as a cover article. So as we saw, there are many different methods like biochemical methods, clinical methods, all has like several disadvantages. So I'm not going too much in detail, but there are many methods which I already talked about, like scanning electron microscope, transmission electron microscope, even dynamic light scattering technology, nano tracking analyzer. All these methods can characterize those exosomes. And even I also showed you some of the data about Western blot, even ELISA, and even flow cytometry. Each of these methods have their own limitations. So how can these limitations be overcome? So here comes the machine learning in picture. So we plan to integrate machine learning and the exosome biology. So I'm not going too much in detail about the machine learning because our previous speaker already talked about this one, and I'm still a learner in this machine learning field. So I will talk about what I did, what I experienced, and what thing can be done, you know. So yeah. So I had a question like, can these machine learning be helpful for identifying the initiating parent cells of exosomes? So you might be wondering why this is very important. Like suppose suppose if you take a blood and that blood is a systemic blood and it has many different kind of cell, because there are like immune cells like macrophage or even different kind of cell it has. If you have cancer, there are different kind of proteins. So it's a mixed one, you know. So how you are going to understand that that a particular exosome is derived from particular type of cell? So to answer that question, we applied machine learning. So for that we chose two different model. One is a retina model, another is cancer-based model. So why I'm saying cancer-based? Because this is not exactly a cancer cell, but this is a cell which is helping the cancer to proliferate. This is endothelial cells, you know. So these cells provide nutrition in the hypoxic environment to the cancer cells. So we chose endothelial cell. And in another disease model, we used retinal pigmental epithelial cell, which is a part of retina. And then we isolated nanovesicles, that is exosomes. Okay. So this is our plan actually. So we first characterized using the nano tracking analyzer, which can give you several different kind of features of exosomes. And we then we used we applied machine learning based several models. First, we divided this in the training data set, 80% and 20%. And we applied several different machine learning models like support vector machine or support vector machine, decision tree, and back tree, all these models. And then we asked like whether it can predict or not. So this is the characterization data. So we clearly see that there is a decreased number of exosomes from the endothelial cells. So this could be one of the important molecular marker itself. Okay, but for the molecular marker, you have to do biopsy, because whenever somebody happens to suffer from the cancer, they go to doctor and they ask doctor साहब मुझे cancer हो रहा है ऐसा कुछ. And then the doctor simply asks for some biopsy, and that biopsy involves a lot of like invasive procedure. They're gonna take they're gonna do some HNE staining or even different kind of staining. So this particular exosome is very crucial for avoiding those kind of scenario, because you can simply take one drop of blood and you can identify what kind of exosome you have. So recently there is a paper about Alzheimer's disease diagnosis based on even the exosome. They detected this beta and tau protein, I think, in that exosomes derived from cerebrospinal fluid. Of course, that is a mouse model. They did in a mouse model. So here I was talking about, so we did a characterization, and this is the size distribution analysis for the exosomes from the RPE cells and the endothelial cells, and this is the intensity. So you can clearly see that the intensity is lower compared to this one. And we analyzed like what is the value for D10, D50, D90. So what are these values? So these values suggest like what is the 10% of diameter of what is the 10% of the vesicles have the diameter like particle size. So you can clearly see that the 10% of the exosomes has around 100, and most of them have around 300 to 400 nanometer. So all these, when we take like several different exosomes, because one particular set has like will have like plenty of exosomes which we cannot understand, and so there comes the machine learning in picture. So we applied several different models and we analyzed its performance by using this AUC ROC curve and area under curve. So, and we found that by using this back tree model, it gives the highest that prediction performance. And here also you can see we found that in the back tree model that it can detect with the highest efficiency. And of course you may ask like what we got. So we found that in the previous conventional method, the sample processing takes around 1 day. Of course, the sample preparation is almost same depending on the technique what we use. And the sample processing takes almost 1 day for conventional methods, 1 or 2 days maybe. And in the AI based method, there is no need of sample processing. And in case of this conventional method, the sample analysis take 1 to 3 hours. But in case of this, in our method, we saved a much significant time, that is around 15 minutes. And of course, this involves a lot of image processing or dimensional reduction. But here, in case of AI based method, we are ready to use, and it saved a significant amount of time. So concluding my talk, so exosome, it can be an important tool for non-invasive liquid biopsy, and the application of machine learning algorithms on the features of exosomes like derived from biofluids, it can help for the development of potential diagnostic methodology to track progression of fatal diseases like cancer and Alzheimer's disease and many others. So thank you for listening, and feel free to ask me, and I'm open for even collaboration. Thanks.
Thank you very much, sir, for sharing your invaluable research with us. Any questions from the participants? You can ask it directly to sir.
Dear participants, if you have any question, please, you can unmute yourself and can ask.
Dr. Thakur, it was a wonderful and precious slides that we received. I am one of the faculty from University of Kolkata. Just I would like to know about this liquid biopsy. That is it effective for the solid tumors? Can we identify the hard tumors by liquid biopsy, or the early detection of any prognosis of the cancer by liquid biopsy? That means I want to ask what is the sensitivity of the liquid biopsy?
So I think you have two different questions. Yeah, one is whether it is applicable for solid tumor, and another is what is the sensitivity, if I'm not wrong.
Yes.
Is it? Okay, so
yes.
going about the solid tumor, yeah, of course it is applicable for solid tumor as well as liquid tumor like leukemia, because it has been proven, it has been like various paper have already shown, you know, in terms of different kind of cancer, including solid tumor and also leukemia even. So it is applicable. It is indeed applicable because these exosomes are basically secreting when they are secreted from the cells, originating cells. So they are being carried all the way through this peripheral circulation, you know. So the chance of whether that particular so like some constituent protein or the mutant protein is being carried by these exosomes, that also depends on the biogenesis of exosome, which I did not talk here because of the time concern. So okay, I'll tell you in brief, because this exosome basically release from the cells by, there is the endosomal lysosomal pathway. So whatever the protein going through that pathway, there is a chance, you know, there is we cannot claim that that it will go, but there is a chance. But of course we have to check by different biological experiment like immunogold electron microscope, or even by mass spectroscopy. There are different methods, you know. So it is applicable. It is indeed applicable on the solid tumor. And coming back to the next question, you talked about sensitivity. Sensitivity, yeah. So there is a issue of sensitivity. That's why scientists are putting more endeavor to develop new method with high sensitivity. And one of the method which we published in Science Advance, that is LSPR and AFM based method. Yeah, it was a collaborative work. And also my PhD work.
So am I requesting if you can share all these thoughts, because sensitivity, why I asked, because the cancer cells that would be short-lived and very small molecule, the detection range as well as the half-life of the cells is also a matter of concern. If we want to use this liquid biopsy for the detection of any marker, so the short-lived of the phosphoprotein, can the phosphoprotein be detected with this liquid biopsy method?
Oh, I think I don't know exactly. You know, I haven't worked in that direction, but surely we can discuss about this. I think I need to check.
Thank you, sir.
I worked mostly on the CD marker, that is glycolytic markers, and also like surface proteins.
Okay.
Yeah. I haven't worked on this.
The phosphoprotein.
Yeah, phosphoprotein. I haven't worked in that direction yet.
Thank you, sir. Thank you.
Yeah, no problem.
So there is one more question from Mr. Rathesh. He wants to know that we are still using non-tracking technology in this. Is it a new technology?
No, we are not using non-tracking technology. We are using nano tracking analysis. You know, so nano tracking analysis is a method. If you have used like flow cytometry, you know, so flow cytometry, there is a pinhole and it can detect like each single particle. So similarly, there is a method called, because the flow cytometry has one of the important constraint is, one of the important limitation or challenge is like the resolution, you know, because the particle size should be more than 500 nanometer. but these exosomes are less than 500. They are around 100 to 200 nanometers. So for that, we need to do immunocapturing, and we have to coat some magnetic microbeads using exosome, and then we need to detect. So that will be again like that will not be a direct method, again indirect method. So again, we have to introduce several control groups experiment, so there is another added experiments for that. But coming back to this NTA, yeah, this is a very direct method and very widely used in terms of nanoparticle, in terms of exosomes. Of course, this was mostly developed for exosomes. Yeah.
So one more question is there by Ms. Richa, I think. She says, can lymphatic cancer be detected by liquid biopsy?
Lymphatic, sorry?
Can lymphatic cancer be detected by liquid biopsy? It's in the chat box, sir.
Okay, lymphatic. I think it's more related with lymph system.
Yes.
Yeah, I think it can be detected. Yeah, it can be detected. Yeah.
Thank you very much, sir. We are very grateful to you for answering to all the questions of the participants. Now I would like to invite Professor Ranjana ma'am to share the e-memento with you from our university side.
Thank you very much, Professor Thakur. Wonderful research work, really. And please accept this memento from our university.
Thank you, ma'am. Thank you. All pleasure is mine. Yeah, thanks.
Thank you very much for accepting it.
Thanks.
Hello, Abhimanyu sir.
Ji, boliye.
Please accept the token of love from our side also.
Yeah, thanks a lot.
Thank you, sir.
Thank you.
For the next session, we are having with us Dr. Subodh Shrivastava. He will be delivering a lecture on computer vision and their medical applications. I would like to give a brief introduction about him.
Subodh sir?
Yes, Subodh sir. Okay.
Yes, Gaurav, I am here.
Thank you, sir. Thank you for coming.
So, Gaurav, are you able to listen my voice?
Yes, sir. Yes, sir. Yes, sir. Your voice is coming clearly.
Okay, thank you.
Welcome, Subodh.
Good morning, ma'am. Very good morning, ma'am. Ranjana ma'am, good morning. Thank you for giving big opportunity. Thank you, ma'am.
Thank you for accepting our invitation, Subodh.
Actually, Ranjana ma'am is my professor in the School of Biomedical Engineering and Department. So I'm very happy to see you, ma'am, at this place.
I am too happy to see you here.
Thank you, ma'am. Thank you.
Now I would invite Ms. Himadutta Barua to please introduce Dr. Subodh Shrivastava.
Hello everyone. I'm Himadutta Barua. I would like to invite Dr. Subodh Shrivastava for his speech. I just like to introduce him. Dr. Subodh Shrivastava is working as an assistant professor in the Department of Electronics and Communication Engineering, NIT Patna. He has 7 years of post-PhD teaching experience. He has 45 publications in reputed journals and conferences and 6 book chapters. He delivered more than 10 invited talks and conducted 9 workshops in the field of AI and computer vision areas. He got projects from private and state. He is a member of the IEEE and is also connected with the Indian Society of Technical Education through lifetime. He received his PhD from IIT in 2014. His research interests include image processing, biomedical image analysis, pattern recognition, machine learning, computer vision, and their medical application. Currently, he is the professor in charge of IEEE Student Branch NIT Patna, UG coordinator of EC, assistant warden of the hostel, and principal investigator of labs such as digital electronics and anti-MC lab. Please, Dr. Shrivastava, please.
Thank you, ma'am. Thank you, Professor Ranja, madam, and thank you, Gaurav. Thank you all participants. So welcome to all of you. So first, I would like to share my PPT.
Hello, sir.
Yes, ma'am. My voice is coming. Okay.
Yes. You are now able to share your screen.
Yes, sir. It is not host disabled participant screen sharing.
1 second, sir.
Dr. Srivastava?
Yes, sir. You can now share.
Okay. Yes, now it is coming. Okay. So now it is visible. Yeah. Gaurav, now it is visible.
Yes, sir. Visual, sir.
Okay. Okay. So good morning, everyone. So myself, Dr. Subodh Srivastava. I'm working as an assistant professor in the Department of Electronics and Communication Engineering. My field is here the computer vision and its medical application. So today I will represent my talk on the computer vision and its medical application. Okay. So basically, what kind of the content my talk will have here. So first, I will tell you about the introduction to the computer vision.
Hello, sir. You are not audible.
Hello. Are you listen my voice?
Yes.
Hello?
Yes.
It is audible, Gaurav?
Yes, sir. Yes, sir.
Okay. Thank you. So my content is here. Means the introduction to computer vision. First of all, you need to understand about the computer vision, and then I will move on the medical application, where I will discuss about the design of the computer-aided diagnosis system for early breast cancer detection and self-learning prosthetic hand based on AI. So basically, means both works are means I have done. Okay, self-learning prosthetic hand. Basically, I got this project from BIRAC. 44.8 lakhs. So I will discuss a little more about that, how the self-learning prosthetic hand can be constructed there. Okay. So basically, yeah. So introduction to the computer vision, the first. Okay. So here, what you are able to see on the screen, first, there is the brain is there and the recognition and eye is there. Okay. So first, what means with the help of the brain human being can recognize the system outside the world. Okay, so that is the brain the having is the importance for in the recognition system. Another thing is here for the visual perception, for the visual the human being basically used to use the sensory organs to gather the knowledge about outside the world. So what kind of the the sensory organ is there? So basic thing is here. The first if we talk about that, the hearing, with the help of the hearing, we can listen the outside the things, taste. Another thing is the smell is there, touch and eye. These are the the sensory organs by which we can gather the knowledge about the outside the world. Okay, but out of these five, which one is having the the most important aspect here? So here the answer is here, the picture, because someone has said the picture speaks thousands of the words. Yes, definitely image. With the help of the image, we can convey the more information for our reasoning. So that is the important thing is there. So here you in the background, you have seen there the recognition with the help of the brain and visual perception, where the image is playing very important role with the help of our eye, the sensor is there. Okay, so two things you need to understand here. And another thing is here, if we talk about in our daily life, we encounters the every day, we encounters the images every day in our daily life. So what kind of the images, what kind of the visual information sources we have? Basic thing is here. If you go back the 20 or 30 years back, so you we have definitely the painting was there, photography was there, then gradually it increases, and the nowadays we are having the digital image and digital libraries, and we are taking the snaps of the each and every moment. Okay, and also we have seen there a lot of the importance of the images in the medical image, because if in the medical image the doctor, with the help of the medical image, doctor can predict the the disease at early stage, and also the forensic imaging application. If we talk about the forensic application imaging techniques, so with the help of that we can find out the criminals. And also we have seen there this application is very much very much familiar for the satellite communication. Satellite communication means the remote sensing, where you have seen there the images are being sent through the satellite to locate the minerals present in the earth. So we have seen there the role of the images every day and everywhere in our daily life. So here you can see there different types of the the images are here, the given. So you can see here some mammographic images are here, for by which doctor can predict the early detection, lungs cancer here. And also the the reflective images the Lena images is there, where you can see there are different types of the noises is also they are having. Okay, so now at background view we are saying that the brain and images and our eyes sensory organ is playing very important role to understand. On the basis of that, I will discuss about the the computer vision and AI. Basically you have to understand the concept behind the AI and computer vision. What is this? Why we are talking about the AI? What is the AI is there? So what in the picture you are able to see here? The brain is there, eye is there, and keyboard is there. On the basis of that, if we talk about that, if we say in the simple words, AI enables the computer to think. By with the help of AI computer can think. And what is the computer vision? Basically, computer vision has the reference with the eye, where the computer vision enables the computer to see, observe, and understand. These are the very important things you need to understand. This is very small thing. This is very thing is the familiar thing is there. The AI enables the computer to think. That is the point is there. And computer vision enables them to see, observe, and understand. The 3 points you need to keep in your mind. Okay, because I will use these 3 points during my lecture. Okay, so here basically on the basis of that you can define the computer vision. This computer vision is basically this is the subfield of the artificial intelligence to derive the meaningful information from the digital image and other visual inputs. That is the important thing is there. What is that? The computer vision. Basically, the computer vision is a subfield of the AI. Okay, that enables the system. What is the purpose of the computer vision? Basically, the purpose of the computer vision to extract the meaningful information from the data. Data, what kind of the data you can take here? You can take the images, video, and other visual the inputs. Okay, so that is the important points is there. Because on the basis of the the extracted information the recommendation can be done, and according to that, according what kind of the information we have reached, according to that the the action can be taken, or the recommendation can be done. Okay, so this is the some fundamental about the computer vision and AI is there. And also if we combine the name of the computer vision and artificial intelligence, so that's name is here the artificial vision. Combining it is called as a artificial intelligence is there. Basically it is having the two parts is there. The first one is the machine vision and computer vision is there. So what is that? You can see here, means in the picture, the artificial intelligence is there, machine vision, and finally the computer vision. Computer vision is what? Basically means computer vision, already I have told you, means mimic the human visual system for understanding. Means what is that? Means to see, observe, and understand. That is the 3 points is there for the computer vision: to see, observe, and understand. And the machine vision is what is that? Means where the machine vision and means the hardware, means computer vision and hardware is combined. So that is the machine vision is there. So basically machine vision includes many aspects such as lighting, camera, part of the implementation as the industrial. Means basically the computer vision plus hardware that indicates about the machine vision. So machine vision, the simply can be means say here, means the automated visual inspection system. So that is now it is clear about the means the artificial, what is the artificial intelligence is there, what is the computer vision, and what is the machine vision. I am coming here, means next point is here. You can see here for understanding the role of the AI is there. You can see here the set is here the AI and subset is the computer vision, and computer vision subset means here the subset of the computer vision is here the machine learning, and deep learning is the subset of the machine learning. Okay, so I will talk about that, and you need to understand the relationship between that, how it can be means understand. Okay, because these fields are very important and challenging field nowadays. First of all, before to means to understand more details about the computer vision, first we need to understand the simply means the comparison between the human vision system and computer vision system. Okay, so human vision system is what is that? In the middle you are able to see the brain. Okay, and brain is associated with the eye. Now in the picture you can see, means a picture is having the means the fruit, means a plate of the fruit is there, means the different types of the fruits are there, means also is there, means the brain. So what is the role of that? Means eye, basically what is that? Eye is basically playing a sensor. Okay, what is the sensor? Sensor that measures the changes in the environment. Okay, means that is the responsible for capturing the image of the environment, because mean in front of the eye there is a plate of fruits is there. Okay, so eye is here means able to capture the images, means it is playing as a sensor. Okay, and now means it is being sent because the eye is connected with the brain, and now the signal is being sent to the means the brain, and now brain is interpreting. So interpreting device for responsible for understanding the image. Like that means now means with the help of the brain and our eyes, we can say that there is the bowl is there. In the bowl, there are orange is there, banana is there, lemon, and etc. Okay. In the similar manner, the computer vision is here. What is this? Means in place of the eye, means sensing device has been placed, and now interpreting device, anything is there. Nowadays microprocessor and microcontroller device may be there, because the means microcontroller device means the whole microcomputer on a single chip. Okay, so that is the interpreting device is there, and with the help of that means we can also means classify and identify the things. Okay, so now means here means the goal of computer vision. We now need to understand about the goal of the computer vision. What is the goal of the computer vision is there? So our objective is first to means prepare a program, means by which means that computer can interpret the image. Okay, what kind of the images are there? So first we need to write a program. What is the program? Means what is the program? Program is nothing but it is a group of instructions. Okay, and that is written based on the algorithm. What is the algorithm? Algorithm is the step-by-step solution. Okay, and what is the instruction? A command in binary that can be recognized by a computer to accomplish the task. Okay, so our objective is here first to prepare the means the program by which means the computer can interpret the image. Okay, so here the second objective is here to gain the high level understanding. from the digital image and videos, I will discuss about there are three types of the basically the levels are there, the low level, middle level, and high level. Basically, whenever we are talking about the high level, means programming is there. So basically, it will deals about the understanding. Okay, so here means our basic thing is here means to see the the offset. Okay, so these are the very important things, and means by whatever the program is being prepared with, our objective is main thing is here to extract the information. Information is what? This is the the features. Means suppose if I want to tell about the this image, so first our objective is here to extract the feature. What kind of the features is this particular image is having? Suppose I want to tell about a particular person, how that particular person looks like. So what is that? The features is there. So our objective is here to tell to anybody, means first to need to extract the useful information in the form of the features is there. Okay. So basic thing is here, means here there is the images there, means I have taken this one. So basically up to now, you can understand basically computer vision deals with the analysis of the image content. Okay, so analysis of the image content means means here you can see means what is that in this image you can observe means first of all you can see the stationary and non-stationary object is there. After that, on the basis of that, if we classify the computer vision work, the first thing is here, the object classification. Means object classification is what? Means what broad categories of object in this photograph? First, you need to understand for the object classification. And second thing is here, if we talk about the computer vision work, the object identification. Means object identification is what? Means which types of the given object in this photograph. So basic thing is here for object identification, you need to understand and you need to tell about that, means which types of the given object in this photograph. Means stationary is there, non-stationary is there. If stationary is there, so man is there also. If non-stationary is there, so man is also there, car is there. Means whether this is the living or non-living. What is this one? With different types of the question can be raised here. Another thing is here for the analysis of the computer vision is the object verification. Suppose you have prepared a program and with the help of the program, means any object has been extracted from the image. Okay, so then what is that? Means this means the object verification will deal about that is the object in this photograph. Means it will work as a verification. Means it will do as a verification. Means what is that? Is the object in this photograph. Means that is the important point is there. Means what is this one is here. Means you need to understand. And another point is here the object detection. Where are the object in the photograph? That is the another point is also there for the computer vision object detection. And object landmark detection is also the another important point for in this image. What are the key points for the object in the photograph? So that is that another point need to be noted, object landmark detection. And another point, if you talk about that, the object segmentation. Say the segmentation is basically to find out the reason of the reason of the interest. Reason of the interest is here means whatever the person, truck, car, traffic light, and each and everything has been extracted now. So that is the segmentation point is there to extract the reason of the interest. And another point is there the object recognition. Object recognition means what? What are the object are in this photograph and where they are. So these are the very important point for means and such types of the question can be raised whenever the computer vision deals with the analysis of the image. First, the object classification. Second is the object identification. Third one is the object verification. Fourth one is the object detection and object landmark detection, object segmentation and object recognition. So these types of the query will be solved. Means whatever the means the program or whatever the content I will take, this one is here. So these are the computer vision work is there. And here now you can see there the face detection, some examples is there for the computer vision is there. Now you can see here a smile detection is there. Here, vision-based some biometric is there. Some examples are there. And login without a password is there. So there is some another content is there. And now the applied fields of the AI. Okay. So these are the some AI fields is there. Expert system is there. Natural language processing, robotics, computer vision, computerized speech recognition, and machine learning. So now our objective is on the computer vision. Okay. So now they need to talk about the component of the computer vision. Okay. So basic thing is here, you need to understand the computer vision. On the basis of that, this computer vision is having basically the three component. Important thing is there. The first one is the image processing. Second one is the image analysis and image understanding. So here now I have pasted here the matrix. On the basis of that, the matrix, on the basis of the matrix, you can understand here. Means if for the, suppose if you want to define the digital image processing, if the input has been taken as an image and also you are getting the output as an image. So what is that? That is the, means the digital image processing is there. Means there is the manipulation of the image is being done. Okay, means our objective is to improve the quality of the image. So that is here the image processing is there, digital image processing is there. And if we talk about the computer vision, means computer vision is here, means if you are taking the, means the input as the image and you are getting the some numerical or some analysis, you are getting some numerical data or you are getting some means the description. So that is dealing about the computer vision. And computer graphics is what is that? Means of computer graphic, means suppose if we have the, means the input as a description and we are getting the image, that is dealing with the computer graphics. Okay. And artificial intelligence is what? Means suppose input is giving as a description and output is also, we are getting the description, means some numerical analysis or something data is there. So that is what is that? That is the artificial intelligence is there. Okay. In the another term, basically, if we talk about that, the computer vision is related to as a construction of the 2D world from the observed 2D image. And computer graphics is just reverse of the, means the computer vision, where the construction of the 2D image from the observed 3D world. Okay, so that is the, means the computer vision and computer graphics both have the vice versa, and digital image processing is the means bridge between the computer vision and computer graphics. And overall the things are coming under the artificial intelligence. Okay, so here you can also understand the image processing and computer vision. What I have told you, basic thing is here. This is the manipulation of the image and process generate the symbolic data. Okay, now also you can understand here in the simple form is there. The input as a image is being taken, or means here the operation has been done some, and image also you are getting. And here image and image analysis, some operation is being applied in middle, and finally you are getting the output as a numerical data is there. Okay, so here the some computer graphics I have already told, this is the just reverse of the, means the computer vision. So basically the computer graphic deals with the generation of the 2D computer images from the description of the 3D data. Okay, so now what kind of the, means the aspect, means the image processing, computer vision and computer graphics is having here. So basic thing is here, we talk about the image processing, the image enhancement is there, means resolution is there, image restoration, computer means image compression is there, and image fusion is there. And for the, means that is dealing with the pixel to pixel. I will tell you what is the pixel is there. The smallest element of the image is called the pixel, and where means the dealing with the pixel to pixel, that is that coming under the image processing area. And computer vision is there, pixel to the symbol. So what kind of the area it is covering? The signal processing, pattern recognition, descriptor, machine learning, and data fusion is there. Computer graphics, what is that is here? Symbol to the pixel, description to the pixel, modeling, and augmented reality. Okay, so on the basis of that, means here you can see some means examples here. The example is there for the image denoising, compression, edge detection. Here computer vision with face recognition, object tracking. Okay, So here some example is there. So in the block diagram, the same thing has been discussed here. Here also now the overall the picture of the means the computer vision is here. So what is this one is here? Image acquisition is there. Means first our objective is to acquire the image. Okay. So image acquisition. So image acquisition will also have some sensing device. So that is because the sensor will measure the changes in the environment. Okay. So that is the important point is there. So that is the image acquisition is there. The pre-processing is what is that? Means basically the removal of the noise and enhancement of the image will be done in the pre-processing is there. The segmentation for reason of the interest need to extract, and the feature extraction and selection means extracting the features, means the attribute or characteristic, and finally means the recognition, means the comparing the criteria, and or we can say that judging point is there. Okay. So, and the machine vision is similar, because I have already told you, where means some hardware implementation also has been included there. So before the image acquisition, so that makes the machine vision system. So here also you can see there, means each and everything is the same with computer vision. So except there is some hardware means component also has been included here. There is the camera is there, illumination is there, and other some processor can be used here. So that is here the dealing with the machine vision system. Okay. So related fields, now you can see there the role of the means here the computer vision, means computer vision, machine vision, and image processing. They are having the vast means the fields. Means here also you can see there it is being surrounded by the artificial intelligence, control, robotics, signal processing, physics, imaging, neurobiology, mathematics, machine learning. So now you can understand the computer vision, machine vision, and the image processing is very important role means in the means you can say simply the computer in biology. That is very important point. This is very burning point for us. And now we need to understand the first component of the means the computer vision. Basically, the computer vision is starting from the image processing. Image processing is what is that? Image processing is basically the manipulation of the image by applying some operation for the purpose of extracting information in the form of the numerical analysis data or resultant of the image. So basically our objective is here in the image processing to improve the quality of the image. Okay, means manipulation of the image by applying some operation. Some operation, I am telling about the some operation. What kind of the operation can be applied? Basically, there are three types of the operation. I will discuss about that. Means the basically the low level operation, middle level operation, and high level operation. Basically, whenever we are talking about the low level operation, where acquisition of the image, pre-processing, basically the pre-processing means the enhancement of the image and removal of the noise and compression come under the low level. And middle level, what is that? Middle level operation is basically the segmentation. Segmentation is what is that? Partitioning of the image into the sub-segment, and feature extraction. And feature extraction means the characteristic or attribute, whatever the features you have extracted, that features may be redundant. So there is the need of the feature selection technique. So that is also that in the segmentation, feature extraction and feature selection can be combined under the middle level operation. And high level operation means that is the pattern recognition, machine learning, deep machine learning area for understanding purpose or for mimic the computer vision. Okay, so that is the important point is here. You can see here there is the image is there. That image has been captured. Now here the camera is there for the means with the help of the digital image processing. Now the process has been done. Now you can see there means picture is means manipulated pictures you are getting here, means that more clear picture. A drop of water on the leaf you can see very clearly on the processed image. So such types of the means the thing we can deal in the image processing. Okay, so here now means basically before to come on the image processing, first we need to understand about the construction of the image. Okay, so how the image can be constructed, that is the important point is there. Means Because during the acquisition process, image may be corrupted due to the noise. Basically, noise is the unwanted signal is there. Okay, due to the misfocus of the alignment of the device. Also, that is the point is there. So different types of the things are there. So different types are, even the dust may be there, blur may be there. Okay, so different things are there. So what is that? Means our objective is here to understand the first, the means the image model construction. If we are able to understand the image model construction, then we can define accurately what kind of the particular image is having, what kind of the noise a particular image is having there. So that is the important point is there, because means the multiplicative noise, because the noise may be categorized as a multiplicative noise and additive noise. Okay, because the additive noise is follow the Gaussian distribution and multiplicative noise follow the means Poisson noise, Poisson distribution, Rayleigh distribution, gamma distribution. That is difficult to remove that. So that is here. First, our objective is here to understand the image model construction. Then I will tell you means what kind of the noise may be there. Okay. So here, what is that? Means the image model construction, basically the amount of light generated by the light source, that is the important, means the amount of light, means light source must be there, nature of the surface, and amount of light that is captured by the sensor. Okay, so here now the image has been taken from the internet, not my image is there. Okay, so now what is that here? This is the means the source of light, there is the bulb is there, and now the leaf is there. Okay, and now you can see here the eye and camera, means such types of the sensor has been placed for capturing the images there. Okay, so what is that? Some terms you need to understand here. So what kind of the terms you can understand? The first, the luminous flux. Okay, light generated by the source, that is called the luminous flux. And there is the 2 points you need to understand here, the illuminance and luminance. Okay, so illuminance is what is that? The amount of the means the light, or you can say here the amount of the luminous flux falling on the given area. Means here whatever the light is being generated by this bulb, it is falling on the this leaf. Okay, so amount of the means the light or amount of the luminous flux falling on the given area of surface is called the illuminance. Okay, so that is the point is there. And whatever the light is falling on this one is here, that light is reflected. That light is means reflected at per unit solid angle, and that is being captured by the IR sensor. Okay, so whatever the light is means reaches up to the means the sensor, up to the means the eye, means and that is called the means the luminance. Okay, so here means if we talk about the noise, means the sometime you have read in the paper the additive noise and multiplicative noise. Basically, during the acquisition of the image, means suppose means if image is having some misalignment or something having the dust or something, what is that? Some scratches is coming on the image. What is that? That is the additive types of the noise. Means during the acquisition of the image. And during the formation of the image, if some particular noise is there, if during the formation of the disarrangement is there, so that is called, means what is that? That is called the multiplicative noise. So you need to understand here, whatever the light is reaches up to the sensor and whatever the distortion is being created during this eye and reflected light. Okay, so that is what kind of the noise is there, that is the multiplicative noise is there. So different types of the filter in the image processing and computer vision area have been developed there for removing those noise. So that is the important point is there, because sometimes the researcher are only having that, means suppose I have taken the image and that image is having the Gaussian noise. Gaussian noise is additive noise, but not. You need to understand the formation of the image. Means suppose if you are going for the mammogram, suppose you are going for the X-ray. So there may be some there may be some problem with the capturing sensor. Okay, so that noise must be identified, and according to that means the filter can be developed, and then the multiplicative noise can be means the removed. Because the enhancement means the enhancement means what is that? Enhancement means the for improving the quality of the image. So that is our objective is here. Suppose if we want to extract the information from the image, so our objective is first to improve the quality of the image. So what is, if image is having some noise, so our objective is what? To remove that particular noise. So first our objective is to understand whether this particular noise is having the multiplicative noise or additive noise. You cannot blindly put there is the only the additive noise is there, only there is the multiplicative noise is there. Okay, so now the here I have written here the U is here, this is the observed image, and N2 and F is the original image, and N2 is the multiplicative noise, and N1 is here, this is the additive noise is there. Okay, means during the formation. But what is that? Means if the image is being formed, basically what is that image? Image is basically the 2D signal. Image is basically the 2D image processing is what? It is the extension of the signal processing. Okay, and it is what is that? If we want to define the image is the 2D signal that varies over the image, that varies over the x and y coordinates. Okay, so x and y coordinate is there, so that is here this is the function of the x and y is there, and I, what is that? IXY is what is that? The illumination at that particular point, and R is the reflectance or transmittivity at that particular point. Okay, so on the basis of that, you can see here, if 0 means what is that? If value means ranging from the 0 to 1, what is that? So 0 means indicate about the total absorption, and 1 is indicating about the total reflectance. Okay, so this one is here means total reflectance or total absorption. Based on that, we can also define this one is here. So basic two things is there, the illuminance and reflectance. Basically the image is the combination of the illuminance and reflectance. Basically illuminance indicate the I and reflectance indicate the R. Okay, so now you can see here the means the acquisition of the image, how it is being done. So basic thing is here, there is the source of light is there. So basically the energy is there. This is the scene element, basically the object. So object, what is that? Object is the target for which the image need to be created. Okay, so object is what? Object as scene element is what? This is the target for which the image need to be created. So now light is coming on the scene element. Now the imaging system is there. Definitely the imaging system will have some sensor device, and now it is being generated. So now in the digital image, so whatever the image or whatever the light is being captured by the sensor, that must be digitized. That must be digitized is what is that? That is having the means the property of the sampling and quantization. Basically the digitization can be done with the help of the sampling and quantization. I will tell you the further what is this one is here. You can see here now the images has been digitized here. Okay, so but before to understand this one, the imaging, so this the way of acquiring the image. The way of acquiring the image is also important to understand this point. How many way by which we can acquire the image? That is the important point is there. So here on the basis of that, we can define that the reflective imaging, reflective mode imaging. Second one is the emissive types of the imaging, and third one is the transmissive imaging. Okay, so what is this one is the reflective mode imaging. Reflective mode imaging is basically the simplest form of the imaging, means that use only the sensor to acquire the digital image. So here you can see here the first, means the source of radiation is there. There is the object. Object is basically the target, which is to be for which the image is to be created, and the sensor is there. Sensor basically a device by which we can measure the changes. So here what is this one is here, means whatever the light is being coming, and that is being captured by the sensor. So in the reflective imaging mode, you can say that this is the simplest form of the means the imaging techniques, and where only the simple sensor is being used to acquire the digital image for the digitization purpose. Means A to D converter. A to D converter basically means basically analog to digital converter is there. Basically it will have the quantization, sampling and quantization, and finally it is being stored in the digital computer is there. Okay, so this one is here, this is the digital computer. Okay, so now this one is here, this is the first types of the imaging techniques is there. So what is that, what kind of the images can be categorized under these types of the reflective mode imaging techniques? Why I am telling about that? For understanding the noises, for the pre-processing purpose, because the what is that, during the acquisition, noise may be corrupted due to the noise or the blur or something unwanted things is there, maybe there. So what is that, you need to understand the types of the acquiring of the image. So here they in the reflective imaging, all digital cameras, scanner, some that capture the images, simple means the digital images coming under this category. And emissive types of the imaging. Emissive types of the imaging, you can see here, there is no need of the source of light is there in the the emissive types. Emissive types of is basically the self-luminous object without the help of the radiation, where images are captured from the self-luminous object. Self-luminous object without the help of the radiation. There is no need of the the sensor is needed. There is no need of the source of light is needed. Okay, and now whatever the radiation is being generated by the object, that is being captured by the sensor, and now the sensor is further doing process for A to D, means where the sampling and quantization is being done, and finally it is being sent to the computer for the storage purpose. So what kind of the image may be categorized under this group for the emissive type? Basically the thermal imaging is there, means MRI is there, magnetic resonance imaging, and positron emission tomography, positron emission graphography. So these types of the image is having the self-luminous image. Basically what kind of this one is here, you need to understand about the, we talk about the thermal imaging. What is the thermal imaging? Basically in the thermal imaging, a specialized thermal camera can be used to in the low light situation for producing the object based on the temperature. Okay, and another is the transmissive types of the imaging techniques is there. Transmissive types of the imaging techniques here, you can also see here, there is the source of light is there, which depends upon the material of the object, because the some materials may observe the light and some material may pass the light is there. So what is the scenario is here, the attenuated radiation is sensed into an image. Okay, so that deals about the the transmissive imaging. So that deals about the transmissive. Simple wording you can say there, the attenuated radiation is sensed into an image. Okay, so whatever the radiation is being sensed, so that can be converted in the form of the image. So what kind of the image may be categorized under the transmissive types of the there? The example is there, the ultrasound image is there, example is there, X-ray image is there, another example is there, mammogram is there. So these are the example, means that is the important thing is there. So you need to keep in your mind, there are three types of the reflecting mode is there. So first one is there, this is the reflective type, second one is the emissive type, and third one is the transmissive type. Okay, so these are the description is there. Okay, so further means if we go here, you can see here the object is there, observe, sample and quantize, digital storage, digital computer, online buffering, display and output and record is there. Simple thing is there. Now the object imaging system, and here the A to D is being used for digital storage, digital computer, online also you can use the some networking. Okay, LAN, WAN, whatever the networking, now it can be sent through the the online or other things is there. Okay, so advantage of the digital image processing, what kind of the digital image processing? Basic thing is here, with the help of the digital image processing, the small correction can be done. Okay, and it is the the post process basically, and after the acquisition of the image, whatever the correction or for example the enhancement, for example is the improvement of the quality of the image can be done through the software, through the programming. So that is the advantage of the digital image processing is there. It is easy to store in the digital memory, yes, of course, because whatever the the image are there, that is being converted into the form of the digital, and also you can understand there. Our machine can understand only the means the binary thing is there. So that is the good opportunity is there. And also here it is possible to transmit the image over the network. Yes, of course. Digital does not require any chemical process. That is very important thing is there. So it is very environment friendly, and that is important thing is there. So these are the some advantage of the digital image processing is there. Now you can see here what is that the description of the digital image. So digital image is what is that? Basically, the image can be defined as a 2D signal. Okay, so 2D signal, what is that? That varies over the x and y coordinates. So you can see here there is the x coordinate is there, and there is the here what is that? There is the y coordinate is there. Okay, so there are the two things is there. And now the digital image, basically, because this is the 2D signal is there, so there that is having the x and y coordinate is there. So what is that? Means at here you can see here the dotted line. Here, dot is there. This dot, this dot, this dot. What is that? This is the pixel. Basically, what is that? The smallest element of the picture is called the pixel. What is this one is here? The point indexed by the row and column. Yes, of course, the point indexed by the row and column is called the the gray value or intensity value, because this particular pixel is also having this some value that is called the pixel value, gray value, or intensity value. So what is the intensity value? Basically, the intensity value is the sampled and quantized value of light that is captured by the sensor at that particular point. Okay, so what is this one is here? What is this one is here? Intensity value is the sampled and quantized value of light that is being captured by the sensor. That is captured by the sensor at that particular point. Okay, so here you can see here each point has been indexed by the row and column. You can see here. So now the image can be defined in the form of the matrix is there, and according to that you can draw the matrix. Here this is the 0 0 0 1 where and 0 m-1 is there. Now in such a manner, here this is the it is the combination of the row and column. Okay, so you can also understand here for defining the dimension of the image, you can multiply the row and column, by which we can calculate the dimension of the image. And quality of the the image number of pixels determine the quality of the image. Okay, so here here you can see here some digitization. Digitization is basically having the sampling and quantization. Okay, so what is this one is here? Sampling. Sampling means here you can first here you need to see here we have the analog signal. Okay, so now analog signal is there. So this is why what is the sampling? Sampling is a process by which we can convert the analog signal into a discrete types of the signal. Here the x the time coordinate is being discretized. Now you can see here the time coordinate is here. Firstly it was the t, and now it is being converted into the discrete form, t, 2t, 3t, 4t, 5t, up to like that t. Now this is what is that? This is the sampling process is there. And quantization is what is that? Rounding of the amplitude. You can see here now it need to be converted into the digital form. So what is that? What is, you can see here, if after the sampling, it has been converted, it is being discretized only the x-axis, not the y-axis. So second step is here, our purpose is to quantize the y-axis. The rounding of the amplitude means assigning the value. Assigning the value is what is that? This is the quantization. And now the coding. Now coding, basically the coding, Shannon, Fanon coding, Huffman coding, there are many coding is there. Okay, for transmitting purpose. Now with all these the analog to digital converter can be created. So you can see here also that there is the scene element is there. Now the sampling has been done, and secondly the quantization has been done there. Okay, so here, so you can see here the sampling, how it looks like the sampling. Suppose if we have the image, the Lena image is there. Now the dimension of the image is here, the 256 or 256 is there. And now you can see here, now sampling is being done. 6x4, discretization is 60x4. Means here, what is that? Discretization has been done. And now the subsampling, reducing the size of the image. Now you can see here, this is the subsampling process is there. And here, this is quantization, means assigning the label. So assigning the label means the value of the, means the each pixel will have some value. I will tell you what is the concept behind that to describe about the value of this one is there. So what is this one is here? Means here you can see the quantization level 256 or 256 is there. So now this is the 256 level. At 32 levels means here this is the 32 levels up to here, and here you can see. So now this is the second level, means now this one. So what is that? You can see here whenever we are means decreasing the levels. Okay, so there is some distortion you may observe there. So on the basis whatever the light is being captured by the sensor, means that must be digitized. Digitized means what is that? Sampling and quantization. Okay, and on the basis of that means the means false contouring and pixelization error can be described. What is that? If we fix the number of the pixel constant, means because for digitizing purpose the sampling and quantization is required. Keep in your mind. Okay, and what is that? Means if we are taking the pixel constant but reducing the quantization level, so what is that? Means it will refer the creation of the false edge, means outlines where the original means the image seen had none. Okay, so what is that? This will mislead about the false contouring. So what is that? Suppose a image means 8-bit is required to encode the monochromatic image, grayscale image is there. So there is the 8-bit is there. Suppose if you are using the 4-bit is there. So here means what is that? If the label is being means the distorted and thing, so you can see there what is that? 8 gray label is there now. The 4 gray labels and 2 gray label is there. Now you can observe the effect on the image. Okay, so there is, it will leads about the means the false contouring. This is the types of the error is there. Another types of the error is there. The pixelization error is there. Means if we are fixing the means the pixel and if we are means fixing the means the labels and decreasing the pixel, so also means that is called means also it will create the error. So these types of the error will called as a pixelization error or checkerboard effect. Okay, so here you can see here this is the 2x56 or 250x6. Now the pixel is being decreased. So now you can see there if the pixel is being decreased, so now the image is distorted now. Okay, so that is the point is there. So these types of the some concept is there for defining the some error is there. And what kind of the images, basically the image processing and computer vision deal about that. So basically the image can be described on the basis of the attribute, color, dimension and data types is there. Okay, so basically the attribute, if we're talking attribute means the characteristics. Okay, characteristics is what is that? This is the pixel based and means geometric. So pixel based means what is that? Means the smallest element is called for that is. So one thing is also you need to understand for the 2D image, the means the smallest elements is called the pixel, and for the 3D image, the smallest means the pixel is called the voxel, because the volume also has been included there. Okay, so what is this one is here? Means the attribute means whenever we are dealing with the pixel pixel based, so that is the pixel means for the processing is there, pixel processing is there, and there the group processing is there, and the means group processing and whole image processing is there, means the global processing is there. Okay, so that is here, that is dealing about the pixel. And another thing is the geometric. Geometric means what is that? Means some we are talking about the some geometrical description about that, means line, curve or something is there. So that is what is that? That is the geometric types of the image is there. Okay, and if we are further moving about that, the color image. So color image is what is that? Binary image is there, grayscale image is there, that is called the monochromatic image. true color image is there, and pseudo color image is there. Okay, so binary color image is what is that? Means the bit depth. First, you need to understand about the bit depth. Means number of bits required to import the pixel value. Okay, so number of pixel means what is that? If we are using only the one bit is there, one bit is required to import the pixel value. Basically, the 2 power n is there. The value of the bit depth is there. So if you are putting the 1, so what is that 2 power 1? Means 2 value you will get. So 0 and 1 you will be there. And also the 8 bit is required to import the pixel value of the grayscale image. That is the means that the value varies from the 0 to 255 is there. And what is that? And sometimes the medical image is also coming under the grayscale image, but means it may have the total bit 8 to 12 bit. So that there is some point is there. Okay, for the monochromatic grayscale image of that is dealing about the medical image, that will have the bit 8 to 12 bit is there. True color is basically having the 24 bit is there, and pseudo color is having the more than 24 bit is there. Basically bands is there, the 3 to 11 bands is there. Okay, so I will show you the example of those image. Okay, and what is here the dimension? Basically dimension the 2D image and 3D image. Okay, so based on the 2D and 3D image is there, and also the data types of the image is there. Signed image, unsigned image, float image is there, logical image is there, and double image is there. Okay, so these are the some data types of the images there. You can see here the grayscale image. So what is the grayscale image? You can see. So here, so grayscale image is basically the 8 bit is required to import the pixel values is there. So that is the grayscale value. That value will vary from the 0 to 255 because each pixel will have some values is there. By the image is basically means there is only 1 bit is required to import the pixel value. So 2 power n. So that value will there the 0 and 1 is there. And here the RGB is there. Means what is that? The means color image is basically the color image is the 2 color image is there. So basically RGB is there. Basically each component will deal about the monochromatic image, that is the 8 bit, 8 bit, 8 bit, and total combination is there is total 24 bit. So 24 bit is required to import the means the color image is there. And now here you can see this is the hands is there. Basically the pseudo color image is there. So there is band is total 3 to 11 bands is there, where means the means some artificial color has been means the included for defining the picture. So that is here the means the pseudo color imaging is there. So this is the example of this is the means the MRI image is there. Okay, so now you can see there the 2D and 3D image is there. So now some synthetic image also can be created is here. Okay, so here the 2D MRI how it looks like, and 3D MRI how it looks like is there. Okay, and here whenever we are working on the some programming purpose, so some programming purpose also the C programming, as we know about that. So there is some possibilities, the types of the image. On the basis of that we need to understand. So here the signed n-bit integer, unsigned n-bit integer, signed 32-bit integer, and unsigned 32-bit integer. Basically the range has been taken from the C programming. So basically you can define and you can defining for the experiment purpose. So that is the important thing is there. Now the synthetic image, basically the synthetic image is that the test image is there, simulated image. Neither it can be scanned or captured. Why we are creating the synthesis image? For the for purpose of the getting the reference image, because the reference image is what means where the image is having the means the denoised image, or you can say that the reference image is that image where means it can be used as a benchmark or test image. Means whatever the means the real time of the image, we can compare that. Okay, this particular the test image and the benchmark image is having the SNR value 1, means that it is the good quality of the image, and whatever the algorithm has been applied on the real time data set, and then you can compare there. Okay, what is this one is here, whether means whatever our algorithm we have applied on the image, whether means we are getting the good data, whether we are getting the good SNR value or we are getting the good SSIM, means the structure similarity index map. Means what is that? Means for the creating the parameter for the comparison purpose, we need to create the image synthesis, our benchmark, our test images there. Okay. And also I have told you about that the digital image processing, I have already told you, means the image processing is having the means the three, means the low level operation, basically the image acquisition, pre-processing and compression. Middle level operation, basically the segmentation and feature extraction, and high level operation, basically pattern classification, identification of the, means the image, means that is dealing about the classification and clustering. Okay. So here means what is this one is here some key stage in the computer vision. You can see here. So here it is starting from the acquisition. Acquisition, I have already told you what is that. Means acquisition of the image here. So what is that? This is the acquisition. So this is the image enhancement by improving the means quality of the image by applying the some filtering techniques or some enhancement algorithm techniques. Okay. So by which we can improve the quality of the image. Restoration, basically restoration means from the degraded image, from the means the means distorted image, and from where we need to retrieve the good quality of the image. So that is, and also it is able to enhance and sharpen the image. So that is that deals about the image restoration. So image restoration maybe the inverse filtering based and blind deconvolution based. Inverse filtering means what is that? Where we have the full knowledge about the object. Okay. So that is means that where we can apply the inverse filtering techniques by getting the means the retrieved image. And also if there is the partial knowledge about the object, so then we can use the means if there is no knowledge about the object is there, so then we can apply the blind deconvolution is there. Okay. And next operation is there is the morphological operation. Basically the morphological operation is the it is based on the set theory that accomplishes the task to extracting the information for the removing the noise, for extracting the region of the interest. For example, the erosion, dilation, erode is there, opening and closing is there. Okay. So basically the erosion means what is that? Means the dilation is what is that? Means the increasing the layer. Suppose there is a hole is there, and if we are increasing the layer, so by which we can fill that particular hole is there. So that is the means the dilation is there. And erosion, what is that? Erosion means the decreasing the layer. Means what is that? We need to open the in that particular hole, we can means the open more. Okay. So that is the erosion is there. And for example, for why we are doing all this example, for our objective is to derive or extract the meaningful information. Okay. So that is the important point is there. And then the segmentation, basically the segmentation will deal about the extraction of the region of the interest. Okay. And then object recognition with the help of the machine learning and deep machine learning and pattern recognition techniques we can do there. And here we can use the some classification techniques is there. For example, the means the means CNN. Suppose if we are using the CNN in case of the deep learning, suppose if we are using the in case of the machine learning, we are using the SVM, if we are using the linear discriminant analysis, if we are using the ANN, artificial neural network, whatever we are using there. And representation and description is also important there. Okay. So with the help of the some edges and half transform also we can apply here for connecting the distorted edges is there. And suppose if we want to send the image from the one place to another place, so that is the data must be means the compressed. So image compression can be applied here. So basically there are two types of the image compression techniques there. Broadly means first one is there. This is the lossless and lossy compression is there. So according to our data, we can apply and then we can transmit from image from the one place to another frame is there. If there is the some image processing application is there, so we can use the some image processing application is there. So these are the some key stages in the means the computer vision. And now the image processing task is there. Basically image processing task up to the segmentation and feature extraction. Okay. And And in the computer vision, what we are including? We are including the means object recognition, means for the machine learning, pattern recognition, and deep learning area. Okay, so if we combine the image processing and means the pattern recognition, machine learning, and deep machine learning, I mean understanding, so that is what that will create the means the computer vision. So there is the some techniques is there for the image processing up to the compression. Means what is the enhancement, restoration, edge detection, segmentation, and compression is there. Okay, so here image enhancement, you can see here now how it is being done. So basically to bring out the detail, simply highlight the certain features of the interest of the image is there. Image restoration is there. So here when the distorted image, from the distorted image has been retrieved, okay, based on the some mathematical or probabilistic model. Okay, so here some original image is there and some distorted image you can see here. There is some distortion due to the motion is there, and here original image, the noise has been included here. So basically these types of the some noises has been included here. There is the original image is there. The Gaussian noise looks like here. This is Gaussian noise is basically means the additive noise of the noise that is being created during the formation, during the acquisition of the image. Poisson noise is basically the multiplicative noise that has been that has been generated during the formation of the image, means during the capturing of the sensor. Okay, and salt and pepper are also, this is the additive noise. Speckle noise is the, this is the multiplicative noise during the formation of noise is there. Motion blur and noise in astronomical images is basically dealing about the this is the dealing about the additive noise is there. Okay, so image implanting, there is example is there. Okay, some application reduction of the speckle noise. Okay, so descapal image, some application image, the remote sensing image is there. Image segmentation, how it looks like, basically the region of the interest. Suppose if you want to select this region of the interest, so this region of the interest can be done. So these are the application of the segmentation. So basically means identification of the object in the image. Okay, and also it can be used for the cancer detection. Okay, Parkinson disease, Alzheimer disease, that is very important point is there. Suppose if we are taking the MRI image is there and a brain is having this sub abnormality, or objective is to extract the reason of the interest of those abnormalities. So that is the important point is there, to extract that particular reason is there. Okay, so here some means on the basis of the electromagnetic spectrum, we can define the some image processing application is there. Okay, so if we are talking about the radio wave, so there is the example is the MRI is there, MRI imaging is there. And some if we are talking about the microwave, so what is that? The radar imaging or remote sensing is coming under this one is here. Okay, for example is there the electron paramagnetic resonance that is being used for the measurement of the free radical is there. Infrared means thermal imaging, and thermogram. Visual light is basically dealing about the endoscope and dermoscopy is there. Ultraviolet light is basically dealing about this one is the biomedical imaging, astronomical or means here this is the laser images there. X-ray image is there, basically the X-ray image. Okay, that is being used for the chromosome analysis and bacterial colony. Gamma rays image, basically it is being used for the nuclear emission, nuclear medicine, finding teeny tracks in the metal surface. In the astronomical observation, you can see there the image processing and computer vision is playing very important role in each and every field is there. Okay, so that is the important point is there to understand is there. Now if we are moving further for the high level, means computer vision is what? To gain the high level understanding. So high level understanding means the pattern recognition. So now our objective is here to also understand about the pattern. Basically, what is the pattern? Pattern is basically the feature. Feature characteristics are attributes. Suppose if I want to tell about this picture, suppose means if I want to tell about the means there is the bold is there, if some means there is the eye is there. So how? First, our objective is to extract the feature, the characteristic. On the basis of that, that particular object can be described. Okay, so that is here. This is the pattern is basically there is the features is there. Okay, so for understanding the features is there, means the feature vector is there. Means what is that? Means whatever the the feature vector or pattern vector, you can say that. Means because the each feature, suppose suppose a data set is having the 100 image, okay, and from the each image we are extracting the total the 10 features from the each image, okay. 10 features from the each image is what is that? So whatever from the particular image, whatever the features is being extracted, that deals about the feature vector, okay. So here means that is dealing about the column, and there is the only one row is there and different columns is there, okay. And feature vector constitute the feature space. Suppose if we want to define the image, so already you have seen there, image can be represented in the form of the matrix, okay. So row and column is there. Basically row will deal about the number of images. So for the in particular experiment, how many number of row image you have taken. Suppose if you have taken the 100 image, okay, in for experiment purpose, so you need to define the row R1 to R100, and from each image, suppose the 10 particular features is being extracted. So what is that? Means so then you need to define x1 to the x10, okay. So x10, what is that? Means feature vector will constitute the feature space. So that is the important for that, for understanding the pattern recognition and feature vector space is there. So here you can see here basically what is that? The machine learning in pattern recognition. Means what is that? The beauty of the pattern recognition of machine learning, what is that? It can learn itself from the data. The beauty of pattern recognition of machine learning is what is that? It can learn itself from the data. So our objective is here to build a algorithm or build a system means that can interpret the system. It is just like as a example is there. Means suppose we are teaching to our child, okay. So first we need to tell about of the child is there. What is this one? This is the blue color, this is the black color, this is the what is color, this one is there. So you are feeding that data in our child. Oh, what is that? After that, whenever our growing child is seeing that thing is, oh, this is the blue color, oh, this is the the white color, this is the yellow color. What is that? This thing is here. Means our objective is here to build the system. So what is that here? That is the PR system is there. So what is this one? It can be the build with the help of the object or video or the feature. So that is here the beauty of the pattern recognition machine learning is what it can learn itself from the data, and there is the classification purpose is there. So here now you can see there male and female is there. What is that? The classification is what is that? What is that? The process of comparing the unknown object with the stored pattern to recognize the unknown object is called the classification. So what is this one is here? What is this one is the classification? The process of comparing the unknown object. Unknown object means what? Means you have already developed the the system. Means now the baby or something is able to recognize that thing is there. What is that? The process of comparing the unknown object or an unknown object, and there is the with stored pattern. So already in the mind there is the some stored pattern. Already there is in the system there is the some stored pattern to recognize the unknown object. So that is called the classification is there. So another thing is there for the recognition purpose, the process of assigning a meaningful label to unknown object. Yes, definitely the label must be put on the unknown object. Okay, on the basis of that, here the male and female pictures are there. So which one is the male and which one is the female is there. So that is the important point is there. So the process of assigning a meaningful label to the unknown object is there. Okay, so here on the basis of that, the classification techniques, we can apply the classification techniques. LDA, linear discriminant analysis is there. Support vector machine is there. Support vector machine also. The ANN is we are talking about that. Bayesian based classification techniques here. So different types of the classification techniques is there. On the basis of that, we can classify. Okay. So here, basic thing is here for classification purpose, we need to understand about that the learning process, the supervised learning. There are the three processes for the training and testing system. Because for developing the model, there is the training and testing. First we need to train the system and then testing process can be carried out. Okay, so what is this one is here? So they are the three various learning approach are there. The first one is the supervised learning is there, unsupervised learning is there, and reinforced learning. What is the supervised learning is there? Supervised learning is where there is the some supervisor is there already MAR data. If we are taking the already MAR data, there is the label is there. Suppose suppose if we have taken the means the mammogram image. So already in the mammogram is there, the doctor has already defined there is the this person is having the something the abnormality. So they have already circled there. So that is the MAR data is there. There is the labeling is there. See whether what kind of the cancer techniques is there, whether this is coming in the benign or malignant group is there. Okay, so benign is basically in the cancer. The for the the starting stage, and malignant is the lateral stage is there. So already this is the MAR data, and with the help of the our CADD system, means the computer aided diagnosis system, means the we are taking the supervised data, and supervised data means if we are whatever the doctor has marked, and after applying the our algorithm or our system, if they are creating the same area for the showing the abnormality. So what is that? It is working as a validation purpose. So what is that? There is it is doing on the supervised learning. Unsupervised learning means what is that? There is no supervisor is there. There is no marking is there. There is the no knowledge about the data. So on the basis of the similarity, means on the basis of the similarity, what is that? Okay, this types of the object can be grouped in the this types of, and this types of the object can be in this particular another type. What is that? Sometimes we are seeing the foreigner people. Okay, this people is coming from maybe the USA or maybe the Britain. Okay, these are the foreigner. And sometimes we are seeing the Indian people. Okay, this is the Indian people, this is the foreigner people. What? Why? On the basis of the some similarity measurement. Okay, we can describe, we can categorize on the basis of that. So that is there is the unsupervised learning is there, and reinforced learning is what is that? Means it is working as just like as a critics. Critics, what is that? Means critics means whether whatever you are doing, it is correct or not. Okay, means there is the teacher. Means that there is the evaluate. There is the, yes. There is the evaluator is there. Okay, so that is the reinforced learning is there. Okay, so what is this one is here? See here you can see the pattern classification. For the pattern classification in the computer vision area, the image acquisition, image processing, extraction of the feature data collection and pre-processing, feature evaluation, and here the main program is there. Okay, so what is this one is here? Basic thing is here, our objective is to extract the feature, but before the extracting the feature, our objective is to do the pre-processing. Pre-processing means the enhancement of the image and removal of the noise, and after that the segmentation or morphological operation is being applied there, and after that you will have the clear means the area, and the clear area, from the clear area you can extract the means the feature clearly. Okay, so and then after that you are applying the some machine learning and deep learning machine learning concept or the pattern recognition concept, and in such a way you means your system means can classify automatically. Okay, so here such types of the things is there, and now the some performance matrix is there for means for evaluating the classifier. So there is the some confusion matrix is there. Suppose the sensitivity is there. Suppose if you want to define the sensitivity, suppose if we want to define the specificity, suppose if we want to define the means the true positive rate, or if you want to define the Wooden index or something, what is that? So here what is that? For the defining the confusion matrix, means you can understand here. I think in the last lecture someone has asked about the sensitivity. So what is that? The sensitivity is here, means the confusion matrix is basically is used for determining the performance of the classifier. Confusion matrix contains the information about the actual data or predicted data. Okay, so what is there? This what is that? Means what is suppose means the actual data is there. This means this is the what is that? This is the predicted data. Suppose means the true positive. Suppose this is the TP is there. So what is the true positive? True positive means what is that? Suppose diseaseous person. Diseaseous person has been classified as a diseaseous person. Suppose the corona person is there. Okay, someone is having the corona, means whatever the antigen test is, what is that? Means antigen test may be misclassified. So what is that? Is if someone is having the corona and that our system is also defining as the corona, the disease person has been classified as a disease. So what is that? This is the telling about the true positive. And what is that? If there is the positive and negative is there, false negative is there, mean disease person has been classified as a, means what is that? As a healthy person. What is that? This is the false negative. And what is the negative and positive? False positive is what? A healthy person has been defined or categorized as a disease. So what is that? That is the false positive is there. And negative, what is that? Means healthy person has been categorized as a healthy. On the basis of the confusion matrix, you can see there in the means the machine learning and deep machine learning and pattern recognition, we can derive the different types of the means the performance measure. So that is the sensitivity, recall, hit rate. Basically, this one is what is that? Means what is that? This TP upon the overall, means TP upon the TP plus FN. Okay, means this TP upon TP FN. Okay, so what is that? It is dealing about the sensitivity or hit rate. Sensitivity is the probability that test will identify a person with the disease when in fact they do have the disease. Okay, so that is what is that? This is the sensitivity is there, hit rate or recall it is called. And for specificity is what is that? Means this is the, means what is that? Specificity is defining about this one is the false positive rate. So what is that? FP upon the FP plus TN is there. On the basis of that also we can define the accuracy. Accuracy means what is that? Mean TP plus TN, and this one is what is, this one is an overall the value. This one, this is the about the accuracy is there. Also you can define this one is the BCR, mean true positive rate plus TNR. Okay, and also you can define the measure, F measure. Also you can define the Wooden index. Also you can define the error rate. Error rate basically what is that? Here FN, FP plus FN. Okay, means the false rate you are calculating here, and that is the overall. So that is what is that? These are the some fundamental. What is that? This is the measure by which we can classify and evaluate. Now I am coming on the my topic, the application, medical application using AI. Okay, so medical application AI, basically I have already told you, means what is that? This is the means the computer vision is the sub part of the artificial intelligence, and artificial machine learning and deep machine learning is the sub part of the computer vision is there. Okay, so what is that? Means here you can also see here the artificial intelligence. What is this here? Computer vision, natural language processing, sentiment analysis, sound, machine learning and classical. Machine learning basically dealing about the supervised, deep learning, SBM, decision tree, reinforced learning, and supervised also dealing about the deep learning, PCA, principal component analysis, K-means, and clustering. Okay, and classical manual encoding rules and boundary condition, and the spectral ratio images and edge detection. Okay, coming on this area, this is the categories. And now means basic thing is here, means the in machine learning basically means what is that? The difference between machine learning and deep learning. In the machine learning means data need to be extracted manually. Okay, means what is that? User defined data. In the machine learning we need to extract data manually. Okay, so that is the machine learning is there. Machine learning means what is that? Means data must be means defined by the user. Okay, so that is the machine learning. In deep machine learning, what is that? Means where data will be extracted automatically. Okay, means suppose in the deep learning you need to only define the reason of the interest, and then we are applying the CNN. So then means data will, and then features will be extracted automatically. Okay, so here basically the training and testing is the important for there. Okay, so here you can see here there is the example is there. In the machine learning, the input is there, and here the manual extraction and feature selection. Means suppose we are extracting the feature, means feature, user defined feature is there. So whatever the features are defined, that features may be means the redundant. So that is the that is need to apply the feature selection techniques. And the feature selection techniques, there are different types of the feature selection techniques is there. Suppose the MR means minimum redundancy, maximum relevancy is there, t-test is there, f-score is there, Bhattacharya is there, t-test is there. Different types of the feature selection techniques is there for reducing the dimension of the features. We suppose whatever the redundant features, if you are applying for the processing of the data, definitely it will take time. So for the real-time application, it is not a good for that. So that is the features selection techniques you need to be applied there. And then you are applying the classifier techniques for classifying the object. Okay, so different types of the classifier techniques is there. That is the based on the statical classifier, non-statical classifier, and hybrid types of the classifier. Statical classifier is basically having the parametric classifier and non-parametric classifier. Parametric classifier is basically means the linear discriminant analysis support vector machine is there, and the non-parametric is basically the unsupervised, means basically the k means is there, k symbol is there, is coming under the statical classifier is there. Non-statical classifier is what is that? Non-statical classifier is basically dealing about the natural language processing area is there. Basically for the semantic types of the classifier, graph theory classifier, decision tree classifier is there, random forest classifier are there. Okay, and for the hybrid classifier, what is the hybrid classifier? Basically means the ANN, artificial neural network is there, fuzzy based classifiers are there. So these are the can be categorized under the hybrid types of the classifier. Basically, they are the three types of the broad categories of the classifier is there. Statical classifier, non-statical, and hybrid classifier is there. And now you can see here in the deep learning area, what is that? Here in the input and output is there. So now here only we are applying the after the the input image, you can extract this, you can apply the segmentation technique. Basically segmentation technique can extract the reason of the interest, and after the reason of the interest means the after applying the CNN or deep learning algorithm, it will extract the feature automatically and it will evaluate, and also it will reduce the dimension of the image. So dimension of the feature, and then finally it will classify the output. So there is some fundamental difference between the machine learning and deep machine learning is there. So you can also see there for your purpose of understanding the difference between the machine deep learning. So what is that? Less amount of the data is required the big amount of the data. Feature extraction always need proper feature selection. Self-learning, no need of the feature extraction. Accuracy, compared to the real, provide the less accuracy, provide the high accuracy. Require the human intervention for the mistakes. Do not require the human intervention. Can function on the CPU. This can work on the GPU. Okay. So here you can also see here. So deep learning process is there. Suppose if we are talking about the CNN, convolution neural network, in the short form, I am discussing about this one is here. So suppose there is the image is there, and here means the convolution is there, convolution pooling, and here is the full connected layer, and there is the softmax is there for the classification purposes there. So what is that? There is the convolution is being done, and convolution is being done, and then means first means the feature is being extracted, and then what is that? And then according to that, again the means convolution and again pooling and fully connected layer is being done for the classification. So how it is being done? What kind of the main important categories is there inside? So basically, first you need to understand for the convolution neural network, you need to understand first the 3 point is there. So there is the image is there. First, whatever the image has been taken. Suppose we have taken the image. So what is that? That is the having the 6x6 image. The dimension is there, and there is the filter size. Whatever we are applying the filter. And now the result means here means it is being we are getting here the value 2. What is basically the convolution is being done. Well, what is that convolution? The convolution is being done from the group of the means the input pixel group of the means the group of the pixel are being taken from the means the input image for the processing. So basically, there is the mask is there. Mask is basically the filter that is being applied on this particular image, and now it is being convolved. So how the convolution can be done? Basically, means you are getting the value 2. How it can be calculated? Means whatever the value, means here now you have the 3 cross 3 value is there. The dimension of the 3 cross 3 means this is the filter size is also the 3 cross 3, and here in the input also has been selected the 3 cross 3. So 4 cross 1 plus 9 cross 0 plus 2 cross minus 1, again 5 cross plus 5 cross 1, again plus 6 cross 0 plus 2 cross minus 1, again 2 cross minus 2 cross 1, again plus 4 cross 0 and 5 cross 1. Now what is that? What you are getting here, this is the value, this value. So now if you here the stride has been shown here, stride is 1. Stride means the shifting of the means the filter. Okay, shifting of the filter is what is that? It is shifting one place. Okay, so it is being shifted at one place. What is this one is here? Means it is shifting. So shifting one place is here. Suppose s is equal to 2 if you are taking, so it will place 2. Okay, so you can see here now the s is equal to 1. So now it has been shifted. So now it has been shifted, means s is equal to 1. You have taken this one is s is equal to 1. So now again now it is being calculated. Now the convolution is being done basically. So 9 cross 1, again you can see 2 cross 0 plus 5 cross minus 1, 6 cross 1, 2 cross 0, 4 cross minus 1, 4 cross 1, 5 cross 0, 4 cross minus 1. Okay. So what is that? Now it is the means now the next value you have calculated. Now this value is coming the means this is the 6. Now again you will move this filter. Now this you can move this filter. Now you can, after that you need to move this filter the downward and downward, and until you have covered all the value. Okay, so what is that? Suppose you can see here the input image is having the size 6 cross 6, but the resultant image is having the 4 cross 4. So what is that? There is the padding is there. Okay, so because the padding has been taken the 0, what is that? For calculating the output size of the output image, what you can do? Input size of the image. Input size of the image is what is that? The 6 is for the means first it is taking for the row. Okay, 6 and plus means suppose the padding has been taken the 0. So this is the 0, 6 plus 0 minus this is the 3. So this one is the minus 3. So 6 minus 3 is equal to 3, and plus 1 is what is that? This is 4. And again this is the cross and s is equal to stride has been taken 1. So that's why you are putting the s value is equal to 1 cross like that. So you are getting the 4 cross 4. Okay. So the resultant image is you are getting the 4 cross 4. So this is the means in such a manner in the convolution neural network, the means the size of the output image can be calculated here. Now the padding, why it is being done? The padding, suppose the padding is being done means 0, increasing the means the size of the image. Okay, size of the image for means you can see there means the edge point. For the means calculating the proper edge deduction, you can do here the padding, because suppose if you are moving this one, so this pixel are not being used. So that is the padding is being applied for fully connection. So this is what is that? You can see here, suppose the P1 is equal to 1. So that is what is that? 0 is being created. So 0 is being created. What is that? Means 0 means because the image in the form of the rectangle is there. So 0 0 0. So what is that? 2 layer has been increased. So 2 layer has been increased. What is that? If you are putting the 1 is equal to 1. So what is this one is here? Means you can retain the original image size. Okay, so original image size is what is that? You can put there means the padding value you can put. So padding value if you are putting is here. There is the means 2 means here. What is that? P is equal to 1. So that's why you can get here the means 2 cross 1. So that is the what is the padding value. Okay. So here means if you are putting the P is equal to 1, so it means the 2 times. It is increasing the it is increasing 2 times value, means the pixel value. Okay. So here means you can in such a manner you can retain the original image. So that is the some few points about the CNN is there. So now the further if we are talking about the means the CAD system, basically CAD system has been combined, has been constructed with the help of the computer vision and artificial intelligence. So basically computer vision is having the pre-processing, noise reduction and enhancement. Segmentation means basically the region, age and structure. And here the region of the interest from the size, location and texture, that is coming from the computer vision area. And artificial intelligence, basically this is the classification, feature selection and classification has been done. So basically computer aided diagnosis system, basically that is in combined, the short form is there, is the CAD is there. Basically there are two types of the CAD is there, means CAD means detection and diagnosis system. So computer detection is, that is the CAD is there. CAD is there. What is this one is there? Usually confined to making the suspicious reason. So suspicious reason is what is that? Reason of the interest is there. So you can see here. So there is the means the abnormality on this one. The X-ray image you are able to see here. You can see there is the CT image, and also you can see the MRI image is there. Okay, so our objective is to find out the means the reason of the reason of the interest. We need to find out the abnormality automatically. Based on the abnormality, our system will define whether the particular image is the means the having the abnormality or not. If it is having the abnormality, so what kind of the abnormality it is having there. So here the CAD pipeline is there, the pre-processing, segmentation, feature extraction and classification is there. Okay, and nowadays this is the means what is that? This is the machine learning based. Okay, so there are two types of the CAD system can be defined here. So you can see here deep learning based. What is that? Suppose the breast cancer is there, so there is the database is there, mammogram and ultrasound database is there. Now a CAD system. So what is the CAD system is there for the based on the machine learning. So basically the segmentation, enhancement, the pre-processing, post-processing, coming the feature extraction, feature selection and classification. Okay, so basically detection and diagnosis. So system has been divided into the two groups. The first one is the detection, that is the CAD-E, and diagnosis CAD-X. Okay, so here is the diagnosis system is there. So there is the pre-processing area and post-processing. First, our objective is to collect the image. Okay, so collect the image from the database. So we have the different types of the database for the mammogram and ultrasound images there. Okay, and now it is being developed, the CAD system for with the help of the machine learning. So pre-processing, and in the pre-processing you need to do what is that? Enhancement of the image and removal of the noise. Segmentation you need to do, and feature extraction. Here the features will be extracted manually, and if the feature is having the redundant value, so there is need of the feature selection techniques is there, and then you are applying the classification techniques, and then evaluation matrix. Already I have told you about the means accuracy, precision, sensitivity and F-score. You can derive all these performance measure from the confusion matrix. Okay, and now if we are talking about the deep learning algorithm based, what is that? Means you can, what is that? You can what is this one? You can directly you can take the image and you can already apply this one, the deep learning. Suppose if you are means applying on the overall the image, only the deep learning, so it will extract the overall the features. And suppose if you are doing the segmentation, and after segmentation means first if you are defining the region of interest, and after segmentation you are putting the deep learning algorithm, so then what is that? It will extract the feature automatically and it will means classify means accordingly. So nowadays we are using the deep learning concept is also there, because it can be means apply on the large data set. So there is some pro and cons of also there for the deep learning is there. So need to the better of the research is there. And now means based on the development of the CAD system, design and development of the CAD system for early breast cancer detection. So what is that? So for early breast cancer detection is what is there? Means here you can see there. There is the mammogram noise is there, and these types of the noises is having the some abnormalities. So abnormalities is basically what is that? Cancer is what is that? Cancer is basically uncontrolled growth of the cell. And in the breast cancer, if we are talking about in the breast cancer, deposition of the calcium inside the breast. Okay, so you can see here the calcium is in the form of the scattered form. Okay, so calcium will means lead about the some cancer is there. So suppose what is this one is here. So there is maybe some mass is there. So means a tumor and mass is what? A control growth of the cell. Means that there is some lesion is there. Okay, if some lesion is there, if some tumor is there, what is that? So that can be means defined as a abnormality. Okay, so on the basis of means the structure of the tumor, means we can define the malignant and benign cancer stage. So basically, benign is the means the starting stage, and malignant is the lateral stage of the cancer is there. And if the mass is the round or the circumscribed, so basically it will deal about the means the benign. And if we are getting the unstructured, just like as the omelette types of the structure, so what is that? That will deal about the means that will deal about the means the malignant types of this there. And microcalcification is what is that? Microcalcification, the micro and macro. Smallest teeny scattered form of the calcification you can see here, that is the micro, and macro is what is that? Some larger there is there. So that is the what is that? This is the some abnormalities is there. Architecture distortion, there is no definite mass is there. Means that is the architecture distortion is there. So that is the important point to find out. This one is the architecture distortion is there. So you can, on the basis of that, the malignant here, the calcification surface style, means on the basis of that you can see that means round, circumscribed, oval. Means will deal about the means if the tumor size is looking like this way, so then you can see that, you can say that there is the chance of the benign stage is there. And if you are getting the irregular, ill-defined, speculated, and microlobulated, so that is the malignant types, maybe the possibility of there is there. Okay, so how we can design the CAD system, because I have already discussed all the means the means steps or whatever means can be used in the computer vision and machine learning area. So basically, if we are going to develop with the help of the machine learning, not with the help of the deep learning. Okay, so firstly, if the machine learning is what is that? So first you need to acquire the image. So then you need to do the pre-processing. So pre-processing is being done for the removal of the noise and enhancement of the image. So you need to also recall the point whether the noise is having the multiplicative noise or the means the additive noise. If the noise is being added during the generation or during the acquisition of the image, so that is the additive noise is there. And if there is some distortion or some noise is being included or is being getting there, so what is that? That is the means the multiplicative types of the noise. According to that, we need to develop the means the filters. Okay, so there are different types of the filter is there. Fourth order filter is there. Total variation filter is there. Okay, so different local mean filters is there. Different types. The winner filter is there. Different types of the filters is there. So there you can apply the different types of the filtering techniques for the purpose of the removement of the means the removal of the noise, and then you can apply the enhancement techniques. For example, you are applying the CLAHE, contrast limited adaptive histogram equalization, means the histogram based equalization techniques you can apply there. So different types of the gamma histogram techniques is coming there. Okay, so new techniques is there. So also you can apply for the enhancement techniques is there. Gamma correction is there. So different types of this thing is there. Now you can apply the segmentation techniques. So different types of the segmentation is there. In the literature is there. Means the region-based segmentation is there. Basically, the segmentation techniques can be defined on the basis of the similarity and dissimilarity measurement. Okay, similarity, basically the region-based segmentation is there, and that is the also is there the manual segmentation is there, semi-automatic and automatic segmentation is there. Okay, and if we are talking about the dissimilarity means what is that? Means based on the means age detection is there, point detection is there, line detection is there. Okay, so some types of the reason of the interest means can be calculated based on the similarity and dissimilarity. basically objective is there to extract the reason of the interest. Basically, what is the reason of the interest in here? After seeing the abnormality, or objective is to extract the means the abnormality. Okay, so once you have means mark the reason of the interest and you have segmented that particular area, then now you need to extract the features. So different types of the features can be extracted. Okay, so different types of the features, for example, you can extract the means histogram based feature, you can extract the geometric feature, you can extract also the wavelet based feature. Wavelet is basically the transformation technique, but also can be used as a wavelet based feature techniques, because it can reveal about the information about the localization and frequency. And then the means the Gabor based feature selection technique. Gabor basically deal about the orientation and feature selection techniques. Basically, the feature selection techniques can be used for the purpose for the reducing the dimension of the means data, and then classification techniques you can apply. So now you can see here the mammogram has been taken, pre-processing, feature extraction, feature selection. Now the classification has been done, and here the normal, benign, and malignant. Okay. So here now what kind of the techniques have been applied? This is my paper. This is the pre-processing technique. For the pre-processing means that total variation based model has been adopted. Okay, means the pre-processing for that particular filter has been developed, and here the segmentation means modified fuzzy C-means segmentation techniques have been done. Okay, so basically the K-means segmentation and fuzzy C-means segmentation based on the clustering based segmentation. So K means what is that? That is the hard clustering. In particular element is supposed belong to a particular group. That is the K means is there. And fuzzy C-means segmentation, what is that? A particular element may belong to the different group. That is depend upon the strength of the association of the particular element. Okay, so that is here, that is the fuzzy C-means. So there is the modified fuzzy C-means has been used. Modified in the fuzzy C-means algorithm, you will see there is the Euclidean distance. But in place of the Euclidean distance, I have put there the mutual information, because the classification was there in the form of the scattered. So that's why with the help of the putting the mutual information, it can be calculated and accumulated. Now the hybrid feature selection techniques is there. Hybrid feature selection, different types of the features can be extracted. So basically total 88 features has been extracted. Histogram based, texture features, geometric, wavelet, and Gabor based feature. Now the feature selection techniques is there. Now the pattern recognition and classification of machine learning area has been done. Means whatever the data set was there, that data set has been divided into the 10-fold cross validation. Basically 10-fold cross validation is what is that? This is the K-fold cross validation is there. So basically this is the training and testing method is there. Suppose if we have the 100 data set in a group, so 100 data set in a group, what is that? So we need to create the 10-fold, means each fold will have the 10 data. And what is that? K-1 will be used for the training and 1 will be used for the testing, and it will run up to the K times for 10 times. Okay. And if there is some misclassification error will be calculated. On the basis of that, training and testing can be done. And another training and testing method is there. Hold out method is there. Hold out method is there. What is that? You can take the data set, 100 data set, and you can take 60 image for the training and 40 image for the testing. And resubstitution method is there. You can take 40 image for the training and 60 image for the testing. That is the resubstitution method is there. And now is the means what is that? Knife. Okay, knife testing is what is that? Means why knife training and testing, what is the each features? Means each data is being taken for the as a testing and testing. So definitely the knife training and testing method definitely will take means will give the much accuracy, but it will take much more time. So now the best out of those best is the 10-fold cross validation is there. Means what is that? You need to create the fold, and it will where in each fold K-1 will be taken for the training and 1 will be taken for the testing, and it will run up to the k times is there. So now on the basis of the training and testing method has been done, and now you need to, once the model has been developed, then you need to apply the any classifier techniques. Suppose the SPM you are applying, or the linear discriminant, ANN, whatever you are applying is there. So definitely it will create the groups of that, means the normal and abnormal image. So here you can see there some experimental purpose is there. Now this is the segmented image. Now this is the, after the pre-processing, the image has been segmented. This is the pre-processing for the cancer detection, breast cancer detection. Here that 88 features has been taken. And now this one is total in the MIS database has been taken for this experiment. So basically 332 images, from the 32 images, from each image 88 image is being extracted. So for particular one image, if 88 feature is being extracted, so that is, what is that? That is the feature vector, and feature vector will make the feature means feature space. Okay, so that is the matrix form is there, means here in at row places 1 to 320 22 will be there, and for column means 1 to 88 will be there. Okay, so there, and after that means the feature selection techniques have been applied, means genetic algorithm based or mutual information based some feature selection techniques have been applied for the reduction of the means the dimensionality. Okay, and then finally the performance, then finally the classifier has been applied. Then you can apply the any classification techniques, okay, for evaluation of performance analysis of the feature selection. And now the CADD system has been done. This is the computer vision based system is there. So now you can see here, this is the original image is there, and now this is the means what is that, the smooth image from the non-linear complex diffusion based filter, and now the high pass filter has been applied, and now you can see here the sharpen and unsharp masking. So with the help of, I mean, the pre-processing has been done here, basically applying the different types of the features. So basically, I mean, unsharp masking and crispening has been applied for to enhance or crispen the image. Okay. So after that, you can see here for means you have applied the enhancement techniques. Na? So what is that? Then you need to calculate the SNR value, means signal to noise ratio. If the SNR value is coming near about the 1, so that's why you can say that the image means whatever the algorithm you have applied on the image, it is your algorithm is better. Okay. So for that, this here you can see here the image, different types of the sample image, the 10 sample image has been taken, and this is the before applying the algorithm. Then means the SNR value has been calculated, and after applying the algorithm, then SNR value has been means calculated. Then you can see there is the means comparison is there, and also you can see there the proposed SNR that is being indicated by the means the red label, and it is getting better in each cases. So that's why you can say that means this algorithm is better. Now this data set. Now what is that? This Clahe contrast limited means contrast limited adaptive histogram equalization has been applied for this one, and now the segmentation is being done. So now before the segmentation process, before to applying the fuzzy Siemens segmentation, means the wave what is that? Wavelet is being done. Wavelet is basically is being done for the compression. Basically means when we apply the wavelet techniques, so there is the approximation coefficient and the detail coefficient. With the help of the approximation coefficient, means suppose there is the image, you can means draw in the 4 coordinates, means LL, LH, HL, and HS is there. So LL is basically the dealing about the low pass area, low pass filter is there. So there basically this is the dealing about the approximation coefficient, means from where you can retrieve about the 95% means information, and rest 3 means that is the detail coefficient that is dealing about the noise or something else. So there is means whatever that means the reason of the interest of segmentation techniques have been applied in the LL techniques is there. Okay, now the different types of the segmentation techniques, what is the means available in the literature review, the OSU segmentation, texture based segmentation, K means, fuzzy C means, and modified. OSU is basically means that is the based on the thresholding based segmentation techniques is there, means where you need to define the optimal threshold value, and texture segmentation based segmentation basically there that is dealing about the texture. K means basically hard, means hard clustering segmentation techniques is there, means where particular element can belong to a particular group. That is the K means is there, and fuzzy segmentation algorithm means particular element may belong to the different types of the group. Okay, suppose there is the ABC group is there, so particular group A element can belong to the B, C, D, any group and modifies such fuzzy segmentation. So there is the proposed segmentation techniques is there. Okay, so proposed segmentation is there. Basically, in the fuzzy segmentation, there was the if you see and go through in the segmentation, so there is the Euclidean distance is there. So Euclidean distance has been removed with the help of the mutual information, because Euclidean distance cannot apply here because the breast is here, the curvy is there. So that's why means here means the mutual information has been applied. So then we can see there, and also another thing is here. What is that? Also the lungs also we have calculated the same thing is there, and also we have seen and observed there this technique is performing better. And now there is also some discussion about the means the feature selection techniques after the feature extraction. Now you need to put the some feature selection techniques. So some feature selection technique, the feature score, relief, MR, and FCBR is there, SBM is there, t-test, Wilcoxon. And these are some feature selection techniques can be applied for the reducing of the dimension. Okay, and now how it has been done. So basically you can see there what is that means here means supervised and unsupervised has been categorized based on these feature selection techniques, that is the having the property. Now the table, the comparison analysis, here the means different types of the the classifier has been used. The net based KNN means k-nearest neighbor, discriminant analysis, artificial neural network, support vector machine has been applied. And then means firstly the relief feature selection techniques has been applied for all classifier, and then parameter has been calculated. And then I found that means what is that means with the help, if we are using the relief and applying the using the SBM, basically SBM is also having the 5 kernel, linear, radial based function, quadratic, polynomials, and multi-layer perceptron. Then you can say this combination relief, SBM, and MLP is performing better. And next MR filter has been also feature selection techniques also have been applied for all the classifier techniques. And then on the basis of these parameter, you can see there again the SBM MLP is performing better. Okay, this perception here also the fast correlation based filter feature selection techniques have been applied, and now you can see there SBM is performing better, but not for the MLP, it is performing for the SBM quadratic. So there is a slight change is there in the observation is there, and also GLFIP is there. Also you can see there now the KNN is performing better. Okay, so again the feature score, now feature score, the feature selection techniques and all the classifier has been applied on these parameters, and then I found that what is that SBM is performing better in the combination of the quadratic. Okay, and like this way means the different feature selection, all the feature selection techniques have been applied. So then on the basis of that, what we can do means we can suggest the best the combination best combination which one which feature selection techniques is need to be taken with the classifier. Okay, because at randomly we cannot use the any feature selection techniques, no? So that is the important point is there. Then we need to do the some otherwise this otherwise we cannot get the accurate values there. So that is the important point is there. And also the unsupervised also feature selection techniques has been done. So here you can see there. So there is some performance is there, and comparative analysis has been done is there. Okay, so on the basis of that, the CADD system has been done. Another thing is there, this self-learning prosthetic hand. Self-learning prosthetic hand means basically this is based on the AI and machine learning based this is the computer vision. Basically it is based on this one is here. Basically what is that it is coming under the machine vision area, basically because the hardware implementation has been also included here. because with this project, I got 44.8 lakh rupees from the from BIRAC. So this project was, what is that? Basically, there is the means escalator, means the there means what is that? Means the self-learning prosthetic hand can be attached on the amputee. What is that? So basically, means what is this one is here. So what was this one? What kind of the problem basically it was having? So basically means cost effectiveness, means whenever we are going to develop some model or some project. So what is that? What you need to do there? So first you need to means see there the cost effectiveness point. Second, the easy to use the prosthetic hand or whatever the hardware implementation techniques you are applying. The flexibility of using the all degree of freedom in the performing the action, means whatever this project has been done in the sense of the means the hand. No? So that is what is that. So then we have checked the freedom of the performing the action. Whatever, if you are going to develop some another means device, so according to that you need to take the advantage over the existing one. Delay associated with the available prosthetic hand. What was that? Means basically it was based on the EMG and other techniques. So what is that? So there is the chance of the delaying for understanding the brain signals. So what is that? So then for minimizing all these delay and freedom problem, what is that? We have means thought means what is that? Means we thought that means this is the means need to develop the means the self-learning prosthetic hand based on the machine learning. Means what is that? First, means suppose if we are means training the system, means suppose we need to train the system first, and once if you have trained the system and that it can be means attach any amputee and it can work easily. So what is that? So basically some proposed section is there. For the means the feature purpose, what is here means feature, action, voice and images is there. Feature has been taken. For the optimization purpose is there. Okay, so optimization techniques is there. Means here some optimization techniques can be applied, the learning algorithm, and now the training and testing. In such a way we can develop the model. Okay, so now some detail of the underlying technology for the solution has been done. So voice activation, computer vision, main database performance and interruption. I mean, these are the some hardware requirement may be used here. What was the use there? Okay, so these are the some project some view and glimpses. Okay, and thank you all. This is the reference what I have means discussed or where I have taken all these content. Okay, so this is the means overall this one. Okay, so thank you, Gaurav.
Thank you so much, sir.
Thank you.
Thank you very much, sir. It was a very informative session, and we are so grateful to you for sharing all the applications of computer in medical field. I would like to invite. Is there any questions from the participants?
Yes, ma'am. There are some questions they have asked. Madhvi asking that, sir, 3D are we using in medical image?
What?
1 second, sir. I have unmute. Madhvi, are you here? You can unmute yourself.
Yes, Madhvi.
Okay, next question belongs to the Rathis.
Yes, sir.
Are you here?
Yes,
you can
sir.
unmute.
Yes, sir. Yes, sir. Is it audible?
Rathis, your voice is not coming.
Okay, sir.
Next question belongs to Krishna Kumar. Krishna Kumar.
Yes, Professor. Good afternoon.
Yes.
Am I audible? Am I audible, Professor?
Yes, Krishna. Yes, please.
Yes, sir. Thank you, sir. Sir, thank you for the wonderful session, sir. And my doubt is how far digital image processing technique is effective in telemedicine field? Because nowadays, due to COVID, we should be decentralized, sir. So how far our technique is effective in telemedicine, sir?
Yes, of course. That is the good question. Basically, means why it is important for the telemedicine, and in case of the means the COVID. Suppose if you are going to design the automatic detection system of COVID and recommender system. Suppose if you are going to develop such types of the system is there. So whatever means you are taking the CT image, whatever the CT image based on the CT score, it can be defined. First means if means whatever the CT image, sometimes the wherever you are going for the CT scan, that image may be corrupted due to noise or something else, because that may not be always the good quality of the images there. So that is the main important thing is there. You need to apply the image processing and computer vision analysis is there, and automatically you can find out automatically. I mean the reason of the interest, mean the symptoms can be after the pre-processing, after the removal of the noise and enhancement of the image, means you will get the exact location of the abnormality. On the basis of that, it can be used as a second opinion of the doctor. Basically, you need to take the opinion of the doctor, but whatever your machine learning, means computer vision and AI-based system is defining about that abnormalities, it will help to assist the means the disease.
Yes, sir. Thank you, sir. But so as per your suggestion that patient has to get first prescription on a direct visit to the concerned doctor. Later on, if doctor believe means, then he or she will give telemedicine treatment for a nowadays pandemic. Is it, sir?
Yes, of course, because means whatever the means the information, because nowadays no one can move and directly can take the consult with the doctor. So that's why whatever the data has been generated, that may be stored means after the pre-process or after the enhance or whatever the means detection of there, that data can be stored in the some digitalized or in the cloud also. The server and cloud nowadays it is being used. Even the ministry also has offered such types of the project to do, means wherever you are able, even if you are means where in the Patna, if you are able in the Bhubaneswar or Delhi or Varanasi, somewhere else, the means doctor can, means the patient can directly talk to the doctor, and also there is the probability of that, means whatever the data, that data may be stored easily, and that may be accessible by everyone.
Yes, sir. So because of that, only in the year 1995 itself, we actually integrated primary health care center. But still, people are believing that by direct going to the hospital premises only, they will get cured. So that kind of belief they are having so far, sir. But
they need time, because they firstly means people are hesitating to take the online classes, but nowadays everyone is taking that. So that's why the scenario will improve.
Yes, so nature is like this, sir.
Yes, of course. This is the next generation.
Thank you, sir. Thank you for the wonderful session, sir. Thank you very much.
Thank you. Nice topic.
Yes, sir. So I just mute myself, sir. Thank you, sir.
Okay.
Thank you.
Another question from Mala Nehru. Are you here?
Yes, sir. Am I audible, sir?
Yes.
Yes, Mala.
Good afternoon, sir. Good afternoon, everyone, sir.
Good
It was a
afternoon.
very nice, informative, and elaborative session on digital image processing. So my question is, with respect to the image processing techniques, is there any difference in handling the 2D and the 3D images, sir? Say, with respect to image restoration or with respect to feature extraction, is there any difference, sir?
No, not basically. Basically, there is also means whenever you are using the 3D, so volume has been already included there. So means already you need to analyze with the Z axis. So what is the coordinate is there? So coordinate means you have seen there, basically the 2D image can be defined as the 2D signal that varies over the X and Y coordinate. But if you are talking about the 3D image, so that is there is the 3 coordinate will be there, means X, Y, and Z, and it will varies over the X and Y and Z. So you need to analyze all this, but all the application, all the feature extraction, and all the techniques you can apply accordingly.
Okay. And the features are going to be the same with respect to 2D and 3D images, sir?
Yes, of course. But volume will be included is there accordingly. Okay. Suppose if you have the 2D image and 3D image, so definitely according to that.
Okay. Okay, sir. Thank you, sir. Thanks a lot.
Thank you. Thank you, ma'am.
Another question from Bala. Please, you can ask. Bala sir, are you here? Bala Morgan? No. Okay. Anyone?
Sir, am I audible?
Yes, yes, Rajesh.
Okay, sir. I have a doubt in the projection of images.
Yes, yes, please.
Actually, I have message there. My question is that if you are using a tomography or something like that, how we are performing the scanning of that body part of a man, means
Sorry?
human. How can we scan the if there is a tumor in our brain, then
Okay.
how we are scanning that image or object using the image projection method?
Okay. Basically, means tomography images is there. Basically, you are talking about the machine area. So what is that? What you need to do there means basically the image processing and image means the computer vision and AI is dealing about the after the acquisition of the image, and means suppose means once you have captured the image, suppose you have acquired the image from the machine. After that means whatever the abnormalities or whatever the means some noise is there, some abnormality, some distortion is included there. So what is that you need to do means pre-processing is basically with the help of the soft means image processing is what is that it deals about the means the correction of the the abnormalities or whatever the things you can see there means after the acquisition of the image. Okay.
Okay, okay, sir. Thank you very much.
Okay, thank you.
Now, Aarti ma'am.
Yeah, good afternoon, sir.
Good afternoon, madam. Yes.
The session was very informative, sir, regarding image processing,
Thank you, ma'am.
additional image processing and all. Sir, coming to 3D image, sir, 3D image for breast cancer. I mean, 3D image
Right.
techniques which will be used for breast cancer. Is it implemented? I mean, are they now implemented in India, sir, or are we going with simulation type?
Yes, it can be implemented, ma'am. Basically, because this is the broad field and research of the matter of research is there. So many researchers are working in this field. So need to explore. Are you getting?
Sir, what my doubt was, are these techniques used in India, sir?
Yes, of course. Yes, of course. Why not? Yes. Because breast cancer is the second leading cause of the cancer after the lung cancer. So that is means if you see the survey out of 10 even means out of 10 each every one woman is having the means the breast cancer. So that is the that is very important and burning topic is there. And also you can whatever the means the algorithm and whatever the techniques you can also apply in the 3D image. Basically the basic thing is there when whenever you are acquiring the image and the 3D image, if you are acquiring or something is there. Okay, so what is that? According to that you need to do means what the means computer vision and means AI what can do, but creation of the 3D image is the another matter is there, no? So that is what is that it is being done with the help of the machine. Okay, so once if you have the machine, then you can apply the computer vision and AI techniques is there. Okay, our objective is what means AI means enables the computer to think. And computer vision is what? Computer vision means what is that? It enables the computer to see, observe, and understand. So see, it is starting from this see, observe, and understand.
Okay, sir. Thank you, sir.
Okay, ma'am.
Aarti ma'am, are you there? Okay. So thank you, sir. Thank you so much, sir.
Thank you. Thank you, Gaurav. Thank you, Ranjana ma'am. Thank you.
So I would like to request Ranjana ma'am to kindly provide the memento to sir.
Dr. Subodh, it was a wonderful presentation, very informative lecture, and nice to hear from you. And please accept the memento from.
Thank you, ma'am.
From
Thank you so much. Madam, I am here due to only you. So my best regard always to you. So thank you so much, ma'am.
So, Dr. Subodh, really nice hearing you.
Thank you, ma'am. Thank you.
And
thank you, sir.
You all are in such good positions that
Thank you.
fulfill my emotions.
Thank you,
I'm
ma'am. Thank you
highly
so much.
charged by that. That is what I want to see my sweet family of students.
Thank you,
I
ma'am.
really want to tell you that thanks to God that I am blessed by very good students. My PhD family is super.
Thank you, ma'am. Thank you so much, ma'am.
And
Thank you, ma'am.
you all are like my children, my own family.
Thank you, ma'am. Thank you, ma'am. Thank you so much, ma'am. Thank you. Thank you, Gaurav. Thank you, everyone. Nice talking with you, everyone. Thank you, Galgodia's families and Vice Chancellor and patron, all patrons. Thank you, everyone, for inviting me and giving such a wonderful chance. Thank you so much. Thank you, ma'am.
Thank you, Subodh.
Thank you.
So, dear participant, we will meet again, and we would like to invite you all for the wonderful session carried out by the hands-on session by the Kangkon Sarika sir at 2 p.m. So please, you all are requested to join with us after the lunch break at 2 p.m. Thank you all. Thank you. Welcome, Kangkon sir.
Hello, am I audible?
Yes, sir. Yes, sir. Actually, sir, most of the participants have not joined it because the previous session stopped at 1:30 approx.
Okay.
That's why they are taking the lunch. So
no problem.
Okay. So we can start after 10 minutes, sir. So please, I am inviting the participant to join. That's why please give me 5 minutes or 10 minutes, sir.
Okay, is my voice clear?
Yes, sir. Your voice is clear.
Okay, thank you.
In the meanwhile, sir, I am requesting you to please share the link of the software. That's why the participant will be able to download.
Okay, the software should be downloaded from the official websites. So I will mention that, and I can give one link to my GitHub repository in the chat box so that they can access the notebooks.
Okay, sir.
In the chat box, I have shared a GitHub link, so
Okay,
where
sir.
they can find the relevant data and notebooks associated.
Okay, sir. Sir, in this link, there are several type of software is listed. So which all the softwares should be downloaded?
Yeah, all softwares are not necessarily needed now. So if anyone, so while I'm demonstrating, if anyone can need that or anyone can need one alternative, then they can access that.
Okay, okay, sir. So we have approx 99 participants. Now we have reached 100. Okay, let's welcome Saikia sir. Mr. Saikia is MSc from the Department of Biotechnology, Guwahati University. He is currently working as a research associate at Bioinformatics Facility, Institute of Advanced Study in Science and Technology, Guwahati. He is Google Cloud certified professional and data engineer. He has co-authored several research papers in very reputed SCI Index journal. He is the member of several scientific society also. So I would like to request Saikia sir to continue his session. Thank you,
Thank
sir.
you. Yeah, thank you, Dr. Gaurav Kumar. So good afternoon, one and all. I would like to thank Galgotias University, Vice Chancellor Ma'am, and patrons for providing me the opportunity for a hands-on session on this FDP. Once again, I'm Kangkon Saikia. So along with my academics, I have been a freelancer, web application developer, mostly scientific applications, and a cloud architect for over 10 years. So as a researcher, I have also been actively consulting in areas of biological data analysis of mass spectrometric and chemoinformatic data, phytochemistry, design and development of bioinformatic pipelines, and database development, and also NGS analysis. So for this session, as entitled on the slide, I will be demonstrating various approaches for drug design, drug discovery, mostly in programmatic and big data approaches with data mining and structure-based designs. So I will be addressing various problems and ideas which can be solved by researchers with expertise in multiple disciplines. So before beginning of the session, I would like to mention that since I would be coding live for demonstrations, I would like to keep this session as an informal one with more of discussions. So without any further ado, let's get started. Okay, for convenience, I'm stopping this video. So let's see the universal steps on computer-aided drug design. So first, we have small molecule library against which from a target, these molecules are screened for its inhibitory or its active effects. Then we investigate specificity of the selected hits from the virtual screened targets, and then we perform again molecular dockings and molecular dynamics simulations and other quantum chemical calculations or other chemoinformatic calculations. Then we try to predict its virtually its ADMET properties, which is absorption, distribution, metabolism, excretion, and toxicity properties. Then we optimize the molecules by designing better molecules so that it becomes easier to synthesis for a chemist, or it becomes easier for biologists to perform experiments and carry out the future trials or future clinical trials or future experiments. So within these 4 steps, so there are several sub-steps involved where most computational expertise is needed, and most chemistry and engineering approaches needed. So for example, when we want to predict, suppose ADMET toxicity, so we need several data and several algorithms so that the prediction could be accurate, so that one drug can be designed and delivered at very less possible time. And in the optimization step, we need the state-of-the-art algorithms such as deep learning or GANs to design better molecules so that its toxicity or its some druggable properties can be optimized. So now, what are the interdisciplinary requirements in our case? So first of all, we need data sets. So every algorithm needs a very big data set, mainly for machine learning or deep learning purposes. So from several wet lab experiments across various laboratories, we are generating data, we are generating academic information, but there are very less places where we can find those data so that one researcher or one statistician or one computer scientist can take the data and generate algorithms or generate predictive models. Now next step is to development of algorithms. We need data flow pipelines so that once we submit a molecule or once we submit a protein, so that it goes through a particular pipeline and then generate results so that these results can be accessed by other researchers and processed accordingly. And next is we need more and more open source repositories. So there are various monopolies of this private sectors where a single software is very costly, and most of the time it is not affordable by various researchers. So in that case, we need more and more open source repositories. I would like to invite the researchers, those who are in computer science or mathematics or statistics background, to contribute towards open source repositories, which are mostly related to biological applications and biological softwares. Yes, and I also personally have contributed in many open source repositories. So next, we want collaborative research across various departments, across various labs and across various universities, so that one problem faced by one researcher in a particular discipline can be addressed to other particular researchers in other discipline. So for today's session, I have selected a very existing topic. So I will try to design a inhibitor compound for SARS-CoV-2 for COVID-19 virus. So this the slide shows slide shows the life cycle of this COVID-19 virus. So here I will explain various steps where we can design a drug molecule, and process it in a manner so that various. Okay. So first we have COVID-19 virus here. So in this step, this virus binds to ACE2 receptors, and this virus get endocytosed in the cell, and then this virus releases its RNA into the cell, and this RNA then get translated into viral proteins, and then this RNA is used to and again this RNA is used in the cell to make its replica, so that this can be packed into many viral particles and then finally release it from the cell. So here we have several targets where we can stop the virus from entering, or stop the virus from replicating, or stop its assembly, or we can expose it to human immune system. So in the first step, we have initial approach, where we can stop the host receptor, or where we can bind the protein to the host receptor so that the virus cannot enter through that receptor, or we can bind a compound to the viral spike protein, so that it cannot enter the host. So next approach is that we can prevent its ribosome assembly, and then again using the peptidase enzymes, so we can prevent this viral assembly and get replicated. And then in other approach, we can prevent the viral exit from the cell. So here I will be targeting this viral assembly, because in first step, so if we try to bind a compound in the spike protein or in the host receptor, so COVID-19 virus easily get mutated to that particular receptor, so that it cannot so that it becomes inactive to that particular drug. And next, in this assembly step, so where protease or where different protease enzymes are involved, so that 3C like protease, it is chymotrypsin like proteases. So here these regions are mainly conserved, so that if we design a drug to inhibit that particular compound, so there are more likeliness that this compound go inside the cell and inhibit that particular compound, so that it can stop the further propagation of the virus inside the cell. So this is my general idea for today's experiment. So now I'm sharing my browser. Okay, so I'm starting from a very beginning. So where first I would like after I have done several literature search, so I have found that there is a viral protein called 3C like protease. So this is the main protease of the SARS-CoV-1 and SARS-CoV-2 virus, which do the work of this viral assembly. So next, first we need to get the protein structure of this virus. So we have a website which is called PDB. This is a repository for protein data. So here we can download the various structures, protein structures, which are either produced from X-ray crystallography or cryo-electron microscopy or NMR data. So in this website, we can search for various structures. So I have specifically, excuse me. So okay. So specifically, every PDB entry is defined by ID. So in a literature, I have found that found a structure which have ID is 7BQY. So this structure points to a COVID-19 main protease in complex with an inhibitor entry. So we have found a structure which where already an inhibitor is attached. Entry is a peptide-based inhibitor, which means it has a chain of amino acids along with several small drug compounds. And this particular structure is produced by X-ray diffraction. So here we can find some general information about the structure. So for example, this is the ID of the publication of where this structure has been taken, or which publication this structure has been published. And again, we have general information such as this structure has chains. Chains means coordinates of some amino acids. It's in a series, so that which are folded in a particular manner, so that it produces a protein. So it has one chain A, which represents this SARS-CoV-2 virus, and then again with this chain, one ligand is attached there, which is entry. So here I will download the structure. Here it is 7bqi.pdb in the downloads. Okay. So first I will explain how the structure format is. I will try to describe how the structure is stored in this PDB format. Then we'll continue to view the structure. So this is the contents of the PDB file. So this header contains very basic information, then title of the structure, and these are various informations. And here we have down below. So these are the coordinates, these are the sequence of the amino acid residues. So these are the heteroatoms, information related to heteroatoms, so formulas, and information related to alpha helices, beta sheets. And again, so we have the coordinates of the atoms here. So we have X, Y, Z coordinates of the atoms with a particular amino acid residue by chain and this atom type and other various other relevant informations. So these coordinates particularly we need to describe the structure. So if we open the structure in a visualizer like PyMol, so yeah, if I try to open the PyMol, so here we have
Kangkon sir,
yeah,
your screen is not moving, sir. Actually, it is stuck on PDB.
okay.
So still PDB is showing.
I should show all windows.
So this one 7B2Y.
Yeah, is it visible now?
Yes, yes, sir. So can you please, sir, repeat, sir?
Okay.
Because all the participants have not able to view the process.
Okay. So after I have downloaded the structure, so I have opened the structure in a text editor. So this is how a structure is stored. But initially, this header part describes very small information about the protein. Then we have title and we have other relevant information. So these are not necessarily, these information are not necessarily regions needed. So these are basically explanatory informations about the structure. And there are some remarks also. Okay, after the remarks, we can get their sequence residues information, their heteroatom information. Heteroatoms means the atoms which are apart from the amino acid residues. Okay, so now we have in every atom entry. In every atom entry, we have mainly we have these X, Y, Z coordinates of the atom and particular atom name, and here we have the chain name and we have the residue name. So these four things are very important. So next we have this here this column describes the amino acid residue numbers, and then if we see that we have one particular chain as described in the PDB structure, and then after that in down below in the bottom we have two molecules of water, and okay, along with another one particular chain named C, which describes our ligand. So this will become more clear when we will visualize the atom, visualize the structure. So also we have connect records, which stores the information where which residue get connected to which particular atom. So here in this case, for example, in this case, for 1120 get connected to 2360 number atom. So this structure can be visualized in various kind of visualizers. For example, I will use here one PyMol, and another visualizers are also available. For example, Discovery Studio Visualizer or Maestro academic versions, or you can visualize the structure in Jupyter Notebook also by using a library called NCLView. So here I would like to fetch the structure directly from PDB, or we can open the structure, downloaded structure. So here I have downloaded the structure and try to open it here. So now we have this structure of our protein, and we can see that this is in the cartoon form. So this particular representation is called cartoon form. So if we try to view it in other forms, so that we can, if we try to view it in lines, so this is the particular structure. So we also can hide the cartoon form here. So these are the particular coordinates, and they are got connected by a particular by these bonds, and these red dots represent the water. So if we try to view the sequence here, which can be viewed in display sequence, then we can see that these are the amino acid sequences. So when I select a particular sequence and say this, so this somewhere it will get highlighted. So in my case, okay, let's try other sequences. Selecting. Okay. So here we can see the particular selected sequences get highlighted. So first, let us try to visualize, separately select this structure and visualize in cartoon form, and then we will go to the ligand and visualize in the sticks form. So first, I'm going to select the residues representing the protein. So here, I hope all the residues got selected, and then I'm going to rename the selection. So here I'm going to name it to, okay, say receptor. All right. So now we have one particular selection here, receptor. So here we have the receptor, and we can transform the so we can change the show as cartoon here. So again, we have the receptor in the form of cartoon. Now next, I'm going to select the ligand. And so ligand is comprised of this particular name. So A, V, L represents particular amino acid residues in the ligand. And then these are particular atoms representing the ligand. So here we have made the selection for the ligand. So now I'm going to again rename the selection as ligand here. Okay, now we have selection for the ligand, and I'm going to show it as lines. Okay. Okay. Now I'm going to make some adjustment to the display so that it becomes distinct. Okay, I'm going to change the background to white and display quality, maximum quality. Okay. Okay, now I'm going to apply some commands so that we can distinctly visualize the ligand. So I have here selected the ligand. Okay, so since I have named the ligand as lick, so I'm going to apply a command to change the color. So here in this panel, we can apply several commands which are listed in the PyMol documentation. And so first, I'm going to change the color. So okay. I have a list of commands. Okay. Let's see the documentation. For now, I'm going to color code manually. Like for example, using by spectrum and by element, I'm going to color code this. So this is the process of doing it without using the command. So here I'm going to color code first. I'm going to color code the receptor using, for example, by chain, by example, by alpha helix, beta sheet, and loop. So here we have beta sheets, alpha helix with distinct color, and we have the ligand here. So now let's see what are the residues it have within the 5 angstrom of distance. So let's I will use indicate by residues within 5 of let's ligand. So indicate so 5 of ligand.
Saikia sir, are you talking because your voice is not coming? So please, voice. So please let the step first. Speak the step first and before proceeding for the demonstration.
Okay, am I audible now?
Yes, yes, sir.
Yeah. So here I will try to select the residues within the 5 angstrom of the ligand. So this is the ligand here. So six color by element. Okay. And I'm gonna color the receptor distinct with ions. Okay, so this is the receptor, and again, this is the ligand here. So now I will try to select. We can select pirates. All within 5 of. Okay, so here. So now we have the selection of points which is within 5 angstrom of this ligand. So now I'm going to quickly rename this selection as now selection within 5 angstrom. So I have the points here. Now I'm going to transform it as sticks. Okay. Now, if I hide the receptor, so these are the particular amino acid residues along with the water molecules, which are shown by plus signs here. So these are particular amino acids within 5 angstrom of the ligand, which is represented in yellow color. So why I have selected is 5 angstrom, because 5 angstrom is the distance for any non-covalent interactions. So we can have van der Waal interactions, we can have non-ionic interactions. For hydrogen bond, we need 2.5 to 3.1 angstrom approximately. So anyways, so we have the selection of the residues was here. So now we need these residues in further virtual screening process. So we need to confirm here what are the residues and what are the residues number where this particular ligand interacts. So now in this selection, I'm going to quickly deselect the ligand and label the residues. So here, again, I'm going to deselect the waters also. Okay, and go right click on that and go to label and residues. So here we have the residues there. So these are the residues important to the active site or to the site where a particular inhibitor binds. And so now we have the information about the residues where a particular inhibitor binds in this particular protein. So now I'm going to prepare this protein for a virtual screening activity across a data set of ligands, and try to predict try to determine the compound which has most binding affinity in that particular site, apart from this entry which is the receptor already bound. So for that purpose, first I'm going to open the okay, let's first talk about the ligands now, where we'll start the virtual screening activity. Is my voice audible? Okay. First, I have opened the Jupyter notebook here, and then I have a library of compounds. So I have a library of compounds here. So these are actually downloaded from one of our in-house databases. So here in this compound dataset, we have a SMILE, which is the chemical representation for simplified molecular input system. And we have a compound class, which are flavonoids, which are main phytochemicals, which are known for their bioactive activities, and they are also antioxidants. And their compound subclass, we have name of the particular compound. For example, here delphinidine galactoside, which is a anthocyanin, and then we have their molecular weight and formula. So here the most important column is the SMILES. So I will open this data set in Jupyter Notebook and process these SMILES to produce a structure, and then we'll try to determine the 3D coordinates and minimize the structure for a virtual screening purpose. So for that, I would like to import. So here, I have already installed one particular library called RDKit, which is popularly known for chemoinformatic data analysis or chemical compounds analysis. So I have used pandas to use data frames and RDKit for chemical analysis. So also, let's leave it for now. So now I've imported that. Then next, I'm going to import this flavonoid dataset in a variable called fvdf, and then simply see the head entries. So here we go. So we have this SMILE columns: compound class, compound subclass, and name of the compound, and molecular weight. So what we need to do now? So first we need to convert these SMILES into the structure. So before doing that, I have also a list of compounds, which are referent compounds, which are already known as the inhibitors of that particular protein. So let's also take a look at them. So these are AMPRO inhibitors. Okay. So we have 10 compounds. Also, we have their SMILES and compound class names, molecular weights, and also formula along with its PubChem ID. So PubChem is, so those who are not familiar with PubChem, PubChem is a database of chemical structures and their properties or any other relevant information to a particular chemical. So here we can browse a chemical by name, or we can browse a chemical by similarity search. For example, if I take this SMILE, that says, for example, I'm going to take the SMILE representation of a drug called lopinavir and try to find its analogs or try to find the similar compounds. So if I query by the SMILE and hit enter, then I will find a list of compounds. So this SMILE exactly match with one particular compound, which is lopinavir, and also we have it similar to another 396 compounds, so another substructure of 10. So one thing we also can do is that we can download these similar compounds and try to dock these compounds along with, but this is beyond the scope of this tutorial. So anyways, so now I'm also going to import the referent drug dataset also, so which is named as. So I'm going to name it in a variable called ref drug df and see the entries. Okay, so now these are the compounds we have here. So if we see the dimension of both the dataset, we'll find that we have 268 flavonoid compounds, and 11 reference drugs. Now, first, we are going to create a dataset where we'll be taking the SMILES name molecular weight formula from both the compound, both the dataset, and also here I'm going to include one from the compound subclass. Okay, from flavonoid dataset. So from flavonoid dataset, I'm going to include the compound subclass. Okay, before doing that, first let's do one thing. The subclass should be uniform in both the data frames. So here I'm going to first rename the data frame with flavonoid. Yeah. Flavonoid data frames. Found subclass here. Type work. Okay, I have to state the columns keyword. Now we have this type, so I have to assign flavonoid DS. Okay, please tell me if my voice is not audible or it is broken. And again, I'm going to rename the reference drug dataset compound class to type. Import the variables. Okay. Oh, got an error. Type not index. Let me check the reference drug dataset. Type here. So I have type here. So now we have joined both the data set here. So next, I'm going to create a RDKit molecule instance from the list of molecules. So RDKit, for RDKit molecules, so we have, suppose if we want to define a molecule, then we can create a molecule instance by using this command camp.mol from smiles. So suppose if I want to paste one smile, say, take the first row for example. So like this, we can instantiate a RDKit molecule class. And then if we want to visualize the molecule, then we have to import one another library, which was, I believe, it was just IPython display. Yeah. IPython console. Import this module and then there we go. So this is the molecule constructed from the first row of this data frame. So now what I'm going to do is I'm going to create this molecule instance in a list and iterate over all the indexes in this data frame and append the molecule instance to that particular list. So for that, this particular block of code. So I'm iterating over every rows in that ligand data frame, then creating an instance of that instance of a molecule from the smiles. Then I'm going to add the hydrogens. So see here in this structure, we don't have hydrogens. So I'm going to manually add the hydrogens, and then after adding the hydrogens, we will embed the molecule into a 3D space so that we will provide the 3D coordinates of this compound. And then after providing the 3D coordinates of the compound, so we will minimize the compound. Minimization means we will place the compound in a particular force field so that its three-dimensional conformations can be calculated. So where here you will use a UFF force field. So, or we can also use the molecular mechanics force field, but we will use UFF for this purpose. So for advanced calculations, we can use other force fields such as Amber or such as OPLS or CHARMM or anything you want. Now I will check if it is not so. If it is not minimized, then we will just print a message that this particular compound is not minimized, so that we can increase this number of steps in the steepest descendant algorithm, so that this compound get minimized. And then we will compute the Castiglier charges in the particular atoms, and then we will finally append the molecule in the moles. So moles now get a very big list. Okay, it will take a while. Anyways, so after that, we'll try to calculate the molecular descriptors. So molecular descriptors means some descriptors of a particular molecule. For example, what will be its molecular weight, or what will be its solubility in a particular solvent. Say, will it be soluble in water, or how many rings does this compound have, or how many hydrogen donors the compounds have and hydrogen acceptors the compounds have. So like this, we have various molecular descriptors, and most important descriptors are which are needed for a structure-based drug design is called Lipinski's rule of 5. So there are 5 particular descriptors. So you can just do a Google search for Lipinski's descriptors. So you'll get an idea about that. I'm not going to talk much about this Lipinski descriptors. So here we have selected log P and calculates molecular weight and see the heavy atom count and how many hydrogen acceptors it has and hydrogen donors. Also, we have rotatable bonds. So this is the main thing, depending on which the docking will be carried on. And then we'll also try to see how many amide bonds present in the particular compound. Then also try to see how many rings are there. Okay, let us wait till this step finishes. So during this time, so let's see another thing. Now, the mostly available tool for docking based calculations is AutoDock. So here we have, hello. Yeah, here we have, this is the official website for AutoDock. So we have 2 AutoDock distributions. One is AutoDock 4 and another is AutoDock Vina. So both of them is based on Lamarck genetic algorithm. And difference is that this AutoDock Vina uses a simplified scoring function, and AutoDock 4 uses another scoring function, and the force field used in both the softwares are different. So here first question is why we need a force field. So when we put the atoms, say 3D coordinates of the protein or of a ligand in a molecule, so first we have to optimize the rotatable bonds in that particular, say in this structure, rotatable bonds in the structure in such a way that the fitting in the particular ligand through the non-covalent interactions should be optimum. So during this process, so we need a force field so that which defines the various interactions, such as attractions, repulsions, or many other parameters. So parameters, so how a metal ion will behave, or another heteroatom will behave in that in that particular system. So with that force field, we have a scoring function based on which we will calculate, we will determine a score on the folding of that particular bones, which are called the torsion, and finally we'll find the optimal pose and provide a score. So across the different compounds, so we'll compute the scores. So from that, with Autoduct, these scores represent the energy. So where minimum energy required will be the optimal fit. So we will try to get the minimum energy in the compound and screen for the compounds with the minimum energy. So here you can download the Autoduct Vina. Now in this tutorial, I will be using Autoduct Vina because it is highly suitable for virtual screening. Then Autoduct 4 you can use for most accurate or most optimal fitting of that particular ligand. So for virtual screening, I will be using Autoduct Vina. So you can download the Autoduct Vina from this website, and then install it on your system. So I have installed here the binary of Autoduct Vina over here, and add it to the path of this Windows subsystem Linux kernel. So if I try to type Vina, so it gets me the executable. So here there are various arguments which we need. So we need to provide a receptor in a particular format, which is called PDBQT. So we have downloaded the particular structure in PDB. Then we have to do some. We have to process the structure in such a way so that we can determine the charge on the particular atoms and torsions, and specify the atom types in this according to this particular format. So next, we can assign any flexible residues there. So for docking, we have two things. First, most of the task you will see in research paper is called rigid body docking, where we consider the receptor as a rigid body, and then we'll try to dock a ligand which has flexible bones and flexible torsions to that particular receptor. Next, we'll assign a space, which is basically a three-dimensional space represented by XYZ coordinates, followed by a center. Right. Then we need to assign these coordinates so that the algorithm will look at that within the scope of that bounding box. Next, we'll provide an output file, right, which will output different poses in PDBQT format, and also we will have a log file. So additionally, we can assign CPUs, so that how many CPUs will be used by the software, and then seeds, and these are exhaustiveness. Exhaustiveness is the parameter which is used to determine the accuracy of a ligand. So sometimes it happens that higher the value of exhaustiveness gives an optimum fit, but it is not true in every cases. And number of modes, how many poses we need is the output of that, and then energy ranges. Also, if we see the help, advanced help, okay. Yeah. So now, here we have several parameters which we can assign to our scoring function. So there are the default values are already set for these parameters which are stated here. And again, if we understand the scoring function properly, then we can adjust these parameters so that better optimal scoring or scoring according to your ligand dataset can be optimized. So here I have downloaded the source code of the file and modified the source code, so that I can generate some logs in such a way that these logs can be parsed as in cloud, or logs can be parsed in various log parsing tools like Logstash or Elasticsearch, so that when we get to screen a very large group of compounds and then we dump these logs into the cloud, then later we have several ETL pipelines which analyze these logs, and then we finally get the optimal compounds for several groups of proteins. Let's get back to the Jupyter notebook. Okay, this step has completed. So we have a list of moles from this step, and then I'm going to calculate the molecular descriptors using this descriptor calculator function. So I'm going to apply this function across every row, every index in axis 1 in the data frame. So it is going to take some time. Now we have the computed data frame. If we take a look at this. So we have this. So these are the values assigned by the data frame. Again, we should have the types. Okay. Now moving forward. So here I'm going to assign a new column called as lig ID, which will store the ligand ID of the compound, and so that later on we can save this particular structure into a molecular descriptor file such as mol2 or SDF, and then we will assign that particular ID to that file, and later we can carry out various experiments or docking virtual screenings or simulations through this dataset, and later we can rejoin that particular ligand with that particular ID with this metadata. So we have lig ID. So here I have renamed the index column as ligand ID. Anyways, while exporting this CSV, it will get converted to a new column. So now I'm going to export this dataset called as ligand dataset to a CSV file. Okay, now we should have this CSV file in the output. Now we have prepared the dataset. Now next we need the molecule in a particular format so that we can process it for virtual screening using Autodesk Girinath. So first we will export the molecule to SDF. SDF is a chemical structure descriptor format, so which stores the three-dimensional coordinates of a molecule. So here we can also use the PDB, but RD kits only support export of SDF or mol files. So first here I'm gonna first export it to SDF, then I am going to convert it to mol2 files, then mol2 files. Okay. So this is the location of the SDF file in the like dataset.sdf and like properties. So in this SDF file, we are also adding some properties along with lig IDs so that every column name is added to the SDF file as properties. I will show that in afterwards. So after I export the file. Let's take a look at the file. Output. Okay, link dataset.sdf. So here we have coordinates of the file. So these are the particular coordinates, atom types, and a header with the name. And then we have the several properties which are inherited from the data frame and added to this dataset. So this file containing whole dataset can be used anywhere for any drug discovery purpose or any chemoinformatic calculation purpose. So in next step, I'm going to import another library called OpenBabel. OpenBabel is a widely used library for molecular or chemical format convergence. So it is written in C. And I am importing here the Python extension of the OpenBabel library. And then I'm going to convert every single of the every single molecule in the ligand dataset to mol2 file, and which will be named by the ligand ID. So here I should have one ligand folder, otherwise it will throw an error. So we have already converted this. So let me back this up. I'm going to create a new folder. All the exports will be inserted here. All right, now there we go. So we have all the files in mol2 format. Now if you see the file, we can see its coordinates and various information related to it, but we'll not find other descriptors first which were listed in that pandas data frame. So now we can also visualize this mol2 file or SDF file using third-party softwares like PyMol, or we can use PyMol also, or we can use Chimera. So I think I should have Chimera installed. Chimera, or we can also use any kind of molecular viewer formats. So here I will use PyMol for this purpose, say. So just randomly select one molecule. So see, this is the molecule in three-dimensional format. Again, this molecule is folded in such a way that this energy of the molecule is minimized. So where this was done when we calculated, when we performed the minimization step. So now we are going to prepare this molecule for docking. Now, so for this purpose, we have to remove the non-polar hydrogens from this molecule and assign the gas decay charges in a PDBQT file. So for this, I'm going to use another Python script, so which is distributed with Autodoc. So if you go to downloads, and there are, okay, you will find that with MCL tools. MCL tools is another library which is kind of helper version for Autodoc. So yeah, MCL tools. If we download the MCL tool source code, then you will find a library there. So here I have got that in WSL home, and there info source code. Okay, so we have MGL tool source here and also Autodoc tool source here. So if we open this folder, then we have whole lot of Python script used in the Autodoc tools user interface. So I'm going to grab one script from here, which is called prepare ligand.py. So which will be available under utilities, I think. Yeah, prepare ligand.py. So I'm going to use this script. Use this script to prepare the ligands for prepare the ligands. So now what this script will do will it will convert this ligand in mol2 format to PDBQT along with some operations. So that such as it will add charges. So we have also options for not adding charges, but we will add Castigar charges, and we will add the polar hydrogens and whole lot of other options. So this whole Autodoc tools is written in Python 2.7. I will encourage any Python developers to contribute to this to this repository so that this can be developed or this can be upgraded to Python 3 modules. So that we can increase the compatibility, and we can also work on the user interface of this software. So first, I'm going to show the conventional approach. Then I will show the programmatic approach. So if we open the Autodoc tools, okay. This is the UI. So first, I'm gonna import the ligand. Was in the data, not in the data folder. Biology, notebooks, data. Sorry. Oops. Put and then ligands. And I'm going to change the format to mol2. So here we go. Let's take this compound ID 90. Okay. So as I have imported the compound here, so it has done some initialization to the compound. So it will prompt you a message. So where we have where it is showing that it kept the charges from mol2 file. So these charges were assigned in the step of minimization, and when we have added the Castigar charges in the RD kit, during the RD kit step. So it have found 24 carbon atoms and 21 rotatable bonds. Okay, so this is the ligand here. So now we can also do some manually other things also. So say ligand torsion tree detect root. So for those who not from biology or chemistry background, so torsion means this rotatable bonds. So here in the torsion tree, so root means the center. So from here we can see this one. This one is a rotatable bond, or this one is a rotatable bond. So now first add the charges again. It's like cross-checking once. So we'll go to edit charges, compute Castigar. So we have here two options: one Castigar and Coleman charges. So Coleman charges are used for proteins in Autodoc, and Castigar charges are generally used for ligands or small molecules generally. So here total Castigar charges are at 0.004. So now we have this ligand ready for docking. So if you compare the ligand here, so you can see that there are other hydrogens attached to these rings, but here these hydrogens are removed. So which were found non-polar by this algorithm. Again, why do we need this polar hydrogens only? Because polar hydrogens are only atoms which make polar contacts with the protein. So that will form the hydrogen bond. And again, another interactions are such as pi-pi interactions or van der Waal interactions. So in that case, we only need the polar hydrogens. So finally, we can save this ligand as PDBQT here. So we can export this as PDBQT in PDBQT format here. So let's save this first as a demonstration. Okay. Now, if we have a very large data set, say we have millions of compound, then this UI step is not feasible. So we need a programmatic approach. So for that, I have created one Conda environment with Python 2.7. So let's activate that particular environment. Initially, let's collect the receptors in a separate folder. We name the previous folder as docking backup. Okay. I have created an empty folder called docking. Now here I will keep the ligands. Which was in the output of the notebook. So ligands. Ligands, and in the ligands PDBQT folder, we'll create a new ligands PDBQT folder where we will store the PDBQT files generated. So we also need one particular shell script, which I have written earlier. Yeah. Prepare PDBQT ligands. Here. Okay, so this is a unique shell script. I'm not going to open it in text editor because some characters may change if we use the Windows platform to open a Linux script. So I'm going to open it in Unix shell. Okay. The files, then we can find the prepared ligand. So let's see the contents of the shell script. So some of you might ask, so why I have not followed the Jupyter notebook? Because Jupyter notebook written in Python 3.8 and this script has dependency of Python 2.7. Also, we can also do it in Jupyter notebook with Python 2.7 environment. Anyways, this process suits for me. So I have created a shell script. So where I have added variables. And the first one is log file name. So where I will store the ligand preparation logs. So I will assign the mol to dir. So which was I have assigned here as ligands. And next PDBQT dir. So where output will be stored. And the process will generate one dictionary file called liganddic.py. So another one, these are just removing existing files and creating new ones stuff. And then I'm going to print some progress during the process. And then, so here I'm using the, Yeah. So here I'm using the script. I might put a tab here. Okay. Now, if we execute this particular script, I think there are some errors. Prepare ligand for the PY. Okay. Now if we see the Python version, it's 3.8. It is not supported in 3.8. So let's see the Python. Yeah, it's 2.7.3. Now let's start. Let's clear the initial files. Here is the files. So it will gonna iterate over 300 molecules and prepare. So we can, if we write this in C shell or another, or say Python shell, so we have the scope to parallelize the process also. Again, a warning is coming up. So 51 atoms. So ligand 51 has some errors in atom. So I'm gonna simply ignore this for this demonstration purpose. So meanwhile, I will show how to prepare the receptor. So for preparation of the receptor, so we also can use another script from Autodoc utilities. But since we have one receptor, so I'm going to prepare it in the Autodoc tools. So let's delete all the molecules. Now I'm going to import. I'm going to read a molecule. Say PDB. Yeah. So this is the molecule. So first, let's take a quick look. So we have two chains as mentioned here. So in this chain, we have amino acids. And with amino acids, at last we will find HOs. So this means water. So we have to remove these all water molecules. So this is because there's the force field used in Autodoc or Autodoc FINA does not support the presence of water. So they cannot make calculations in the presence of water. So we need to remove the water. And next, we also need to remove the ligand from here. So in which pocket it will get docked. So if we select the ligand, yeah, can we see? So this is the ligand here. So we can also change the visualization type here. So say this. So this is the ligand. Now simply I'm going to select and first delete selected atoms. So first I have removed the ligand, then I'm going to remove the water. So now we have only the receptor. So another one most important thing I forgot to mention Is that so?
Hello.
Hello.
Am I audible?
Yeah.
Sir, how we can find the pharmacophore of a given ligand molecule?
How you can
find the pharmacophore of a given ligand molecule? Is it possible by the help of this tool?
Yeah, it is possible, but not using docking, but you can use machine learning based approaches. I will discuss that later in this demonstration.
Okay.
Yeah.
And when we choose the protein molecule, is it mandatory that we have to validate the protein structure?
Yeah, it is mandatory. It is most importantly, you can, if your grid dimensions are within the coordinates of the residues, so then you need to validate that particular residue. So if it is outside the grid, then validation is not needed for the particular protein.
No, for the validation, which tool we have to use?
Yeah, for validation, yeah, just wait for a minute. I'm telling you. First, let me show you that. So say we have this particular. Okay. So say sometimes what happens, so 1 or 2 amino acids may be missing from that. So we can add that particular amino acids using homology modeling. So you can use softwares like one software was there called WeedCode. But because I forgot the spelling of the code. Yeah, one software which is widely used is called CADD. Okay, and another one software you can use is MMTSB. So this is a tool set of some scripts. So here, so what you can do, you can analyze the structure. Say you can analyze if you execute the script, then you can analyze the structure. If anything is wrong in the structure, or after that you can fill the missing chains or missing side chains, or if any atom is missing there, then you can fill it by homology modeling, and then you can do a small restraint minimization and on the protein, then you can use it for docking. Hello.
Yes, sir.
Yeah.
Sir, my question is that while we do the modeling or docking, is it mandatory to validate our protein structure? If we are not validate our protein structure, then if directly do the modeling and docking, then our approach is correct or is the wrong way?
Yeah. Say, suppose you are docking a ligand here. So in this place, in this particular place, you are docking this ligand. Say you have a missing residues or some structural errors in this particular area. So then you don't need validation of the structure in docking. Okay. Then if you are docking in this particular residue, say in this particular residue where this particular residues where your active site lies, and these residues, if had any error, then you need to validate that structure so that these residues are correct, or every single atom of that residues are present in that particular region.
Okay, so for the validation, which tool we have to use for that purpose?
Yeah, for validation, so there are several types of errors. Sometimes what can happen is sometimes there must be a missing residues here. So for that you need to do a homology modeling. So you can use one software called Modeler. So also you can compare the amino acid sequence through a blast search and find the most likely amino acid which will be placed in that particular region. So after you have placed that amino acid in the particular region, sometimes what happens that amino acid coordinates might not be in the correct orientation. So for that, to obtain that correct orientation, you need to do some energy minimization using a force field. So for that, you can use this script, or you can use the Chimera, or you can use the Swiss PDB viewer, so where you can do some restraint minimization, and so that these coordinates of that particular amino acid residues which you have inserted are configured properly.
Okay, so may I know what's the threshold of the homology modeling?
Threshold means?
I mean to say, what's the range up to which range we can say that our model is good for the modeling?
Okay, it depends on several factors. Okay, during homology modeling, so you can see the Ramachandran plot, or there are some other parameters also. So which you have to analyze during homology modeling.
Yeah, that's why I'm asking that in the Ramachandran map, there is some sort of a threshold that if it is homology, then can we proceed for the docking?
Yeah, exact. For that threshold, I cannot tell you exactly what will be the threshold, but that depends on the modeling or accuracy of the model.
Okay.
Yeah.
But there are some sort of the threshold, because if we follow that rule, then we can go ahead for the docking. If the structure is there is some sort of a range, so that range vary from one lab to another lab. As I see, most of the people are taking the structure which is showing the more than 30% of modeling. Then they told that we can take the structure for the docking and modeling. And some people told that if the structure is showing up more than 40%, so exact data I don't know, but was the exact range, you can say.
Yeah, I'm also unaware of the exact range.
Okay.
Because every time I have used this, I try to find the X-ray crystallography structure which is most accurate, or where there is very less number of errors, or very less number of missing residues or missing side chains.
If the structure we have to take is the X-ray structure is not available, the NMR structure is available, so can we proceed the docking and modeling type of study?
Yeah, you can process with the study, but accuracy, what it depends. So if some residues are not present here, suppose, so you can also dock the compound in that place, but in experimental purposes, so that accuracy will not be optimum in that case, because that residue might play a crucial role in some interactions, say.
Okay.
Okay, so back to the discussion. So now here first I have removed the water, remove all the heteroatoms, and then I'm going to add the charges. So charges, add Coleman charge. So here we have added to the Coleman charge. Then I'm going to export this file into PDBQT. So for doing that, we have to import into grid. So first, after import, you will get to export as it is PDBQT. So I'm going to export it into our current docking directory. Here I'm going to name it a receptor. Say.
So, sir, may I know how many pockets in this protein molecule?
What?
How many pockets? How many pockets are in this
pockets?
molecule?
Okay.
Yeah.
7. Just wait for a minute. I will explain after I save this compound. Okay. So for binding pockets, you first have to manually analyze first. So if you find some information in the literature, so where their binding pockets are already mentioned, then you can take directly pockets from there. Or if it is not present there, so you can analyze here. So first you can analyze from the structure. Say if I first let me delete here. Hide the water here. And also the can say. Okay, then okay. So here first you can see for probable pockets first. Next you can use other softwares also. Say one another software available is ICM Pocket Finder. So you can use this software to find the pockets. But this uses a similar approach if you analyze visually, but uses this algorithm to find the most pocket-like places here. So main approach to find pockets is that you first download a structure containing structure complex with a particular inhibitor or first. So then you will get to know the pocket, and if you don't have that particular information, you can do a whole body docking. So if you suppose do a whole body docking in this place, so you will find some potential places where suppose if you select 9 binding both, so if you find some potential places in this protein structure where that particular compound will go and bind. So that is the second approach you can do the pocket. So in some proteins, say suppose if you see the structure of the alpha amylase, so there you can find you can see directly some pocket which will be distinctly visible if you see the mass structure.
So how many pockets in this protein molecule, sir?
It depends. It depends on the protein molecule. So there are some proteins which contains one active site only, or there are some other protein molecules which contains active sites along with some allosteric sites. So where some particular get bind or regulated activity of the protein. So it depends on the protein molecule, or this kind of informations can be retrieved from the literature.
Okay, if the protein is already having an inhibitor molecule, okay.
Yeah.
If we remove that inhibitor molecule and we have to insert our lead molecule, so is it mandatory that that my inhibitor molecule will bind to the already existing pocket which is already having the inhibitor molecule?
Yeah, that also depends, because some inhibitors are competitive inhibitors, or some inhibitors are allosteric inhibitors. So if there are some competitive inhibitors, then they will go and bind to that particular pocket before binding the substrate, say. Or there are some allosteric inhibitors, so which bind in the allosteric regions. And so it depends. So it depends how you design your how you want your drug to perform. So it go and bind to the allosteric site, or regulates the protein, or directly competitively bind to the place where already inhibitor is attached, or so that it can upregulate or downregulate its activity. Okay, any further questions? Okay.
One more thing, if we do the docking and then we do the energy minimization by the help of some Gromax or Amber or Charm, so is it mandatory that first of all we do the energy minimization, then go for the docking, and then we have to do the docking, then again do the energy minimization and simulation?
Okay, the energy minimization step is one crucial step. So first, suppose when we determine a extra crystallographic structure, so we have the coordinates of some heavy atoms or some particular major atoms. So suppose when we model the structure, suppose hydrogens cannot, some two hydrogens stay close, or some hydrogens or some other groups can stay close during the X-ray crystallographic structure determination. So these kind of problems can be solved by a small minimization step. So that step is called a restraint minimization, where you don't do a hard minimization. Suppose you don't do a 1000 picosecond minimization, or with maximum number of steepest descendant steps. So we do simply 50 or 20 steps, so that major which are easily correctable atoms, they got relaxed, and the protein go restrained. So on the other hand, when you do molecular dynamics, so there we have another system, solvent system, and a new kind of, suppose, say, force field. So we have to minimize the protein in such a way that that protein behave in that particular force field or with the water system. So for docking, we don't need that hard minimization which we do with Gromax or Amber.
Okay, if suppose my protein is the transmembrane protein.
Yeah.
So for the transmembrane protein, first we have to do the energy minimization, and if we do the energy minimization, then what's the role of the energy minimization and how it is significant to do the docking?
Okay, just let me select a receptor here and hide the surface, and let's show the sticks. Now say in this particular structure, say let's focus on this fragment. So here we don't have the hydrogen atoms. Okay, so because this structure comes generally from a X-ray crystallographic origin or any determination step. So suppose we have added the hydrogen atom, so it can get added like oriented like this or this or this. So hydrogen atom can be oriented in anywhere in this three-dimensional space. So how will you know that what will be the optimal position of that hydrogen atom, where that particular optimal position of the hydrogen atom in this three-dimensional step? So that kind of problem is solved by a restraint minimization step. Hello?
Yes, sir. Actually, my question was that I am having a transmembrane protein.
Yeah.
So in that case, is it mandatory that we do the energy minimization, then we do the docking, then again we do the insert the membrane, and then do the
for molecular dynamics, if you are talking about the molecular dynamics simulation, it is very important that you have to do the minimization of the protein in that membrane system, and then you perform your calculation or perform your simulation. But in case of, if you want to dock, do a simple docking or do a simple virtual screening, then you can skip this step. Or if you do a simple restraint minimization with, say, less than 10 or 20 or 50 steps, then it is better.
Okay.
Participants, if you have any question, please you can ask, because I have unmuted you all. Arup sir?
Sir, can we, sir?
Yes.
Can I ask a question?
Yes.
Yeah,
Yes.
sure.
Sir, can the dissociation constant be calculated by the by any of the software that by molecular docking when there is a molecular docking, can we calculate the dissociation constant or the association constant of the target molecule with the parent molecule?
Yeah, you cannot calculate it because that association or dissociation constant will be a simple experimental value. Okay, when you perform an enzyme kinetics in a particular say in a particular solvent system or in a wet lab experiment, then only you can calculate it. But in docking, you will only find that how the compound will bind there. So you will not get to know that it will associate or dissociate or what kind of regulatory activity it will do directly if you do simply docking. So you will suppose you have a particular place, you have a particular compound, and you can only determine how that compound will fit in that particular place.
Okay.
Okay.
And in case of non-competitive inhibition, is docking possible?
Yeah, if you know a particular site, suppose there it can bind, then you can do the docking in the particular site, and then you have to go for wet lab experiments so that if inhibition occurs or not through by some particular enzyme assays to determine if inhibition is becoming non-competitive or so.
But in case of any publication to any research paper, this has to be validated through the wet lab experiments, right?
Yeah, it has to be validated. So majority of the thing is to be validated through wet lab experiment. But with docking or using in silico approach, you have to you can minimize the possibilities. Okay, so you can use the optimal compounds for a wet lab experiment, say.
So if anyone published the paper, what is be the weightage of that paper with the without doing the wet lab experiment?
Yeah, nowadays if you want to publish, suppose a good impact, say, or 3 or 4 impact, say on an average 3 or 4 impact, so it needs to be followed by wet lab experiment.
Okay, thank you, sir. Thank you.
Any other question?
Sir, can we find the dipole moment?
Okay, dipole moment. Yeah, technically you can calculate dipole moment because there are lots of quantum chemical calculations tools, say ORCA or Gaussian. So there you can calculate the dipole moment. So quantum chemical, but it is very time consuming because it takes very huge number of calculations to do so.
No. Is there any tool or software by which we can?
Yeah, you can. One of the tool I know. I think in Gaussian you can do that. Gaussian, or there are other tools also. Say ORCA. One I know is ORCA. Yeah. So this tool also you can use the quantum chemical calculations.
Okay, thank you.
Any other questions? Okay, now let's move forward. So I have saved this structure PDB-QT. Now, next I have to select a region where this particular molecule will get docked. So I will quickly show that particular region by drawing a grid box. So here I have defined a box. It's a simple 3-dimensional selection box. So we can move this box by this offset and set the size of this box here. So one of the most important thing is that this spacing angstrom value. So for Autodoc Vina, it uses 1 angstrom by default. So if you put 0.375 in its spacing angstrom, so your docking experiment will fail or it will give erroneous results. So you must put this value to 1. But Autodoc, you can assign the spacing parameter separately, but in Autodoc Vina, there is no provision for that. So you have to use 1 spacing angstrom, then adjust the parameters you're requiring. So now I have grid parameters already defined, and so which I have calculated earlier with the ligand binding sites. So first I am going to select that from there. So receptors. So these are grid dimensions. So see here I have added spacing 1 angstrom and 32, 36, 30. So quickly, I'm going to show where it is, where I'm putting the selection was 32. $36. 30. The next second offsets is 4.449, 4.637. Okay, so I have selected this particular place. So here our docking activity will take place. So I have selected this place based on the inhibitor compound which was already attached there. So now you can save the grid dimension file as output.
So this is a chain A or chain B?
It has only one chain. So chain B was the inhibitor compound. So I have removed the chain B. So only one chain is present here. Okay, now I have the structure and I have the grid file. Say, so I have created a configuration file. So I have added receptors in the name by 7bqi final.pdbqt, center x, center y. So these are variables from the grid. And then I have specified the output file name and then specified the log file name, then exhaustiveness value of 8. So for more accuracy, you can increase it to say 20 or 10. So this is the average value I have seen in publications. And index number of modes 3. So this will output 3 particular docking poses. So I'm going to simply copy this file conf and then final receptor to the to a test folder, say. Okay, these two files we need for docking, and then we have lichens pdbqt here. So first I'm going to demonstrate a simple docking using command line, then I will do it in Jupyter Notebook. Now say I'm going to create a folder called test. It is 90 compound. Okay, so I have the conf. So I'm going to use B now. Lichen is say 90.pdbqt. And I'm going to output of a conf file. Okay, so I'm not sure about this lichen tag. Let me see the help. Just lichen and config. Okay, so in this output, see, I have modified the output format in the source code. So it prints a standard log format, which is acceptable by various cloud computing applications. So now here it is starting a docking task. So it will take a couple of seconds, and then after task will complete, I will get the binding affinities. So let's see how it is done in Jupyter Notebook. Till here we have written the ligand file and export it. Next step is to virtual screening. So here I've imported the required libraries, mostly OS, because we are doing lots of path or directory creation or operating system related operations. And then we have listed the ligands here. And then we have copied the required file. So required file will be the receptor file and then receptor file and the conf file. So what I have done in this step is I have created, we go to the old docking folder, docking backup. So here I have created a runs folder, so where I have added a receptor folder programmatically. Then I have created single folders for every ligand. Then within the every ligand, I have copied these two files from the receptor folder. Then after that, I have added the PDBQT ligand file, then perform the docking using this step. So here I have run the docking job with this command, and then I have got this log. Then I have stored this log in a particular text file so that all logs can be all logs are summarized into a single text file, and I can analyze the text file separately. So finally, I have saved the text file. So if now let's see the docking results for a single file. So here we can see that we have three modes as we mentioned in the conf file. So we have binding affinity minus 8.5. So we have same binding affinity 8.4. So here we can analyze both the modes, but further it will depend on the interactions present. So if first mode have more hydrogen bonds than second mode, then we will prefer to take the first mode because hydrogen bonds are more strong than other type of non-covalent interactions, say pi-pi interactions or pi-sigma bonds or other van der Waals interactions. So based on that, we can select the first mode, but it is not always true that this first mode will be optimum. Sometimes in experimental conditions, the second mode can also be valid. But we have the possibilities here. So this notebook simply do that job I have as mentioned. Next, I will try to analyze the docking results with another notebooks. And again, these notebooks are available on my GitHub repo, so you can download all the data there and play with these notebooks.
Yes, sir. My question is that if we are having a library of a different type of ligand molecule, then within a command, we can execute all the molecules.
Yeah, you can execute all the molecules, but not using this conventional Autodoc Vina command. So you will need another supporting program, so as I have done here. Say here I have in this step. Yeah, in this particular step, I have docked 300 compounds to this particular COVID-19 3CL-like receptor. So in this, so see here you have the log for 300 molecules. We are also developing one library which is based on Autodoc Vina, which will in future allow to dock multiple molecules in Autodoc Vina. So it is in the early stage of completion.
Actually, I'm having 10,000 molecules in my library.
Yeah.
That is the drug for repurposing purpose. So how I can execute whole molecule?
Okay, for you can execute a 10,000 molecules. So in this tutorial I have shown, so here you can download these notebooks from my GitHub repository. And if you have some programmer who is good at Python, then you can see the codes and do the docking for 10,000 molecules. So following all these steps only, you can do that. So I have mentioned all the steps in these two particular notebooks.
Okay.
Again, we have also another approach also. Suppose one thing I forgot to mention during ligand preparation step. So if we see the ligand preparation, suppose we have, say we have 1000 of known compounds, say they are flavonoids, they may be phenolics or anything else. Now we can even predict their tautomeric forms, say using techniques such as R group decomposition, or we can see their likeliness in PubChem, or we can also manually attach several groups so that the ligand dataset can be increased. So that is one another approach of virtual screening. So now after it is virtual screening of 300 molecules completed, so I have saved this text file with 300 logs from 300 compounds. Then in the next step, docking result analysis. So first we have imported pandas, and the docking output file I have imported here. So now I have done a simple lexical analysis to parse this log file. So see here, if in the earlier tab, okay. Okay, in the earlier tab, so I have specified a specific format to generate these logs. So see, it always start with a start run. Yeah, start task program, and then it ends with this plus signs. Again, from this task. Okay, yeah, and this with this separator, and within this separator we have a tag called result, and within this result we have a table. So in this table we have this table is in the form of markdown. So there are a lot of markdown parsers in the throughout the code throughout various code repositories. So you can use a simple parser or write one for you. Then you can parse these results. So in the end, I have collected all the ligand IDs. Yeah, I have collected all the results in a dictionary with ligand ID, directory path, and the table. And then I have converted this markdown table into a data frame. So which looks like this. So in this data frame, we have different modes for a ligand. So for ligand ID, say for ligand ID 0, we have 3 modes and different affinities. Similarly for our ligand ID 3, similarly for ligand ID 1, 2, like we have around 900 compounds here, 900 poses here. So from these poses, I have now selected first mode for this demonstration purpose and converted the data type for our ligand ID and affinity to a float or numeric type. So then we have analyzed this, and I have next joined the ligand metadata there, with this ligand ID. So suppose for now we can clearly see that suppose is malvinidin 3O comeral glucoside, we can get the binding affinity E2 -9. Okay, this is the compound name in this column, and yeah, here this is the compound name, and this is the binding name. So in our data set, we have find that theophlavin 3,3-O dicalate has highest affinity. So this is a compound which I believe present in tea versus tea-like beverages. Next one, cyanidin 3O glucoside is anthocyanin. So this has minus 9.7 affinity towards that. So I have roughly gotten results of which flavonoid should be optimal, and see these are performance, more performance than the other are standard drugs, so which are listed down below in this data frame. So now next we need to do is that, so after we have docked the compound, so after we have docked the compound, say with 90.pdbqt I have docked, then a file is generated called result with a single log. This log is a single file for this particular job, and with this result we have the different pauses. So it contains, so this is a molecule which is folded in such a way that it fits the receptor, but it is not visible now. After we add the receptor here, we'll get to see. So let's quickly open the receptor here. So if we open the receptor, since these both share same coordinate values, same coordinate system, so we got to see that this compound got bounded here in this pocket. So again, we during the docking process, we have selected three pauses, so we can navigate to next pause like this or like this. So a previous pause or like this, three pauses. So n number of pauses you can add, and like that you can verify their binding modes in that particular pocket. So next, what we need to do is, after this binding mode is complete, we need to see the different interactions. What are the different interactions happening between the atoms from the amino acid residues to this binding molecule? So next, for that, first we need to split the results. So that is also programmatically we can do that by using a simple command called Vina split, which is distributed with Autodoc Vina. So we will split that file into different modes, which will be stored under several folder called pauses. So I'm going to show you that the previous folder. Okay, docking backup runs to say, therefore for compound 90. Okay, I have it here. Let's say notebooks. Yeah, so for compound 90, we have created a another folder here called pauses using this block of command. Then I have run a sub process in this Python shell, in this kernel with executable called Vina split with input of this particular result file with ligand pause, prefix pause underscore, and flexible residues pause, prefix chain underscore. So this will split that particular file into the number into the n number of modes there, into three separate files. So then we have got a real-time output from the sub process and printed it below. So now, which has resulted that a new folder with this particular pauses. So these two files I have created separately, so ignore that for now. So these three, pause1.pdbqt, pause2.pdbqt, and pause3.pdbqt are created separately after execution of this command. Next, in this current working directory, I have selected this the first three ligands with highest affinity, and I have processed that. But in real case scenario, you need to process all the ligands and store it in your local dataset, so that this can be used further in machine learning or any other task, computational task. So now I have selected all the ligands within this list, and then I have created a complex of that file, a complex of that pause with the receptor. So what the end result is that I have created this complex as for pause 1. Similarly, you can create a pause for all complex for all three pauses, but in this tutorial I have created for pause 1. And so if we open this complex in the PyMol, So see, here we have the ligand and the protein file here. So here, this is the final complex we want to further analyze the interaction. So for analyzing the interactions, we can use one library, which is open source, called PLIP Interaction Profiler. Interaction. Okay. So this is the library we can use. It has a web interface and a GitHub repo also. From the GitHub repo, you can download this whole set of code, which is Python based, and you can extend this library and write your own code to create a complex for multiple to generate the parameters as interaction parameters for large number of files. So what it will give, so let me show you. If we go to docking backup and if we go to notebooks, it is complex, and let's say analysis complex. So it has given us this structure, but this is not important. You can generate this structure on your own by using Pymon. But most important things are this data table. So we have the information of hydrophobic interactions with within what distance the hydrophobic interactions are taking place, and we have also the hydrogen bonds. So we have several different hydrogen bonds with which ranges from around 4 to 2 angstroms here. So a whole lot of hydrogen bonds here, and also we will have other interactions, but in this UI we'll get to see on the hydrophobic interaction and hydrogen bonds. But if you do the if you download the command line version and do analyze your own, then you can get another results describing various properties, including their molecular descriptors in XML or RST format. Also, you can modify it to get a JSON format so that it is more convenient to process in other applications. So in my case, in my several experiments, what I have done is I have simply extracted this information and put that information into the MongoDB instance so that this information can be analyzed later. So now after we have the information, so a basic part of partial screening complex. Now we have a rough idea of the flavonoid compounds which have the inhibitory activities of that for that particular SARS-CoV-2 protein. Now next step is to verify. Next step is to verify our hypothesis that this particular compound, say this whole first 10 compounds, can inhibit this SARS-CoV-2 protein. So now we will go to the wet lab and perform another assay. One of the most important assay for this protease enzyme inhibition is FRET assay, which is fluorescence resonance energy transfer. So we check their enzyme activity based on fluorescence emission and excitation. So after that, we can conclude the inhibitory activities. But with this step, drug design is not over. But with this step, there are several other challenges for drug design. Say now we know that this molecule can possibly inhibit this particular protein. Now, how will you know that this particular molecule will behave in your body, or whether it will be absorbed in your body, or whether it will be eliminated from your body after absorption, or whether it will show some toxicity in your body? Say it may have some side effects to your liver or renal toxicity or any other toxicity. Say it might, if the protein is targeted to cross the blood-brain barrier. So how will you know that this compound will be distributed within the blood-brain barrier? So this kind of calculations can also be done. This kind of not calculations, predictions can also be done in the using the machine learning approach or using their virtual approach. So initially we can use one simple tool that is ADME. Okay. So let me first see the smiles of the compound.
Sir.
Yeah. Hello. partial screening ligand. Okay. Yeah, let me quickly grab the smile of this particular row. So we have TF merged.
Sir, I'm having one question. Can I ask right now?
Okay. You can ask.
If we are having a molecules like 2 or 3 molecules is showing the same affinity, then what's the criteria to shortlist that this one is the one of the best ligand molecule?
Okay, if it has same affinity, then you have to see the interactions. So as I have said that if there is more number of hydrogen bonding than other non-covalent interaction, then this compound must be more tightly fit in that pocket than other non-covalent interaction. And after that also you have to do some molecular dynamic simulation. So in the molecular dynamic simulation, you can see the RMSD. Say for if you run the molecular dynamic simulation for 100 nanosecond, then within the 100 nanosecond, you can calculate that whether this compound get bounded to that site or it get released it after 100 nanoseconds. So like that, you can also calculate MMPBSA based energy calculations, or also you can calculate density functional theory based this orbital energy calculations, and so that more so that you can get a more confidence on the pause, and say whether that will be stable or not. So first, say part is not defined. So I'm quickly going to copy this name and search in PubChem. Okay, I'm gonna copy the smile, canonical smile. Oh, I have pasted the smiles here and run. It has already some machine learning based models which are already deployed. So we can paste one smile here, and then it gives us the various parameters, predicted parameters. So we have Lipinski parameters here. Lipinski parameters here, and we have absorption parameters and distribution parameters here. Also, we have some chemoinformatic parameters. So like that, we can analyze the ADME properties. Also for toxicity, we can browse another server called ADMET or ADMET server. There are many servers. And again, you can also write these models of your own. So there are one database called CHEMBL, CHEMBL. So let me show you quickly. So this is a database called CHEMBL. So here I have already browsed a property for this particular protein, which is COVID-19 3CL protein. So if we see here, so we have found that there are 8,702 records of its inhibition, or with IC50 values we have 170 records. So we can directly get this fetch this data, and we can generate one regression based model, say, so that we can predict the inhibition also. Okay. With this, I would like to conclude today's lecture. Actually, there was a lot more, but due to time constraint, I am concluding in this with this. Is there any questions?
Thank you so much, sir, for the wonderful and informative hands-on session. So I would like to request if any participant has any query, please ask. Okay. Saikia, sir?
Yeah.
Yeah. Anything else if you want to demonstrate, then you can, or you can conclude. So.
Okay, for today, I think I should conclude because it will take another very long time because I have some machine learning models.
Yes, sir. Yes, sir. So I would like to request Professor Rajna Patnaik, ma'am. So kindly facilitate our invited speaker.
Dr. Saikia, very well presented, and really we are thankful to you that you have accepted our invitation and presented this very informative lecture. And,
thank you, ma'am.
Please accept our e-memento. This is for your honor from our university.
Thank you.
Thank you.
Thank you. Thank you so much, sir, for accepting our invitation and providing such a wonderful, informative session. I hope that participant learn a lot. If any participant want to collaborate with you, they can easily find the email ID and the details on the Saikia website. Yeah,
I'm available anytime.
Yes. Thank you all. Thank you so much, sir. Okay. Thank you, sir. So, dear participant, for tomorrow we have a great speaker, and we have lecture from the various renowned speaker like Ballabha Devi sir, who is lead data scientist in the Tiger Analytics. They will make you learn about the application of machine learning in biology. Professor Lalitam Agrawal sir from the IMS BHU, Varanasi. They will learn. They will make you learn about the radiation therapy and use of the computer. We have Dr. Munendra Singh from Johns Hopkins University. They are expertise. He is expertise in the deep learning and medical image processing. We have again Dr. Sudeep Paul sir. They will talk about the on the cranial motor disorder detection system, and we have also Atul Kumar from Lund University, Sweden, and they will make learn about the hands-on session on computational genomics and neurodegenerative diseases. So I cordially invite you all to kindly join the session at 9 o'clock. Thank you all, and wishing you a great learning ahead. Thank you. Thank you so much.
Thank you very much, sir. My request is that can you please provide the study material?
Yes, sir. Yes, sir. Sir, all session has been recorded, and I have posted the link of the same in the WhatsApp group. They will be permanently. You can anytime visit the Guwahati University website or the link prescribed by me, and you can open the video. The video always remain on the Guwahati University YouTube channel.
Is there any G drive so that we can download?
Sir, you can easily download, sir, by the YouTube link.
Okay.
Yes, sir. And I will also provide the PPTs from the speakers. I have already requested all the previous invited speaker to send their PPT. Someone has sent their PPT, someone has not responded. So as when I received all the PPT, I will compile it and send to you all the participants.
So can you provide the coding, today's coding, whatever sir is telling?
Sir, I have sent the link.
Okay.
Yes, sir,
you can easily download from the that link.
It's there. It's there. I have seen that. It's in our WhatsApp group.
Yes, sir. Yes, sir. Okay. Thank you all. And please join at 9 o'clock.
Sure, sir.
Okay. Thank you. Thank you so much, sir. Sorry.